It's also hard for studying because you need to rebuild the system somehow in the lab.
Yeah.
Or if you cannot make in situ studies, or like do the studies in this ecosystem.
It's super hard because you need to build incubation chambers and place the incubation chambers here, and then place inside the incubation chamber some food that you can trace and collect water before and after the incubation. So it's super challenging.
Yeah. For my bachelor thesis, I did not work myself so much on nutrient cycling in coral reefs, but a little bit. And the PhD student that I worked together with, he was also using incubation chambers in tropical coral reefs. It's also challenging, but it's even challenging, near to impossible yet to do that in the deep sea.
Exactly.
And find out about the nutrient cycling in here.
If it's challenging at 20 meters, imagine at 3,000.
Exactly.
So yeah, this is the community we have. It's kind of repeating. So these are Endorids, Emisidius, and Asconema, Megatriala, and then the little crust. I mean, in the little crust is where the real diversity is. So that rock will give us a lot of work tonight.
Yay! Oh, the next shift.
Yeah.
You go to sleep. Paco can do it.
Paco.
Yeah, so we're organized in shifts here with the research teams. So we have 12-hour shifts. We are working from 2 in the night to 2 in the afternoon, and then the other shift takes over. That's Paco from the sponge team, and then also Anna-Helene and Jenny from the macrofauna team that take care basically of all the other critters we come across. And next to Anna-Helene, which is on our shift, who will come with Sergi in about an hour, I guess, to take over from us. Yeah, so sometimes it's like yesterday we had a dive until, I guess it was around lunchtime. I already keep being a bit confused about the timings, but then we had the dive, we were in the rough shack, our team, and the next shift.
What is?
Oh, what's this? Is it a sponge?
Yes, looks like.
It's
I'm
a
not
crazy
sure.
sponge tube.
I'm not sure, but what was that?
Now we're going up the hill.
I'm going too fast, just
No, that's point one.
well, we start here, and this is the first transit. I don't know.
No, I mean the notes. I mean the
I don't know. We don't have that information now because the inertial navigation system is not giving us that information.
Maybe you can slow down a little bit now that it's so crowded.
Okay. So
Just a little bit.
Wow.
She started. Yeah.
Oh, on its way to eat the sponge.
Yeah, the other team asked us to collect that sea star. So
It looks like a little turtle.
I guess that this is not a good terrain to stop. I think that was
Yeah.
an island. Ask for it.
Do you want to try to start?
Can you?
Where is it?
There.
We passed.
Maybe go.
We passed.
Oh, we go on for now. We leave something for the others as well.
Okay, we go. Go on. What's this tube? That's a cycle. Now that I know what was that. That's a cycle carriage.
We saw those yesterday as well.
Reduce to point one.
I think. Thank you. I think. Let's see if it appears again.
The slope increased.
Let's see.
Oh, there's a sea star. Ooh, a Dumbo.
Could you stop?
Oh, thanks for spotting. I was too distracted.
Thank you.
Oh, wow. And hey. Oh.
Lasers off, please.
Yes.
Shit. Sorry. Could we turn the lasers off?
Wow. And also fish swimming by. Oh, it's beautiful with these flappers. So these, these fins, flappers that the Dumbo octopus has on its side,
Stop, please.
they
Stop.
they led to the name Dumbo because of Dumbo the elephant. It's not the ears of the Dumbo octopus, but it looks a little bit like it, and it's flying with it.
Look at that.
Oh wow! Ah. Such a beauty. Showing off for us.
Turn on the immersive camera and send it out, please.
Can we work with the exposure somehow? Because it's so
It won't do much.
too bad.
Because it goes with the gain, and I
Yeah.
was talking with Jason yesterday.
Yeah, not too bad. So we unfortunately cannot work on the exposure and
That's fully out.
That's 5:15.
It's more like
Powered on.
so we now also put our immersive camera out
Yeah.
that can give us a 180 view. So that was the thing you just saw further down. What a beautiful octopus.
That's nice.
Oh, we caused a little bit of a sandy sandstone for for Dumbo, but
Okay. Shall we keep going?
yes, let's keep going.
This guy here.
Yeah. What is it? A snotty fish? No, snailfish maybe? Yeah.
Okay. We keep moving.
Yeah, we leave Dumbo in peace. And let all the dust settle again as well. Which will be relatively quickly. Now, the last glance of Dumbo. Bye bye. Not really centered. No, almost, yeah.
Crush it.
So we want to get to the highest point of this.
So we're continuing now up
Guys,
the
that was
hill.
beautiful.
Probably, thank you. Turn the sextant back in. Probably we leave it out.
Sounds good. Yeah, to leave it out for now.
Okay.
And then when we sample, we take it in. Because we cannot have the immersive camera out at the same time as we
Oh, yeah, those
as we
the laser
sample,
back. Oh, yeah.
because of the bio boxes that will be
Lasers back.
drawn in and put out.
Places are okay. Yeah.
So we always have to be mindful of where the equipment is at the moment.
In the beginning here.
Oh, a lot of these sponges again, of the little ones as well, on all the rocks.
So here you can see that the community is a little bit different. We don't have that many Lisa and Dorits.
Yeah.
We would restrict a bit.
And we don't have that many at Sinelli there.
Point one maybe.
Good ROV, if you continue movement please, bearing 030 at point 1 continuous.
030.1 continuous, okay?
Okay, thank you.
In this frog you can see
The little small.
that one, yeah,
spotted.
for instance.
Yeah.
That one.
And up there is.
That's Conema.
Yeah, and next to it is that that's baby Oscar. No, is it the new species or baby sponge?
It look like something different to me. I don't know.
And what about these crusts? Are they budding? No. It's just their structure.
Hard to say. So these are species that we haven't collected yet. So. Yeah.
So many species. How many sponge?
Look at that.
Ah,
What is that?
carnivore.
It's carnivore sponge. Yeah. There you go. Carnivore sponge. This wall is completely covered in anemone over there.
Yeah. So not all sponges are just peacefully filtering out of the water. Some are catching
vicious
fishes,
predators.
yeah, predators with their sticky bubbles. Or how do they do it?
Well, yeah, they have this mucus and the little critters get attached, and then they mobilize the cells, and then they do like an external digestion.
They poison them.
Not really. They
No?
just catch them and then they do digestion. So yeah, the crustacean get attached to it, and then it cannot go away, and then it's digested. Yeah.
So a little bit like a Venus flytrap,
Yeah.
unless they don't have the thing too close with some carnivorous plants on land.
Look at that. What is that? Fancy. What is that? What is that?
It has these big pores like the.
What is
No,
that?
it's different.
What
It
is
looks like a
that?
brain structure.
Yeah.
What?
Look, the new species over there,
Yeah,
and then
but
this
much
thing
bigger
that I
than
have
before.
no idea what is that. No idea.
Wow. It makes crazy structures.
The rock is too large to collect.
Yeah, unfortunately. But these new undescribed species here, they look much bigger than before, the little ones we
This
saw.
one is more.
Yeah.
I mean, you have to be careful because here there are many things that all look alike. If that one appears again, maybe we can use the vacuum to just take a little bit of the grass with the vacuum. What do you think about that?
Yeah, when it's so on a rock, it
No,
would be quite clean.
Not take the rock, but just take
Yeah,
a piece
yeah.
with the vacuum. Maybe will come. Maybe.
Could be an option, yeah. Let's see what else comes up. Oh, there's a big shrimp.
Yeah.
Can you zoom in on that one? No, it didn't like the attention. Oh, it wants to show off. Oh, so cool to see it swimming with all its legs. Walking on air or on water.
It's a big rock.
Yeah, with fans and
Could we stop and have a look at this rock?
Because
No? Okay.
yeah, and just to understand the shape.
I just want to have a nice view because I think it's pretty. You see?
yeah,
This
with
rock.
all the crust. Ooh, it's really pretty.
Look at that.
So many different species on there. So yeah, the hard substrate is really popular. Also more worms on this, or worm structures. Oops, that was too much pressure from our thrust, from our propellers for that one. If they fly around and they settle somewhere else, then they can just attach to it
Sure,
and
but
regrow
just-
and settle somewhere else.
just if the stem stays, an individual will grow. So
Yeah. Sponges are pretty hardcore.
yeah, they are very
They
adaptable.
take everything. You can see from the currents here, I guess, that from how the sediment is on the rocks, that it's coming
Yes,
from
the right.
a certain direction, and then
You want to keep
Yeah,
going,
but
or you want to keep focus on this rock?
Nah, we can keep moving. Thank you, Antonio.
so all of these, presumably, was new species among them.
Yeah, among them. Yeah, just that there are two that are very similar,
Mm-hmm.
and it's a little bit tricky to
really.
tell from this distance,
Yep.
you know, with the movement and all that.
Where
This
are we
is-
on our way up the hill? Still going further up, right?
Yeah, I guess that we are about to get the summit.
Yeah, they're really cool structures from the volcanic activity.
Mm.
Big Asconema there. Oh, a big anemone as well.
Well,
This one.
oh yeah. So that's, look at that. It's super beautiful.
Ooh, and this Asconema is going in that direction there.
You can see how much sea snow is in there.
Mm-hmm. So the organic matter that is flowing in the water column. Also marine snow, because it's, looks a little bit like snow, but it's really, important part of the nutrient cycling within the ocean as well.
Sure, that's formed on the water column, and you know it's a gathering of organic matter and bacteria, and then it's falling from the very surface.
It's an important energy source also for the deep sea.
Yes.
Because we don't have any plants in the deep sea. There's no photosynthesis, of course, because there's no sunlight, so there's no generation of energy through photosynthesis in the deep sea possible. But of course, nature is creative, and there's another form of energy creation. It's called not photo but chemosynthesis. So there are bacteria, organisms that can create energy from chemical turnovers present at hydrothermal vent areas. And that's another way of generating energy in the deep sea. But also this marine snow is very important for getting nutrients down
That's
there.
the one that we saw before. That we said I would use the slurp. This one.
To me it looks even like do you see at the outcrops it has this bluish color that looks similar to the other encrusting, as if this was in a further stadium of it or so. Does that make any sense?
Say it again.
Because it has like this blue surrounding that is similar to me like the one below, so it almost seemed as if the white one grew out of it.
Yes. But you see that the texture is different, the amount of holes and then the
Yeah,
color.
but I just thought, is it the same? Is it growing on top of each other? Is one like growing further than the other,
I
and
think
just
it's-
turning white later on.
I think it's two different species, but the one that is in the very top has more tissue on the center, and that's why you see the difference in the color. So this is just more bumpy on the center
And
of the
when
sponge.
we go further up, it even gets more orangey. But
Well,
that's again a different species.
That's sediment, I think.
Yeah,
I think. Is it the
it's also from below.
Or it's complicated? If it's complicated, don't worry. Don't waste time. You think we could try to
Motion
back
try
this
lab.
one?
Try to be a bit more slower with the pen and tilt.
Yeah.
Yeah, go with that.
But we're settling down for now, right? And then we first have a look and could be possible, yeah. Oh. And just like that, the view has cleared again.
Great.
So we're gonna zoom in on this these interesting sponges.
Okay, now let's take our time.
Yeah.
One interesting thing to me was on our left we have some rocks with those orange colors. You noticed?
On the left
On the
side,
left side.
yeah, there seem to be some mineral structures.
No, we saw a lot of those rocks in hydrothermal fluids because
Yeah.
that's alteration with the temperature, so they modify the rock. And
It's interesting this thing that has on the top.
Yeah, it's not. It's the coloration, this orange. Like it has a little bit of sediment, but
Maybe because it has moss
yeah.
to protect
Yeah.
the sponge from the sediment.
So maybe it incorporated some of the sediment.
Exactly, that's what I think.
And down here,
That's an Assyrian, colonial Assyrian.
so that's
That's not
not
a
a,
sponge.
not a sponge, but a tunicate.
Tunicate.
Can you-
What I don't know
Oh,
is this
hey.
crustacean.
English, yes. So just be going on the orbit this morning.
Can you zoom in a little bit more?
This is
No.
maximum.
I didn't look in yet.
Sorry.
Because it has very long legs.
Yeah, because it's
And could you also zoom out a lot, even though this rock is very interesting, but we saw also interesting,
Yeah.
like the metallic crusts.
Yes, I
So
will
the-
go there. Just let
yeah.
me finish
Yeah.
with the rocks so
Yeah.
I don't have to come back again.
bridge RV. Can we stop
What's
the
this
movement
fuzzy
for now?
ball down there?
That's
stop.
an Alicona.
So we're now zooming in on this rock to have a really close look what is all growing on there and get an overview of it. We have also crinoids, these feather stars, and to the left we have different mineral structures or different forms of rocks, metallic crusts maybe, that are known for areas of hydrothermal vents as well. So we are at an inactive vent site, and see the colorations and the shadings of metallic elements here. But at the same time, we see a lot of life right next to it. So in case this would just be a lot of
Beautiful.
beautiful life all around it.
Yeah, super beautiful. And there are like gravels.
What did you see?
I think that this has like a kind of a different texture than the rest.
Yeah, I was thinking about if, but there doesn't seem to be, if that white part would be any bacterial mat that sometimes grows on
You
the
think? Okay.
fluid, but doesn't look like on that image.
Yeah. But you see it?
Yeah, it
It's
seems
kind of like two
all-
layers. The whitish and the reddish.
And it's quite covered with sediment as well, in some areas at least. Can you zoom out again? Yeah, so we have a high community also of species that we don't know yet here on the, like this sponge that we were just looking at,
So,
and also some of the little sponges we've seen before and we've collected to find out what they are. And we see a lot of these Lysodendrix species as well. Lysodendrix
like those green ones.
specimens. Yeah, the feather stars. Ah, yeah, you can really see the stem nicely here, like little palm trees. Ah, just
Nice,
swaying
yeah.
in the current.
Beautiful.
Yeah.
See, you can see them.
Oh. And behind it is also a crinoid sitting on a Lysodendrix.
Works with different colors. I like it. You see
See, all the sea snow.
Yeah, all the marine snow coming down.
all those rocks have
Happy
different
feeding.
colors.
Yeah,
This-
also.
Well, I'm not a geologist, but normally they say that this means that there is any kind of heat that made this alteration in rocks.
Yeah.
This kind of that we're seeing right now.
Yeah, so we probably see the remnants here of the volcanic activity of this active vent site that it once was, and now it turned into an inactive one. But yeah, we see the remnants of this activity, and we see a lot of life all around it, and a lot of species also that we don't know yet. Or some that we know, but that we don't fully understand yet, because there's still so much to discover here.
And many species that we know because they have been collected only once, maybe very far away from here, and then we find it here. Like, for example, yesterday, Paco Cárdenas identified a species that we collected some days ago that was found next to Greenland, but on the other side, on the strait, not on the Denmark Strait, on the other strait, which I don't remember the name now.
But it was so on the other side that now we have the first specimen from somewhere else, and we know, oh, it actually distributes.
Now we can
all over.
re-describe the species, we can say something about its ecology and about its geographical distribution, and step by step. Antonio, do you think that we can try to scoop this thing? To search, to search.
Try, yeah.
It's very soft,
Yeah.
so we don't want it to be nice and in one piece. We just, if it breaks, it's okay.
And just a little piece would be enough.
A little piece would be enough. Yeah.
So maybe from this side or so, try to.
It
Leave it
would
like
be.
this inside.
I would like to get some of the reddish.
Okay, we give a try.
But do you know what I mean? That down here it's more bluish. Here is also more bluish, and then it turns whiter, and then it even turns more orange the more it grows.
Yeah.
And you can also still see that.
Yes.
Yeah.
So this is a different species for sure. But I think that this is a colony. So I think that there are two species and two individuals. But I think that the large one on the borders is thinner, and that's why the color is slightly different. And then on the upper side, it has this kind of mucus that probably protects the sponge from the sediment, and at the same time it catches and incorporates the sediment. But it's just a guess.
Sounds legit.
If now we can take a piece, then we can check that on the lab and see what's going on.
And from the supposedly species 2 that is sitting on species 1 would be really quite cheeky to just cover all of the other specimen.
Could be as well, you know, sometimes they just fight for the space and they grow one on top of the
Taking
other.
away all of the surface from the other.
So this is cast 5.
So we have a slurp also for sampling that you can see on the left side, and we will try to get a piece of these species that we don't know yet. As Julio just described as well, to get a better understanding about what is it, how is it formed, how does it grow, and what are its characteristics.
We leave the lizard endurance for the next team, or we collect him now?
We can leave it.
Yeah.
Next team?
Yeah, we have like 5 or so already, right?
Yes, okay.
And we have a lot of time left. So,
What chamber do you want?
One.
Chamber 1.
Yeah,
You ready or?
I'm fine. Yeah.
so with this we can create a suction flow into different compartments. That's fine, right?
That's great.
That's enough. Yeah.
Enough.
Yeah. Thank you.
Okay, I fell off.
We have different
Thank you very
compartments.
much.
Yeah,
Great
thanks.
job.
We have different compartments in the back where the hose is leading to. Yeah, and in one is now these new species, or these species that we will have a closer look at and find out if they are indeed undescribed yet. Yeah, and yeah, the rest of that encrusting sponge will just grow back on that.
For sure, yeah. For sure. Yeah, that was a very precise work.
Yeah.
Okay, we can keep moving. Yeah, thank you very much. Fancy we keep moving?
Just 1 second. Could you turn the lasers off, maybe? And then we have just a few seconds of just this image as well. Thank you. It's really cool. Maybe even get the immersive camera out for a view.
Right,
Yeah?
yes.
Yes.
It's still out actually.
Sorry?
It's still out. The camera's still out.
Yes. Because we are perfect. Yes. Yeah. True. We didn't need to put it in because we used the slope.
No.
Yeah. Oh, perfect then. No bother. Okay, then I think we're good to continue. Turn the lasers back on and go on transect. Thank you.
Right, lasers are on.
Very interesting site here.
Yes.
Oh,
I guess that this is the very top of this little hill.
yeah. So maybe formerly this was, there were also chimneys here. So,
Maybe.
so taking off, continuing transit.
Yeah, continue transit 547. This is 7327. This is the summit.
And we have bigger sponges here.
Yeah, more food, more current.
More food.
Plenty of these arendorids.
Yes.
That's a super successful species.
Yeah.
See again the stem.
Oh yeah. And looks if there are anemones on it?
Yeah.
Or crinoids. Yeah.
Something's going on there.
It looks so weird. Also with the little like eyes on top of it.
Look at that. Look at that habitat. Look at this habitat. This is super nice, super interesting, super unknown.
And these different fan-shaped sponges, are they all? Some of them are different to each other as well.
Yeah, but I think that those are the ones that we collect at the beginning, and then there is some Emigilius, some Rosselli there.
I was wondering what's sitting on top of those, of the brown tube. Yeah, no, it's too much to see everywhere.
Yeah, I'm trying not to leave that.
Yeah, yeah.
Look at that.
Ooh.
Ah, that's a budding thing.
Oh, so it's cloning. Reproduction time for this one.
It's very large, yeah.
Yeah. So it's creating a lot of little sponges here. Oh, videotape change in 10 minutes.
Yes. I don't know which species it is because it's confusing me. Looks like that's inelida, but it's not. So it could be Stelladorids. You remember we collect something called Stelladorids at the some days ago on the ROV2. But that one was orange and this is white. So I don't know.
Messing with your head, the sponges.
Yes. Yeah, we can move on. See.
Oh, there's really like a street.
They're all facing the current.
Yeah.
You see all the fan-shaped ones. They are oriented the same way.
Yeah. Yeah, we have like two ripples here.
So that is telling you where the current came from.
Keep coming with 0.3. I mean, you want to keep going with
Can we-
0.0?
Can we follow this crest a little bit?
Oh, jelly coming in and out.
Yeah. Like that. Or maybe even the other one. That is a little bit more crowd. Thank you so much.
This is another 65 meters, so if you was doing 090. Probably would be the 090 at 0.2 because now
So we're changing our direction now a little bit on our exploration dive. We're going along
Hi, good morning.
the height of the summit here, the top
Bridge
of the
ROV,
summit.
if you could move off please, bearing 090 at point 2 continuous.
090.2 continues, okay?
Fish.
Oh yes,
you.
The
fish
first.
also hanging out here.
Who are you? This is super impressive.
Look, what are all the little
habitat.
streets here
I
from
don't
this like to the next?
I don't know. I don't know, to be honest, Fancy. We should maybe stop and check.
So much to check out here. It's crazy. Or is it?
It won't
Nah,
focus.
is it maybe a carnivorous? But it's like lying down.
It feels like coral to me. But
Wait, it looks like polyps.
like polyps, yeah, like
Yeah.
little polyps.
What?
I mean, yeah. It's just that I have the camera in front.
Yeah.
Antonio fits in it. Look at that, those are huge, yeah? Huge ones. There must be a lot of food here. So, I think that this is the new species.
Ah, so
Really?
the new
So-
sponge on the rock, yeah.
Yeah.
Did you take a sample in one of the rocks that you collected?
Probably.
I think that there was a, but
Was another.
I'm not sure.
We'll check. We'll have to check
I'm
later
not
in the
sure
lab.
at all.
So maybe better if you see a good size of a rock.
Yeah.
Yeah.
It's good that you let me know which is.
I will brief you now because there's a little bit of many things on the biobots.
And how about this Nigeria? Is California?
Yeah, sorry. Those are polyps.
So we're now looking closer at these, the little polyps. Might be cnidarians. Some sort of them, at least. But we're not sure.
Yeah.
They form these little streets or they hang out together.
Well, I don't know. And if I look at the bottom, there may be too much sediment.
And a little sea star.
See? Yeah, those are polyps. You see? Those are polyps.
Yeah. All over.
Let's see if I can
People
focus
animals,
this
right?
a little bit.
Yeah, maybe.
We'll have to change over the recording in 3 minutes.
Oh, yes.
Okay, on the hour.
This is.
Yes, I have 5 minutes here because I didn't click OK for 2 minutes or so.
Yeah, I just put it on my clock because 3 minutes.
Okay. Yeah.
Just
Thank you.
This is interesting. This is interesting as well. I don't know what
Some
this is.
symbols.
This one? No.
Or
Looks too.
Asconema foliatum or something.
Those two.
Yeah.
Those are polyps. Those are corals.
But those are individually placed there, so those are the colonies.
I'm not sure, to be honest. Look at that. Look at that.
Those are massive.
Yeah.
That's huge. That's now one over there.
Wow.
I mean, we have collected this one, not this one. I wonder if it has a stem. If it has a stem, maybe it's Sacocalix. If it doesn't, then
Super
I
big
have,
pores
I have
as
no
well.
idea what it is.
It looks super spongy.
We collect the crinoids.
Oh, yeah, I saw that was great.
There's a skate there.
Skate. The one, I think that's
The
just the
crust?
sponge that
Yeah.
fell over.
Oh.
And now there's like this crest, and my idea was to follow it for a while. What is that? Oh, there. It's a rock. Yeah,
white?
nah,
Yeah.
just
Yeah.
sediment. Sorry, Jason. Sorry.
Check.
No, I think it's just sediment.
Super interesting site.
Yeah, indeed.
It's a treasure.
Yeah, it is a treasure. Have you an idea what these little polyps that we saw right there? There were little polyps all across the floor.
Maybe they're a small anemone.
Yeah. So behind us are already
We have this
Anna, Helena and Sergi.
stems. You see?
Yeah,
We're
No
taking
idea
over
what-
in a few
no idea
minutes.
what is that.
but I saw something really weird, which is that this stem and then crown of sponge.
But
And then was
that can be sarcocalids. So I wonder if this is just all sarcocalids that died or whatever, and this is the stem. But I have no idea, to be honest. So maybe we should stop for the change, so I can brief you and we don't miss anything.
Maybe here on the side?
Okay, stop the RV.
Yeah, maybe with in the sediment.
Bridge ROV, come to a stop, please.
Stop.
Thank you.
Facing the.
Yeah, that
of
would be
course.
great. Yeah. So we're coming to a stop now to hand over to Sergio and Anna Helena and tell them all about what's already in the bio boxes and what we've seen. And
You see that? It's an asconema, and the other one.
yeah, there's a really interesting structure of a really big sponge. They grow really big here on the summit. Okay.
It's a
and 1 minute to tape change.
Me ha caído la
me he visto.
me ha caído
Ponerla
aquí.
aquí, ¿no?
Sí. Y aquí ha sido error mío porque...
Maybe you want to do it in English? Go Anna Helena.
No,
Sorry,
no, it's
sorry.
gonna be
So here you have the solendorids, you have the crinoid with the sediment, you have a piece of the fan shape.
Yeah.
Here you have the solendorids, that was my mistake. I should have put it
Oh,
here. I was
thanks.
thinking on the sponges.
This is already one.
But we can meet that was, no?
Better not to.
Well, then there we go. We have it here. A piece of a large actinelide that was on the huge rock fell here as well. Sorry. And here you have a rock. So
Yeah, the big rock.
normal space
Yeah.
here.
And this is
Luckily that
the
big
this is
fit.
the mug, right?
And this is the mark.
Yeah.
And then we...
But you collect the
Should we put the coordinates?
Yes. Let's
Here there is just a
change.
little interesting spot.
Now.
You can put
So
one
stop
here if you
recording?
want.
Yes.
The solenoids will have
Cool.
one stop on a rock,
Yes.
few. No.
2, 3, 4, 5,
Ah, yeah.
or even 6. Good job, guys.
Cool.
Beautiful.
So now we are, I think we are at the beginning.
Thank you.
So you follow more or less the
Yeah.
concept.
More or
Tape
less,
change.
You didn't finish it, right?
No, but we may expect this to go down now.
Okay.
But just
Couple of you.
Okay, are we ready to start recording?
Yes, start recording again.
Start now.
Yes, great, thank you.
That's it. So any more sampling that you should do?
Okay, so now we will hand over to Angelina and Sergi. See you later. Hi there.
Good luck.
Helena and Sergi taking over.
Just,
We have had slightly lazy morning, watching the beautiful live stream together with the rest of you, while Franzi and Julio have been down here with you. So we're going to have a little bit of a look, and then I guess we are flying on, right?
Yeah. Let me just.
So, ooh, nice shrimp. So this Mohns Treasure really lives up to its name. It's a beautiful treasure of a spot. Oh, that's also some really nice crinoids sitting there.
Yeah.
And
wonder
I
if
would
it's the same
guess
species.
this is the Bathynomus leucopis
Shrimp.
shrimp, yes. If so, that was actually one of the species that were described already in 1876, I think, as one of the first species described from the Norwegian Sea expedition that was led by Mohn, whose bridge and treasure this is named after them, together with Georg Ossian Sars, who was a crustacean specialist in Norway at that time. So that's kind of nice to find Sars's shrimp on Mohns Treasure. They went out in summers of 1876, 77 and 78 to this general area, but they covered all of what is today called the Norwegian Sea, and they did see a little bit that there was a ridge here, but not really, so they were not at this location at all. Please don't be confused about that. Now, which sponges are we seeing, Sergey? Lots of cool things, right?
Yeah. So we're seeing some of these fun-shaped sponges that Julio is trying to put a name on. So we have a bunch of them. Some of them are like 50 centimeters or even more wide, which makes them
Really
huge
big,
animals.
right?
Yeah. So the fun-shaped ones that you see, top right, those are the ones that make most of the biomass, most of the animals in weight, say into this area. You can see the current upwards.
Yeah,
Goes
it's
to
a
the right.
relatively strong current here,
Yeah.
but right now I don't know which way we're facing and which way that current really goes in eastwards and westwards, but
Thing is that it's quite strong, and this is what sponges also use to feed on. So they face current so that to maximize the food that they take in.
that's smart.
Yep.
Yeah.
This is why some different species have come to the same idea of building a fan shape structure. Some others, as you can see here also in the image.
Can you turn off the laser for just 10 seconds? This is the real life picture.
So in other cases, as you can see here, also in the middle of the image, this brown, long structure, this is a sponge, Asconema, genus Asconema. That was one of the first ones that we saw just at the beginning of the dive.
So this is actually, the brown thing there is in the same genus as those gray lace things,
Yeah.
the macaroni
Yeah,
that we have seen earlier.
That
But
we
this
saw.
is not the same species, right?
not really. Yeah. This species appears to be restricted, let's say like this, deeper water. So the other Asconemas, the one that you mentioned with these undulations or
Giant macaroni.
yeah, giant macaroni. So these occur in shallower waters. But yeah,
That pink thing coming in there is a Lysianacid amphipod, so they are scavenging animals,
looking for food all the time.
looking for dead stuff laying on the seafloor somewhere. It's also quite cool to see with all the little pebbles and cobbles here, the smaller rocks, that's also, of course, not sedimented that much over, and that's again because of this current that washes away that very fluffy sediment.
So it gave us the opportunity of landing on sediment and just observing this beauty, this landscape of mostly filter feeder organisms.
But we're looking at the ridge, right? Yes, so we're looking right at the top of a small ridge that we're following.
Yeah.
So maybe do you want to do something here, or do we want to continue following the ridge?
Yeah, let's continue a little bit.
Yeah. Are you guys ready to fly?
We are.
Yay! Ready to rock and roll.
Yeah.
And could we turn the lasers on again then, please?
This is, yeah.
Thanks.
Thank you.
So we have a zoom in.
This is wrong.
Time is wrong, right?
UTC one.
Yeah. Can you tell me the time?
UTC time is 6:09.
6:09.
Yeah, we are slightly losing track of which proper ship time it is, because we just get up at strange hours in the night and do our shifts and go to bed at strange hours in the middle of the day. So after a while you just get into how long time has it been that you have been awake and worked, and we have to check all the screens what the proper time is.
Spin round it, we lost it. I don't know. It's still 0, about 0, 0, just.
So just a reminder for those of you that connected recently, we started the dive at 2,785 meters, which is a long way down, and we are now just more or less the same depth, so 2,757 meters. We're trying to position the ROV. And for those that you are connecting again, we saw a Dumbo octopus, right?
Yeah. About half an hour ago, I think. So,
Yeah.
Yeah.
5:15 hour.
Yeah, almost an hour ago now, actually. And we are at the Mohns Treasure area. This is an inactive,
Straight up the line.
an inactive vent area. So that means that we know that there has been venting happening here previously, not in the time where we could have observed, but historic. And so what we're seeing are sediments that have been slightly enriched by this minerals that are being taken up by that hot water that comes in a vent, an active vent size. So the sediments here are in that way slightly different from an area where there hasn't been a vent earlier, and that is maybe what is making it
There's fish.
a little bit more species rich. That is at least a theory. But this landscape is, of course, partly formed of having been that hydrothermal vent earlier in historic times, and or prehistoric, who knows, not historic, and all of the other tectonic movements that are happening in this area. So lots of ridges, lots of seamounts, lots of dramatic landscape everywhere. And on this dive, we're trying to follow a little ridge, as you can see. So it's going down on both sides, and we are sort of following that top of that ridge. So a hill thingy somewhere here, and since this is coming up a little bit from the surrounding landscape, this is a perfect place if you want to be catching that current and find the animals that are or food that is available in the current. So that's why we see so many animals here on the top. And they're really phasing out, right?
Yeah. So they're facing current.
And you can see how that sandy mud is right behind them, where it's sort of in the shadow of the
Just trying to
current,
look
so it's
for
a little bit more protected.
organisms in there, like
Yeah,
they just stand
soft
out.
sediment organisms are also really cool to look for. There is a small shrimp. Yeah,
Bridge arrow,
it's
you can
already
move off
a little
please,
bit of
bearing 045
sorting
at
in that
0.2
sand.
continuous.
So
045 and 0.2.
I
but
did-
here we're back to those pebbles and cobbles.
Yeah, it's the perfect habitat for these organisms, and they are sessile. They need to settle on a rock. The larvae of these organisms is the one that is gonna get to a new habitat. It's gonna find the right rock and it's gonna start growing. After many years, probably hundreds of years, and you're gonna
check that out.
and then we have some images. We have these in mind also, how they look like. It would be wise to have a look at these rhinos and see whether they're two species or
Yeah,
just the same one occurring at different depths.
because at 1,000 meters depth, they were almost the fashion statement of the sponges, right?
Yeah.
They were standing there on top of every single sponge almost.
Again, let's see if we can spot these rhinos.
So this sandy, muddy part of the landscape is an excellent place if you are a worm, or if you are some of the smaller groups of smaller crustaceans. I especially like what we are in normal language calling the comma crustaceans, Cumacea. We call them the comma crustaceans because they look like a tiny little comma, a little ball with a tail on it, and they are great in this kind of sand. So if we had a sample of the sand, I am sure we would find lots of worms, lots of different species of crustaceans, not only the comma crustaceans, and probably also quite a lot of mollusks in there. So it's a different type of habitat, but very close by hard habitat, but a little bit more difficult to film. So it's harder to show you on a live stream.
There's another species, the white, fan shape. You see this white one for which
Are you zooming in on it now? Oh yeah.
So there is an Nordic now. Really? So big.
Is that the Licydendorix? We have started to learn the Licydendorix sponge
Look, these
name.
are the small anemone that we see. We saw.
Oh yeah.
Place.
Oh cool.
Can we stop here? Yeah.
Okay, so now we have the lasers. Could you please have the lasers off? Thank you.
Beautiful.
Yeah, look at those.
A little bit too bright. But so we have many things
Oh,
in here,
amazing.
right?
And they're using the ridge of the rock.
Yep.
It's like, okay, this is our rock, but we are
One
going to use
is following
only
the
the
other
ridge
one.
of it. And there is one of those non-stalked crinoids in the back,
Yeah.
on top of the Licydendorix. Is that the Licydendorix, or am I messing up now?
I think it's a Licydendorix. There are
Yay!
quite a few one.
Ooh, see,
on-
it's starting
on
to wave its
footage.
arms. Maybe it's
just
saying hi to us.
Put also in context the Asconema. You see the Asconema on the left-hand side?
The brown thing?
Yeah.
That is cool.
This is an exactinellid glass sponge that lives quite deep. We couldn't see it, at least during our dives at around 1,000 meters depth. Yesterday we saw it, and now we see it again at a similar depth. We compare with it yesterday. But these small cnidarians or anemone, or I don't know, that you see in here at the very center of the image. I cannot go farther with focus. Those is the first time that we saw it, right?
Yeah.
Shall we collect one of these rocks?
It's I think a big rock. I think it's too
could
big.
be. You can just try to find
A small
smaller
rock.
like this one. What do you think?
Do you think it's possible to pick up this rock, or is it still too large?
Just this one, Let it go forward a bit.
I think it's here. Yeah.
I'm down in the camera. Go forward a bit,
So we are going to try to pick up this smaller, hopefully it's a bit smaller rock with
Smaller
the...
than the previous one. The previous one was challenging, right?
Sure.
It's very difficult, even when we have the lasers, to really understand the size of things when you're just looking at it at a zoomed in video. But look at how those shrimp are standing up there on the
Yeah.
sponge.
The
What
one
sort of sponge is that? That's the big white
Yeah, the one that we need to identify, the one for which Julio already collected a small
Oh yeah,
piece.
we took a small piece in the beginning of this dive. And that's
Want me to move closer now?
yes.
So we're gonna get a little bit closer to the specimens that we want to collect.
So the ROV has really cool arms, but they're not super long, so we need to be slightly close to the specimens we want to sample so that the ROV arm can actually reach
We cannot
the
see
rock.
now.
But
yeah,
we
it's
need to retract. We need to take in the sextant.
Yep.
So we're going to take in the big camera you see, so that we can sample without destroying the big camera.
Yeah, thank you. I think so, right?
Yeah, the small, because you have that sponge thing, and then you have the big, and then you have the small.
So we start with this bio box. What do you think?
Or we can. It's no. That's
This was the mug, yes.
That's the mug. Yeah. Then let's put it in here. I think it needs to go in the starboard tray after a while. So it's a little bit like a game of Tetris to find the good place to place the different samples that we are picking up. Because we want to make sure we don't mix them up when we get them back to the lab, so that we have complete control over where we picked up the specimens that we are sampling.
The one with the white or the one next to it?
It's the one with the white.
Breakfast, I only go for the yogurt and all. But there are eggs, I think, and bacon.
The one with the white stripe?
Yes,
Yes.
That one.
the one with the white stripe. The white stripe is a series of little animals, which are the things we want.
And there's gonna be also some sponges attached to this rock that Julio and Paco will certainly work on. So let's just get
And this is going to be cost 6.
cost 6, yeah.
So I've made, it's just to write down the number of the bio box.
Okay.
That's missing.
Yeah, I'll put it. Once we know that it's gonna get
Yes,
there.
we have to find out which bio box we land in.
Oops. Oh, didn't catch.
Yay, it did.
Did you grab it?
Yeah.
Awesome. But it's been it's kind of disintegrated, right?
Integrated.
Yeah.
I'm not sure whether this.
Fell apart, right?
It fell apart, yes.
Yeah.
Just disintegrated.
That's how
Do you-
rocks here are.
Do you want to talk about yours and
Then
segments in the bin?
let's empty what's between the jaws into a starboard bio box, and then
Yeah, we need to find another one. Right?
Yeah. But we can put that in the same, right?
Yeah.
So the thing about these great manipulators is that they have very, very strong jaws, or what is the equivalent to our fingers.
Top left, if you want.
Top left, top
The whole.
I think there's a few.
Yeah. Let's try. So, but the whole arm could, easily break our arms if it just closed over. So it's a very difficult thing to sample. So find things.
We need another lizard. Do you mind if we put it here?
We can put another lizard and oryx there. That's perfectly okay.
Do you think we should go for another rock with this anemone?
If we find it small enough in the close vicinity. That's great.
Awesome. Thanks, Will. We're gonna try another one.
Okay.
Just in case we don't smash it.
Okay.
It's not we're blaming. It's something really soft. You imagine this rock like being like hard,
yes.
but
Yeah.
they're not, right?
They just started there.
Yeah, I think these rocks are really easily breakable.
Okay.
Don't use them to build a house.
Not really.
Six.
Let's try to find one. I think there's one in here. Not that far away, but I don't know if you can reach it.
Maybe we can zoom a little bit in again.
One just behind. Yeah, that one. You're right. So we're trying to get this one.
Oh no. Now we just move this a little bit.
Yeah, that's the one. So it's again. So let's go now.
So we're gonna try to get a second cobble.
Going like this?
Sorry?
Like this? Instead of like this? Well,
It's how it's possible.
yeah.
I mean,
Yeah, but it won't be going flat. Which one is that one or not?
The one with the wing.
Maybe now the jaws.
Where's we can get this?
that looks very good.
still there, right?
Yeah, there's.
Have I got it?
Yes, I think you have.
I don't know whether this was a one, but yeah.
You have a rock.
Oh, it's just a... Is that the one?
Yes, it is. Perfect.
Same by box.
Yes.
We are.
This is the same cost.
Yes.
Awesome.
That's great. Thank you. Did you also want? You also want to listen
Listen,
and
there
daughters?
is, I think it's a
Yeah.
good place, yeah. Now we stopped here. Yeah, almost there.
Mhm. I think it's sticking to the clothes
Yeah.
now. Sometimes I wanted to have a toothpick for those cloths and clean them. Help relieve.
Great job.
Perfect, thank you.
Thanks a lot.
You now wanted to have one of those
One of these
spaghetti.
you collected before. We need a few more.
So the reason why Sergei wants to sample another of the spaghetti sponges, the Lysodendrorix,
Or this one.
is, like we have talked about earlier, that this is part of
There's another one.
a study of a very, very widespread species, this Lysodendrorix. It lives shallow-ish, at least 1,000 meters deep, and it lives deep here almost, well, we are now at 2,750 meters depth, almost 60, and it lives in all of the Norwegian Sea, so a widespread sponge, very, very common, and at the same time it's a brooder, so it cannot spread easily from place to place or get in contact with its congeners. So the study that Sergei is working on is using the genetics to find out how connected these different populations are. If they have contact with they don't have any larvae in the water, but maybe they have contact that the little babies are floating around in the water, or maybe not. So this is the big reason why we're sampling several times the same species here, that we want to understand how not very easily movable species can or cannot have contact with the other populations.
Yeah. And you may have noticed that this species is quite common, so really, really abundant in this region. So taking a few individuals or pieces of some individuals is not gonna affect these species. So then we're going to extract as many information as possible from these specimens when they're going to be used, not just for this, but also for this eDNA characterization.
Yes, because another thing we are doing is are you going to use them as the active filter? Yeah.
Natural samplers.
So we can use sponges because they're filtering so much water as a natural biofilter in a way for all of the other DNA that is floating around in the water.
I think it's just caught
You can imagine if like that Dumbo octopus that was swimming by an hour ago, or the fish that we see, or the other sponges and the crinoids we're seeing, everything, they lose a little bit of DNA to the water. So we are using water and sediment to get an environmental sample of the DNA, so a picture of what is in the area, in the environment, from the DNA that is shredded or even maybe for some larger species peed out or whatever. To make sure that we really can, from a list of different DNA sequences, say which species these sequences represent. We also need a library or like a connection card between the sequence and which species it is. So we are always sequencing all of the species that we are sampling to build up this library of species name to the sequence, so that when we are later doing environmental DNA, we have a library that we can connect with for getting the right names.
You can grab this one if you want.
Without a library like that, we would only be able to say, okay, so we have 15 or 25, or I think in this area we would have 125 or something. But with that library, we can also say which species are those 125 different things. And now we are sampling the Lysodendrorix, and look, the ball of spaghetti has like a stem in the middle. I didn't know that.
Yeah, this is the first time that we noticed that, right?
Yeah, that is cool. They always just look like a ball of spaghetti, and now they have this central
like
figure.
White broccoli, right?
Yeah, I am changing my name of that species from spaghetti to broccoli.
This specimen.
We are comparing everything to food, and we shouldn't be hungry because we just had food. We just had breakfast right before we came into the shed here. So that is, I don't know. We're getting food all the time on the ship, and maybe we are thinking about food all the time.
Yeah, we need to do some sport
at
some point.
There is a gym on the ship,
Great.
so small, but very nicely fitted gym. We have a rowing machine and spinning bicycles, and even a treadmill so we can run. So there is the possibility of spending some energy to work off all that lovely food that we're eating. Look at that sample. That is great sampling work. Again, I just have to do a shout out to our wonderful ROV pilots. They are doing beautiful work all the time for us here. So with the other group, we went with hearts, so we're doing hearts to you guys too. This is the new, very famous, well, not new anymore, this is, like 5 years old, cool heart. Hearts to all of you.
So anything else that you feel we should be collecting?
No, I think I'm good from here.
Yeah.
Are you good from here as well?
Yeah.
Then, whenever our pilots are ready, we can fly on.
I just thought
So we're gonna follow in this crest or the very top of this hill or mountain to see whether we see any other beautiful sponges or non-sponges.
Sponges and other animals.
Yeah. These dominate here, but there's plenty of
smaller and bigger things all in between, right? So.
Just center op jouw camera, zeker te zien.
centering.
Center?
Okay, now.
So we're going to continue, as Sergio said, along this field or ridge that we're seeing now, and it's really, I think it lives up to its name. It is a treasure.
Yeah.
It really is a treasure.
I mean, it's boasting biodiversity. I mean, those are animals. They live there for many, many years, and they're doing their job. They're like filtering all the time all the food that gets from the surface and from other areas in this sea bottom. It's really cold seawater, right?
Yeah, the temperature is minus 0.65.
Yeah, quite similar from yesterday.
Yeah, I think we're in the same type of water mass. We're not too far away from where we were yesterday. So it's similar water masses, I think.
Yeah, can do.
But they are
Just need to say.
rich in nutrients because there are so many large animals that need all of this food. So
Yeah. There was no food. Yeah.
yes, please.
Thank you.
Thank you.
The amount of food is related to the amount and biomass and weight of animals that are living here. So you can imagine that this is a really productive water mass. And sponges are getting food from it all the time.
So further up in the water column here, the most in biomass, the largest amount of specimens would be from a copepod. There is a lot of copepod called Calanus finmarchicus in these waters. And it is, even though it is a tiny little planktonic crustacean, it is so important in these waters for as food for the fish that are also swimming up. Not the benthic fish, but the higher up fish, pelagic fish, that it has a common name in Norwegian. It is called the rauåte, so the red food. And this red food or red feed is full of these omega-3 oils. The whole animal has a big blob of oil inside it. So the animals that are eating this copepod are eating almost like little happy pills of omega-3. And some of them, of course, will not be eaten and after a while die and fall down to the seafloor. So there is some nice, what we call pelagic benthic coupling, in the way that the food or that the animals and plants even at the top that are swimming and floating around in this water, 2,700 meters of it, after a while when it dies it will fall down to the seafloor. And much of what is now in this sediment, it will of course be also sand and mud and stuff, but there is also quite a lot of organic parts in this mud and in the waters right above the seafloor, of course, and this is what all of the animals that we are looking at here now are eating. So when we have these large sponges, some of this organic matter is still floating in the water at this depth. And when it's not taken up by the sponges or the feather stars or all of the other animals that we're seeing here, it will land on the seafloor and after a while even be sedimented down a little bit. So the first maybe up to between 2 and 5 centimeters of that mud layer will be full of food and animals.
This rondish specimen.
Yeah, that was cool. It's beautiful.
You may have noticed there's some different colors of these fan-shaped sponges. The white one is the one that we just collected part of. But
Okay,
you see,
like can you move
left-hand side of the image there
at
is an
0.2
orange,
Oh yeah,
orangish
an
ones.
orange found there. Yeah.
This is indeed another species.
That's it, thanks.
Do we have samples of that as well?
Yeah, I think so. And Julio and Paco already put a name on this species. I'm gonna check my notes. So Julio's told me which is the species. I'm gonna share this with you in a minute.
So I have been so fascinated today that I have not been remembering to log as much as I should. That is bad of me. Ah,
So this Stellodorix flavellata, the one that I was just mentioning, the orangish one, also fun shape. Look at that. Those are the, again, these small white lines that you see there. Those are the anemone, little anemone that we just
yeah, that's
picked.
amazing how they're building lines. Wow. Look at that. It's like they're sort of, we're going this way and we're just spreading in a long line.
Oh, I'm sorry.
It's a little bit far away and we're
Yeah.
flying, so.
We have the problem again to get, yeah.
Let's
Oh,
just,
it's fixed.
okay, that's good.
You see these lines?
They're crazy.
Nothing underneath in the back.
Let's just leave it a little
Yeah.
bit and see. We can do the pan and tilt, right? Yeah.
You can change it on the software if you want, but it's not as easy to use.
Yeah,
You have to use
yes.
mouse.
Again, this Stellodorix flavellata on the left-hand side of the image. You see,
So,
it's
do you think,
orangish.
are they always along the side of, right next to the mud?
Mhm. Whenever they find a rock.
But they are not here in the middle of the rocks, so they're always
yeah.
on the edge of the rocks. Or am I just projecting
No,
something now?
no. Yeah, you're right. Maybe the currents are.
Different down there.
Yeah.
Maybe the food
Stronger.
availability is different. Or maybe they just landed there by chance. Who knows?
A combination of these two, I guess.
Oh, there was one of those fat sea stars as well.
Yeah. Do you want to
No.
get up? No. This is on top of this huge sponge.
Can you get it up there?
Yeah. So we're continuing the exploration of this area. Beautiful.
I think there are still some crinoids sitting on those Asconema, the brown Asconema, which is not the
Not
same
the
as
same
the
one,
no.
And you can tell from the different shape and the different color. And this small lines. Oh, this is something different, right? What is that? Those are the small anemone again.
Yeah, but they're not in a line.
No. Well, if you get closer, you see more or less the line.
Yeah.
So we have some samples and we will try to get the species, to identify which species belong in
Reach
here.
RV bridge.
Oh, maybe several kind of lines.
Can we change the
I think they
direction
like to
of
stand
this
next to each other.
movement to 090?
They remind me of when we went to at
090
elementary school when we had to
1.2,
line up
okay.
in a crocodile all the time.
Okay, thanks.
Look at that. What's that? It's going to fill down.
Yeah, because that can also happen, right? A sponge can sort of lose its balance and fall
Yeah.
over. But here we have two different sponges on top of each other, yeah?
Mm-hmm.
So, Lizodendorix holding up another one.
And then on top of it, there's
Yeah.
this Stellodorix.
Right above the two lasers, we are talking about those
Yeah.
sponges that are on top of each other there. Okay, I will make sure that I'm taking more notes now because this is embarrassing.
So we're gonna try to move somewhere else, following this bridge.
That's the end of the line there as well, the transect.
Oh, we're in the sunset. Okay.
But yeah,
Shall we move to the other one?
yeah, we can move to. We have planned several transects today, so I think it's a very short hop to the next transect.
600 meters, I think.
Yeah.
Probably not that short.
So we are at the end of our first transect, and we are planning a little bit of a hop. It's probably fastest to do an up and
Yeah.
yeah. So we will go a little bit off the seabed so that we can move controlled and a little bit more quickly to the second transect. This is 600 meters away from us, so it's not a very long hop. It's just to make the pilots find it a little bit more easy to fly fast if they don't have to fly right above the sea floor.
I think Julio just mentioned that the big one may not be the same species, and he would like to have
The big one like that one.
that big one. I think we
But,
can.
but not here. Let's do that on the next transect because I think it's going to be relatively similar. And now we have started the flight.
Okay.
So sorry, Julio, you have to wait a little bit to get that big one.
Other side, 050.
And we'll try to fix this. Oh, okay, it's fixed now.
Yeah,
So
just
sometimes,
needed a little bit of resting time.
Slowly, gently.
That's where
So everything on the ROV gets used pretty heavily when we are flying here, and everything is down in that minus 0.6 degrees cold waters at 70 Celsius. So below the freezing point of fresh water, but remember we are in salt water, so that's perfectly doable. But it's a very cold area for the ROV to be in as well, and it is at 2,760 meters depth, so that's a lot of pressure.
Yeah.
That is really a lot of pressure on everything that we have put down there. So our focus is sometimes a little bit not happy, but it's coming back to us. So we are off on that little up and
A
fly.
little fish.
Yeah, that's a nice fish.
Oh.
That's
look
definitely
like,
one of the benthic
stressed.
ones that is, where am I going to find food now? Yeah. Demersal fishes. Ooh, and look at those round sponges
Yeah.
laying in the sediment.
So, fish that we just saw.
It's hiding right behind a stalked
Yeah.
crinoid. So
Yeah.
it's a good background for that stalked crinoid. You can now see how that crinoid is actually off the seafloor.
So there's a bunch of these crinoids. You can see the stalk, so they are sessile. I don't know how long, like 10 centimeters or something like that,
Yeah, I guess so.
in order to put them above from the sediment.
And yeah, you can see several in this picture of these sediments. And now we're off flying to the next part of our dive, the next transect. So before we do dives, we sit in the lab and look at available bathymetric charts for the area. This is one of the areas that have been studied quite extensively, so we have good bathymetric charts of this area, and then we can identify areas where we think will be interesting things to look at and examine and maybe sample. And then we plan transects where we want to study more carefully. So we have now done our first transect of the day, and we are on our way to the second transect that we have planned. And I think for this place, for Mohns Treasure, it was actually Julio who was doing the planning of the transects. So he was sitting down with all of that bathymetry information, and I think he even had what we call backscatter information. So that's a little bit information about how soft or how hard the seafloor seems to be from echo sounder information. So the bathymetry and the backscatter comes from using the echo sounder of lots of different vessels in this area especially.
Okay, that's it, thanks.
And the harder and faster that signal is coming back to us,
We have a fish.
the harder the surface is. So that's how we can say something about if it's soft or hard bottom from bathymetric, or the same type of information. It has been collected the same way. 20 centimeters?
20 centimeters
Yeah.
long. Yeah. So this is what why we are using the lasers to identify
that's
how
a
big.
good
animal.
information for that. If you just see a fish, it could be 2 centimeters or 20 centimeters if you don't know from the lasers. There is more of this. Oh, that is,
I think there's some sabellids also here.
yeah, some sabellids, so that would be worms,
Yeah.
in addition to the stalked cranoids that we have looked at earlier.
Yeah.
And yeah, if I could have, from my research, what I would do quite often is to find an area just like this, and then set down an epibenthic sled is what I call the instrument I very often use. It's like a plankton net that is pulled over the seafloor,
Yeah.
and then I can sample a very short distance with the animals that are sitting right on or right over or right under the seafloor. All of these small animals that I talked about. And since I am a specialist on small crustaceans, I can then spend days and days and days just sorting or even trying to identify the specimens that we find in a sample like that. Because even though they're invisible on this camera feed, because we don't get close enough to see anything that close, of small animals, I would say, then I am certain we would find lots if we had a sled sample.
For sure. There's another fish there.
Oh yeah.
So these sedimentary bottoms are also filled by animals, smaller animals.
Yeah, it's a good place to hang out and eat if you are a fish.
So we are now finding sponges because sponges, they need hard bottom to where to settle.
And you know by now that Sergi is our sponge man,
Well,
together with Julio and Paco on this ship.
oh, look at that beauty!
Yeah.
You can see its face.
And that definitely looked like a sabellid on the side there. So worms are tons of in this mud and sand. As I said, all the way down to maybe 5 centimeters deep is a lot of organic matter that had been snowing down through the water column and landing on and in this seafloor. And look at all those holes everywhere. There are indications of lots of animals.
Burrowing animals.
Or just hiding or being down there looking for food or whatever. There's lots and lots of life here. Ha. Definitely want to come here with a sled one day. That would be
Yeah.
fun.
You you will get something to work on, like lots of things to work
Oh
on.
yeah, then I would have tons of things, not tons because metric tons are really big, but I would have lots and lots of animals to look at.
So we are 2,000 and...
764 meters.
Yep.
I think Jason, who is running the live stream from up in the dry lab, has managed to put down the depth on your screens now. Since a lot of you have asked about the depth in the commentary, we are
big things there.
always working on
Let's just
trying
find
to
out.
help you. So if you have any questions, please feel free to
Ooh, sponges again, right?
put your questions in the commentary, and
This...
we will be told
what's that?
down here also what questions you have so we can answer. Isn't that a sea star?
Is there a sea star? Yeah. I
It's
think
a
so.
sea star. Trying to find food in the mud. Yay. But up there is, I think, little rock.
not
No,
sure
it's not.
what
It's
the-
not a sea
a
star.
sponge.
It's a sponge.
A
Oh,
sponge,
it's
yeah.
always a sponge.
Yeah.
You always get right. You have all these sponges.
Okay.
Okay.
Sabelle, it's in here. Those
It is a world of sponges. It is.
things in the middle.
Oh yeah,
So they,
and a tiny little jelly.
so sponges, they get any chance to grow if they find any of these rocks. There was just one rock in here, and it was colonized by sponges. A few more in here.
Yeah, look
I
at
guess
that.
same species as the ones that we
Oh,
just see.
so what's the pink thing up there?
Let's find out.
Is that a sea star?
That it is. Yeah.
Ha! So I have the sea star after all. Ha!
Ah.
That's good.
There's a mountain here. You see? This is.
An octopus.
Oh yeah, that's a little hill. Oh, is it an octopus? Can we slow down and zoom in?
Yeah.
Can we zoom in?
So there's interesting things in here.
Yeah.
Which ROV can we stop the movement for now?
Let's hope so. Are we zooming in? Yeah. So it's a small one. It's
It was a sea star, right?
not sure. It looks a little bit sea starry to me.
Yeah.
There are somebody who are mumbling.
There's a foot.
Let's just check. It's super close to us. Oh. Is it eating one of these crinoids?
Oh, it's
What do you
yeah, it's a sea star that is eating on a crinoid.
No way.
And I think it looks like a hymenastra or something like that.
Can you turn off the laser screen?
So this is
Beautiful.
one echinoderm eating on another echinoderm. Yeah, eat your friends, eat your enemies, eat your neighbors. So we should say sea stars have the mouth on the underside. So like
This is really
in the middle of the belly in a way. I mean, because the sea stars are radiosymmetric, so they have a symmetry around the midpoint in their body. So, and the mouth is on the underside, and the out opening or the anus is on the top. And so it's sitting here and eating. And maybe it's eating I think it's a stalked crinoid stalk
Yeah.
that we see here. So I think it's eating on the crinoid because
yes.
its mouth is not probably touching the seafloor here. Even though the tips of its feet are touching the seafloor.
So now that we're here, let's explore a little bit. Right?
So can we look
Start.
up there?
Don't have a problem again with
Yeah, if you can zoom in on that thing there. Let's see if that is another stalked crinoid or if that is a sabellid.
this mound in here.
That thing there.
Yeah. Got it.
It's a crinoid. That's a stalked crinoid.
Ah, yeah. Yeah, yeah.
And
Probably
it
the same
has a
species.
snail, is it, on the side, on the bottom here?
Yeah, could be.
Yeah. So this is another echinoderm, and they are, as we talked about, using a stalk to get a little bit off the seafloor to get hold of that food. That is cool. But there was this little mound with
Yeah.
like a little hole right next here. Yeah,
No
look
one,
at that. And there is another sea star behind that. Ooh, and look, it's a sea pig.
Oh, yeah.
Ooh,
I will come.
we
Yeah,
have
we're
to look
waiting
at it.
for it, right?
Alpedia, the sea
We were looking
pigs.
forward to.
Yeah, but this is a mound somebody have made.
Yeah.
But look at
I
that.
would prefer to.
This is a small Holotheria.
Yep.
Yeah, a sea cucumber. Super tiny. We call them sea pigs. They're deep sea Holotheria. In the, is it family Elpidida or something like that?
Yeah.
So, sea pigs. Oink oink.
Yeah.
They don't say oink oink, but I say oink oink when I see them because they're little pigs.
So, as opposed to sea stars are predators, these sea pigs, they feed on the organic matter retained in the sand. So they are like processing all the time a little bit of drops of particles that get into the mouth, and then they use organic matter retained in the sand to as a food, and then they produce fecal pellets. These pellets
Poop.
are just this poop.
They
That is
poop
like
like
that,
sand.
compacted, and in some cases they're like long pieces of
Long strains of sandy
sand.
poop.
Yeah.
Yeah. But this is a cool picture because we have three echinoderms. We have this sea pig, and up
Yep.
in the upper right corner we have a sea star, and a little bit further down, but still on the right, is another stalked cranoid, isn't
Yeah.
it? There,
I think so.
kind of almost invisible. There's another stalked cranoid at the very bottom there.
Yeah, here.
So this is an echinoderm world. So
Do you think we should
echinoderm
get
is,
a little bit closer,
I think it's impossible to get a little bit closer without
flying a little bit.
yeah, but that's okay. The thing is that echinoderms are called echinoderms because they have spiky skin.
Yeah.
So all of them are sort of united in a group because they have spiky skin. We are using Greek words in Latin.
You do.
Perfect. Let's fly on.
Thanks.
That was a nice echinoderm
Yeah.
stop, huh? Yes. The spiky skin stop.
It's not that we're speaking just about sponges. There's
No,
some
we
other
only
creatures.
we have been speaking a lot
Yeah.
about sponges, but now we had an echinoderm stop. That was great.
Yeah, yeah. And small one, this holothurian, this sea cucumber.
Pigs, sea pigs. I love them. They're really cute.
Yeah. But these sea cucumbers have lost this protection that they have like a small bits of skeleton in the soft tissue that they are made of. Unlike other echinoderms, such as sea stars, for instance, you touch this sea star and you can feel this is hard, right? This is the
Yeah,
skin
if you pick
is
up a
super
sea, if you pick up a sea star at the beach, it will feel very
hard. Yeah,
hard, right?
this is the skeleton that produces this. Okay, I think we have here. So there was a
That's a shrimp,
shrimp.
and there are some. Oh, it's another anemone.
Yeah, sorry, these fan-shaped sponges again. Yeah, getting some trouble with sponges.
But we are on our way towards the next transect, so let's fly on to the next transect. It was just so nice seeing those spiky skin animals.
Yeah, we'll just move to the next one.
Yeah.
Okay. That's super nice. We get some water.
Bridge, RV. Okay, we can continue with the previous movement.
There is another
direction
fish
005
laying there.
At 04
So,
knots.
but we will try to have some sort of a movement so
Okay.
that we don't stop and look at everything, since we want to get to the next transect. So we're going to try to be slightly efficient now. I don't know if it's possible when we're sitting here and seeing all these cool things. We will make an effort about actually getting to the next place,
Yeah.
right?
So, how long have you been already?
Yeah, 1 hour and 45 minutes. No, 1 hour and 15 minutes. There's lots of fishes on the seafloor here, right? That is, and they're all just laying around. Okay. I've had my breakfast.
Well, the sea star was having its breakfast, right? With the pilot.
Yeah, that sea star was having its breakfast. And there is a lot of these stalked craneways there. Are the lasers on again?
No.
I don't think so.
Lasers, yeah.
Yeah, thank you.
Okay.
I always tend to forget the lasers,
Yeah.
but they are such a good help. There is another little jolly passing by.
Okay, cheers.
I will.
Been there.
I don't saw any beams. I don't know.
After breakfast?
Breakfast.
Yeah, breakfast is
Shot
good.
already, like, or? Shot your breakfast already?
We have had our breakfast
Thank
before
you.
we came in, thank you.
Ah, okay. Hey, well, all right.
Enjoy your...
Yeah, okay.
So it's breakfast time at the seafloor and it's breakfast time in the ROV shack as well.
Is to go faster than normal.
There's another little sponge coming up. We are climbing uphill slightly, or no, not very much.
Not very much, maybe a slight incline.
Yeah.
Yeah, it is a slight incline, not much. So yeah, just 51, so yeah, 51 pounds.
Yeah.
Coming down. Actually,
Look at all those holes here. That's a lot of, almost like little burrows. Things are living down there.
Full life.
Full of life. Another sea star. Do you think it's having another breakfast?
At least it looks like a similar species, right?
Yeah.
Isn't it?
I was wondering if this is. I will check if this could be the.
Yeah, it seems like we are climbing now. Mostly sedimentary, but some rocks here and there.
I can check if we are supposed to start our. See, I think it's this one. Oh.
Have a name for this one?
So we were at. Oh, it's only something like 50 meters up from the previous
There's a
stop.
huge sponge on the left. Very big one.
So don't you think it looks like this?
It does. In
Yeah.
an aster. Yeah. Antonio.
That Derek knows better than me how to fix it.
Yeah.
Now we don't need it.
But is the focus that is not working?
No,
The zoom. Zoom.
The zoom is hanging. It's lacking. It's sort of sticking every now and then.
You know that it's dependent on the software, but the zoom, I'm not sure.
And try again. Let me just check.
It is.
Oh no, it's working now.
Yeah, it needs a little
No.
bit of a break.
See? You're doing nothing.
And if you just do a small touch back, it stops.
Yeah, so maybe it's
It's
just
coming
sticking a
back.
little bit.
No, it's not coming back, right?
Yeah, yeah.
And now, so it's working, not working, depending on. Hold on. So it's back now.
Let's see how it goes.
Have they had a similar ratio? Audio?
No. Actually, during the shift, it was working fine, I think.
Yeah.
Yeah.
Fully didn't say anything, so.
Yeah. Because we have the time like that close up, so. I guess I shouldn't get the fisheye, right? What do you think? It's not the most beautiful.
Yeah, it's not.
landscape, but
It's a biologically interesting
Yeah.
landscape, but again, maybe not visually the most stunning. But Sand is also beautiful in its own way.
Gonna tell Julio to
That's a water box. So, can I just stick that up inside such a way? Or the bike on the right?
Yeah, with this one.
Yeah, we'll put them up there actually. I forgot to take it when I went for breakfast. I'll give it to them.
I'll just put
We can try to.
Way more than anything.
We can try to take them. Remember to take them out when we're swapping.
Oh, thank you. Normally I take it out myself, but I use
It was a two cups of tea morning, was it?
yes, two coffees, strong
Ooh.
ones.
2 coffee morning.
Yes, I like the coffee.
live again sponges.
I thought everybody in the ROV shack were tea people.
I am normally, but
Yeah.
if I'm tired, which I am on the night shift, I like a coffee.
So it's the day shift that are the tea people. It's
Yes.
easier to be tea person on a day shift.
It is, yes. I drink a lot of tea as well. Normally coffee out of those machines on most vessels means great, but it's okay on here actually.
We have a good coffee machine
Yeah,
here, yes.
Yes.
So we are well looked after. We are. In case all of you guys listening in are wondering if we're getting our good coffee, then we really are, yes.
Yeah, I buy my own coffee, so I like a decent one. So just pour up and all.
I am a little bit of a coffee snob at home, so at sea I always only drink tea.
Yeah.
Is it shrimp? Yeah.
So we have a bunch of casts over
Look, there are these right big sponges to the sides there. Oh, we passed them now, but they're coming up some more
Yeah.
up there. And it's amazing how big they become. And at the second that they find just a little rock to hold on to, they start living there. So there is no unused habitat anywhere. At least 3 different species, right, of sponge on that one rock?
potentially even 4.
4, yeah, okay. And there are some more sponges coming, and there is a little rock with a lot of things on it. Lots of little things.
Yeah.
But it seems we're coming to a more pebbly area. So again, then immediately we see that there are more sponges that we are passing. I think this fits with the symmetry we are crossing over, so it's a little bit, we're getting to more hilly areas, right? So we are, this is the Lysodendorix, right?
Yeah.
Yeah.
Big one.
Big Lysodendorix.
All over the place.
All over the, and some fan-shaped sponges, right?
There's
And I just
overhanging there, right?
remind you all that this is still on the hop between 2 transects, so that's why we're not slowing down right now or not playing extra. Oh, that's a big boulder. Not doing lots of extra stops here because we wanted to get you to that next transect that we have chosen, which is hopefully going to be also super nice and interesting.
Indeed. For sure it will.
But it's nice to pass over these little ridges of boulders before we go back to the more soft bottom again, where cobbles or even pebbles make up the hard bottom for the occasional sponge.
Yeah.
Yeah.
Paradise for sponges.
I would say actually this is paradise for worms and little crustaceans.
And you may have noticed that we haven't talked about carnivorous sponges yet.
No, we haven't.
Seen.
All of the day,
Yeah.
no carnivorous sponges. Ooh, that's the day. This is why this is a heaven for little
Yeah.
crustaceans, because there's no carnivorous sponge to eat them. Right here, anyway. But we have seen some of those big predators, the sea stars.
Yep.
And we saw them snacking on. So that's that's a fish, right? No, that's no. Come on, Anna Helena, that's an amphipod.
Yeah.
Oh, stupid Anna Helena sitting here, not even seeing her own animals. And I even washed my glasses this morning, so I should have seen that. That was another amphipod there. Yeah, we're just continuing on this, flying over this sandy, muddy, small animal heaven. And I think we are almost halfway on our hop. So we will continue there. Oh, there is a fish swimming away from us. Yeah. Okay, I'll log the fish.
Sorry?
Are we heading to
Yes. Yes.
So that's another rock. Another
No sponges.
Well, there are lots of little sponges
Lovely
around
small
it.
ones.
Yeah. Lots of little things around that one. And do you still see the stalked crinoids?
Not really.
They are very good at hiding, right?
But they normally leave like trails
Yeah.
where they have the stalk, and I can see it now.
But we are coming to another field of harder seafloor, and again, directly more of those bigger animals. So, bigger animals, I mean sponges, right? Bigger animals, sponges.
Bunch of them. This predominant, the most of the habitat is sandy, or
Yep.
we're
Sandy
not gonna
mud.
find sandy mud yet. We're not gonna find many sponges right now, but maybe. As
But
we
there is
start
some
to learn.
encrusting things on that, maybe, the grey.
Red fish.
And another little field of cobbles. So just a reminder, we are saying pebbles and cobbles and boulders, and I'm looking at this list in front of me here, where boulders are larger than 25 centimeters, cobbles are smaller than 25 centimeters, and pebbles, the smallest, they're like smaller than 6 centimeters. So I think here we have a combination of all three. This is a list that some geologists have helped us make, so I hope if you are listening and you are disagreeing, then you can talk to your geologist friends and find out about this. But this is definitely like a cobble boulder field, I would say. And they're a little bit, I think we're coming to a crest because these are kind of sharp around the edges more, so these are more fresh. And there is an anemone sitting on it.
Yeah.
And there are some sponges here and there, but really not so much. Oh, and there's a fan shape there.
Yeah.
But I think also from the bathymetry that we're going over a little bit of a
So we're now at
crest at 2,700 meters.
Yeah.
Yeah. That was quite a sudden change from the sand to the cobbles, right?
Yeah. Still climbing.
Yeah, but that's the fun thing. You're in this landscape, and it's really. Remember, this is a very three-dimensional area, right? You're not. When we look at the map of the sea, we always think it's just this flat, white, blue thing, but. And I have to admit, outside right now, it's a relatively flat blue thing. It's really beautiful weather out. Almost no waves whatsoever. So it's almost possible to think that this is a flat thing, but here down at the seafloor, it's really three-dimensional and wild landscape, even if a lot of the landscape is made from sand and mud. We have all of these rocky fields, we have all of these hills and little mountains and crests, and I think going for a hike in this landscape would have been a quite hard job.
Yeah.
Yeah.
This area is dominated by this Sulfid-Hügel complicata, right?
Yes, so you are in heaven.
Yeah.
Yeah,
Even
Sergi
just
heaven.
At the top, we have some
I have
of
to
these fan-shaped sponges, but you can see that the most dominant species is this Sulfid-Hügel complicata.
Yeah.
These small bushes, spaghetti-like.
The balls of spaghetti.
I
That
think we
seems to be
reached
the top of that
yeah.
little hill. We're passing this little hill on our way to the next transect that we have
Yeah.
planned for you and for us. So yeah, and since we are on a hop between two transects, we are probably not going to focus so much on getting super pretty pictures of the seafloor right here, but fly a little bit halfway, efficiently at least, to the next stop. But there are still some sponges to be seen, so
Yeah. all the
Sergi
time.
is always happy in here because he can see the sponges all the time. We can down like that.
Yeah, that's fine, thanks.
A little bit of a sandstorm as we're going down.
Yeah.
That's okay.
So I think we're going to be crossing this sedimentary area for a few meters, and then after this we will start transect number 2. It's going to take us a while to get there.
Yeah, but now we're on our efficient little run.
Yeah.
There is a really nice big fan-shaped thing there on the side.
There's our
Oh, and
huge-
look at that hole. What's that?
it's probably
Can you
our
pan
one.
over to it? Yeah? No, it's too late. I think there are some really nice burrows in here. That's another thing that you can find in these sandy areas, sandy, muddy areas. You will find animals that are digging burrows and living down there and either using it as their home or as a place to hide when they are hunting. Let's look there, for instance. I think some of these areas are really areas for little holes.
Yeah.
Only with the sun. It's raining over the rocks.
It should face downwards. The lasers, I cannot see the lasers now. Well,
Yeah, they're there.
oh yeah.
Lasers. It's hard to see the lasers when they don't hit anything in our visual field, because then they're just sending out there. Then they need to. But they are twinkling a little bit in all of that marine snow that is coming down. And some of it is still animals that are alive, and some of it are quite a lot of the marine snow. I should point out, sounds a little bit disgusting, but they are little fecal pellets from animals that live further up. So
poop, I say.
down here at almost 2,700 meters, a lot of the marine snow are tiny little pieces of poop. But there is a lot of energy in it still, so
You can tell.
yeah.
These huge specimens of sponges.
Are they poop eaters? Other animals,
I think
poop.
they don't.
they don't really care
yeah,
if it's
don't
alive
care.
or not when they eat it.
Small stuff they can filter,
Yeah.
and they get into these conocyte chambers, and they process it. So it's not that they have a gut, but they have like small chambers all over the tissue
Mm-hmm.
that are in charge of getting food, and then also of pumping the clean seawater into the water column. They are filtering all the time. We were saying the other day that depending on the size, they can be up to they can filter up to 20,000 liters per day, which is like a crazy amount of water.
That is crazy. They are little filtering machines, aren't they? Is that the Stylophobula again? I thought I saw. I will have it as a question mark.
Is this jellyfish, this
The red jellyfish?
red one, is coming every now and then?
I don't know, but I can ask a friend.
We got one, but
So I guess we're gonna get there like right before we start the new tape or something like that.
sorry.
Another fish.
Yes.
Close a little bit.
just gone off this, so this is 300 to that line, yes. It's not so.
It's 005.
005. Yeah, but this is
But
the
it's-
error, yes.
We don't know if it's 300. It's maybe looking for this, I would say that it's more 310.
I think this might be a Cordula.
Could be.
There as well, maybe.
Yeah, we'll play with
This
it. So.
this would be an area for it again, right?
Yeah.
Yeah.
At least
Because
it was,
that is
1,000
one of
meters.
that is one of the sponges that
Yep.
do like to live on sedimented
indeed.
areas. Oh, but at least there are some. When we come closer, we see the
Jellyfish again.
stocked crinoids.
Mm-hmm.
Then there are these little things that looks like balls on a stick as well.
Yeah. If you see any of those, just let me know. I will try to.
I think I get them
Zoom
mostly
in.
down here at the bottom of our picture because further up they are so invisible. That's a swimming thing. What's that?
Do you want me to
No.
to say sponge?
So,
I wanted you to say sponge. We have to, every now and then I have to hear the word sponge.
This may be potential candidate for you now.
You know that my life on land is going to be very empty of sponges, so maybe can you just record it for me? I can have a recording, and then every day I can just play Sergi say sponge. That would be great.
It's gonna be in YouTube all these videos. Yeah, we still have to
Yeah.
fix this.
The next one.
How long for the tape to end?
We have
15
15 minutes more. Which fits very well with the
I
time.
think
as
they're
well,
gonna
yeah.
get there more or less at this time when
Yep.
the tape finishes. And there's gonna be a shift coming.
Mhm. There is something on the seafloor.
Is that a what? I don't know. Rock?
A rock with some, or maybe just another piece of sediment that is thrown up a little bit.
Are you in a hurry?
Hey, Robert. I think this
This could be, yeah.
Stilo Cordula, right?
Look,
And
this is the...
this is the one. Can we just take a photograph? Oh no, too late.
Vessel point. So you're jumping between the and the vessel. It's not a big issue, but just for you to know if there is any way to just keep it
it's the range of feet.
Yeah, but I think I will write question marks to the Cordula.
it goes for the vessel. Yeah. Yeah, just for you to know.
We have noticed it before, but so it's not smooth as the other one's feet. Then we
What time is it?
at the end.
10 to 8.
Fix that and smooth it out. It's absolutely
Well, that was the air conditioning kicking in again.
Yeah.
We're sitting right under the air conditioning, so and there has to be an air conditioning in this shack because there is a lot of computers and electronics in this shack, so we need to cool it down. But it's not always super comfortable sitting right under the air conditioner, so that's why we are dressing up in a lot of clothes in here.
Wearing my jacket, yeah.
I don't know, not have a windbreaker on me, so I all of a sudden felt that cold wind down my back. But if we concentrate on the beautiful animals, we don't feel the cold.
Look, this is the jelly, right?
Is that the jelly?
Yeah. I
Yeah,
think so.
let me get ready with the
I
camera.
don't know whether it's gonna be
It is.
far.
It's coming? Yeah,
Yeah, there'll be other
there will be other.
ones.
It might of course seem a little bit silly to sit here and use our phones to snap the screen, but that's to just have a quick image until we can look back at those films that we're making now. So we are of course, in addition to sending this live stream for scientific analysis, we are saving the film so that we can bring it to land and analyze it. So it's not just going out there to YouTube, but also to our large computer disks. It's a shrimp hanging out. Hi, shrimp.
Something I can try upstairs. I'll try increase the interrogation rate. That is the water depth as well. If we had it at 2 seconds, sometimes it's enough time for it to come back. So set to 4 seconds now. 3 seconds should cover it. And if it's just a slightly faster interrogation, it will be smoothing that out.
There is a little rock being a black shadow, and then we see the sedimentation different on the two sides of it. So there must be a little bit of a current also all the way down to the seafloor here.
4 seconds. 4 seconds.
That looks like a little burrow again, right? Yeah.
There's only one beacon that's going to go with that anchor. It should be the 2.03. So
The responder one, okay.
yeah. I'll try 3 seconds and see if it's compatible with
Okay.
that.
Thank you, Robert.
We're steadily getting closer to the starting point of the next transect, and then we will have a little bit more of a normal slowdown and flying. But right now we're
That's a
flying
fish.
over a beautiful little fish, and we're seeing quite a lot of those little red jellies.
Not to altitude now, because otherwise I have no command and look at downs on
And
I'll take them off and see.
I would say I just got an answer from a colleague
Mm-hmm.
who, let's
Just 10 minutes
see,
for the tape to be
yes, who says that
jelly.
yes, the jelly is Crossota sp, maybe a Crossota norvegica.
Use the last doing shipwrecks.
And he asks for a picture, so I have to try to get a picture. I will try to get. It's very nice to have colleagues who can help you on the fly with these things. So thank you, Joanne. Joanne did that, yes, and
Joanne.
I am going to send you a picture, Joanne, if you are also listening in, but on email now, from Bergen to the ship, helping us trying to identify things we're looking at. So a Crossota, yeah, Crossota. Maybe a Crossota norvegica.
Sense, norvegica makes sense.
Well, we're in the Norwegian Sea, so I think it's okay that some of the species are called norvegica here.
It's a solitary sponge.
So, but maybe there is a tiny little solitary rock right under it.
She made it there.
Yay! Found a hard floor thing to sit on and then poof, go on.
Who is the one that you're
Right, so I'm going to
to the start of the line and
Yes.
then
I think Julio's coming and friends, so we come.
Yes, I think we will have a handover right in time to start on the next transect. So, did you already put on lots of warm clothes? You are ready with the warm clothes.
It's super cold.
It is.
Yeah. Well, it was just that the air conditioner just kicked in, making sure that we're not heating up the shack too much. What is that?
Did
Yeah,
you collect something?
Yes.
we collected this right at the very beginning. Mostly. There was a rock with this small anomaly. Okay. We have some sponges attached. I don't know. I think it's that when we try to take the rock.
Disintegrated. Yeah.
Break the things.
Yeah.
Okay.
And then on this one there is complicated.
But there is some sponges.
But we were also after doing this sampling of these big sponges, it's convoluted thick sponges.
Have you done it?
No, because we thought we can have plenty of time in the next spot. We just wanted to go to the next
It makes sense.
Bridge RV.
Go ahead.
So you can go ahead with this.
Can we reduce the speed to 0.2?
So we wanted to collect this.
There are some really obvious
white
burrows
one,
here.
but there was no white one.
Yeah.
Okay,
Took a
thanks.
video of one of those, super beautiful.
The white sister.
We didn't find this, the big flat one.
Yeah.
It wasn't there.
So if we find it, should we collect it?
Do you think? Yeah, no?
Mm.
Yeah, because we want to characterize everything, all the most common organisms in here.
There's some
But we're coming up to another
Yeah.
rocky little place.
Yeah, there's a lollipop there too.
Yeah. I will.
No, this is the
And then
correct
not-
position. So
Look.
This is
for
the
angle of
Oh, yeah.
jelly again.
I will sit down and make a photo for you, Jean.
Uh-huh.
person to
Um,
fix the feed.
There's
there was a
this
fish.
We,
Oh, there is an amphipod sitting on a stalk that we just another fish. Oh yeah. If you see these stalks with amphipods on them, then we really like to have
Rocks.
like sticks sticking out, right?
Yeah.
Then we would really like to suck up the amphipods.
yeah. Hi, Francine.
Have you seen this? Like a little crustacean, una arañita, like a little spider, super small. It was a close-up before.
I
yeah.
think there was something.
5 minutes to
We were
tape
taking a
change.
close-up in the
It's super
Sea
small,
star.
super small,
like the right-hand side, yeah.
This we don't want.
Well, it's too small. Yeah, you find it.
Nothing is too small. My average size of my amphipods is 3 millimeters.
So for
My
those
word, still.
amphipods,
305
Yes. Really, I mean, there is a lot of beauty in the small animals.
Or 300.
How full are the sample boxes?
Not super full.
You still have some.
You have some space still to fill in.
And also for the other two.
3,
Yeah.
4. This is for the mug.
Yeah, that's the mug, so that is unused, but so we have now filled in here. And that's what we have filled in. And we filmed sea pig.
Yes, I saw that. And
Yeah.
there was also a little sea star you flew.
In
It was
here
like
you
a
can just use.
really cute
It was the
conglomeration.
echinoderm heaven little minute there.
Yeah. Yeah, really cool discoveries. Also the sea star that was on top of the prime.
Yeah,
Right,
that was
yeah.
cute. That was breakfast going on.
Mhm.
Okay. So we are really getting close to the start of the transect, and I think it will fit very well with the change of shift since Julio and Francie have come to take over from Sergi and me.
So
And they have dressed warmly because they're sitting here in this air-conditioned wind,
How much?
and at the same time we can get the next transit started. Everything will be good.
So
Yeah, there is another of those jellyfish.
I'd like to start on transit.
Yes, so it will be actually quite perfect. We hand over, you start to transect, and then let's see if we get to do another hop and fly or not.
yeah, this information needs to be filled.
Yes, we need to fill, so that was the start, we had the start already there, so we just fill in the end and then we can, could you fill in the start of the transect one? No, we didn't.
I did.
In here.
No, I did not.
Yeah.
But
you can,
you have
you
it
can
there,
copy it
yeah.
from there.
So let's just fill in the end of the transect.
Yeah, it's here.
Yeah, we didn't do that,
We haven't written it yet.
but we have written it here.
Well, you have written it on the
At the
on
end.
this, right? I guess.
When
Yes.
we start runs, no,
So for the start is here.
start here. Okay, so we have the coordinate.
You can use that one.
We have been a little bit excited today, so we have forgotten to take all of the
Yeah,
details we should have. We have
There are a lot
quite a
of
lot
details.
of different sheets we need to fill out details on, but let's now at least start on this T2 just to make sure that we get that one correct. And then we can find the information from the, because we have it in the computer for all of the others.
We
T2.
don't get to the other side.
Yeah.
We don't checkpoint. Bridge, are we?
Go ahead.
Okay, can we reduce the speed to 0.1?
Wait, what?
1 minute until tape change.
Somewhere in the middle of that red band.
Just to
More in the hedge of the orange band. If he was jumping between your position and his position, that's why he does that drawings. So you are on that edge.
See, they are really making a little line there. Maybe it's a good place for the water currents to come. Okay, 4 seconds. Yeah. Change tape.
what is the coordinates?
Okay, recording again.
Recording.
Can you tell me the coordinates? There you go.
Just the last one.
Then we will
Can you
let.
tell me the coordinates? I cannot see them.
Yes, I can write them down.
Yeah, sorry.
And then we will let Sergi and Francie take over. Are we here? Yes.
Sure. That was it. Yes.
Okay.
So it's 4 for the and 3 for the
Okay. We are letting Francie and Julio take over and enjoy the next shift. Changing places in this is quite challenging.
Yes.
Bridge Arvin, can we change the direction of this movement to 010? Okay, thanks.
Alright, hello. Julio and Francie are back again.
See you soon.
And ready to start a new transit here on Mohns Treasure. Thank you. See ya.
Okay. Yeah.
So here the line is just to have an idea.
Okay, let's see what the next transect has in store for us. We're gonna go further up on the slope. What are we now? 2,700 meters. We're gonna climb further up.
You prefer to go
We will have some
now?
rocky areas and some.
back scattering was showing more to the right.
I'm also at the same time listening to them. And we also had some more softer sediments in between for sure, as we also have here in front of us.
This way, and then let's see what's going on.
But as we have seen also already with Annelien and Sergei,
and then let's see what's going on
there were some really beautiful discoveries that they had there. Yeah, let's see now. Oh, a sea star there in the distance.
Because the back scattering was showing harder here. So that's why I'm a little bit.
So we're going in, getting in the groove now and to start to find a spot for the new transect line.
Yeah.
Did you take the coordinates of this yet? Are we officially starting the transect?
No.
No. Not yet.
Very good.
Another sea star. On the search for some sponge flesh. Now we see some lonely sponges down there.
What is that?
Has a weird shape again. Huh. I think this is the place of the weird sponges here.
Yeah.
Shrimp?
Yes.
And we see also some of the stalked crinoids on the seabed. The feather stars, which we will have a closer look at one specimen later on when we finish this dive.
It's being recorded for the So my question is, should we try to put it correctly, or we just know that it's 60 degrees clockwise and keep going like this?
Can't do that. Correct.
Because all that is just sandbar. All that. Or
Probably, yes. So,
They will need this information to process anything. Like we can correct not.
yeah, I'd say so, yeah. There's no signs of really any rocks on the earth.
that's
Yeah.
all.
Yeah, that's a shadow behind it.
Yeah.
Just the one.
Yeah. But the rest is sand waves.
Well, if it's like downstairs,
So it's
So we see more rocky sediment and sponge communities coming up in the distance, where they have more substrate to attach to. And I think I see already there a Lisodendrix on a rock. Yeah, so any hard substrate
Yeah, those are it's
that we find are often already occupied by a lot of sessile or also mobile animals.
Oh, you can tell how important is the substrate.
Yeah.
In this image now we see where things are just every little rock that
Yeah.
appears from the sand is completely colonized.
So some are specialized, they need hard substrate to attach to, for example their larvae, to then grow into sponges and corals or similar life forms, and others are more adapted to also live in the soft sediment, to dig into it, dig burrows, dig through the sediment, also turbate the sediment through that, and so adapted to different surroundings. Are we at the start?
So we are probably here. Yeah, that's fine. Just keep going. And see that outcrop
In the middle of that orange roughly.
Yeah. So I think I should keep going in the same direction.
Shall we call it start of transect?
Yeah, let's call it the start of transect.
Yeah.
Yes. Thank you. Let's see.
Start of T2.
So this is 10. This is 3, 2, this is
And again, for the start of the transect, we're diligently noting down the coordinates and the depth where we are. So we're roughly at 73 degrees north and 7 degrees east, and almost 2,700 meters deep.
Yeah, look at that. Can you slow down a little bit?
Oh,
Yeah.
Julia spotted sponges.
Well,
Interesting sponges.
Is this a carnivorous sponge?
This with the many branches, twigs.
Yes, it is. Look at that nice rock.
Or is it? So it's not a hydrozoa.
This is a test, huh?
Just checking. Maybe how you would tell the difference.
No, I think it's a carnivorous
we would need
sponge,
higher
but
resolution.
I'm not sure if I have an answer.
Okay.
If I had it with my hands, then yes. But
Ooh, and nice fans.
yes, nice plants. These are endorids.
Yeah.
There was something orange over there, but it's too late. And then let's try and check out. So that's the rocks coming there. So yeah, this is again the same community with Actinellidae, these are endorids, some Hemigidius, Arcopherus from time to time, and plenty of little encrusting sponges, shrimps. I don't see Rosellidae, which is interesting.
We've seen Rosellidae at the similar depth at the other side,
Yeah.
quite a lot.
Yes, but not now, but maybe, who knows? Maybe means nothing. Let's see.
And Rosellidae is one of the glass sponges, but we also see other glass.
Rosellidae, sorry, Asconema. I have so many things in my mind. Asconema.
That we don't see Asconema at the moment.
Now we don't see Asconema.
Yes,
That's
but
it,
also not Rosellidae.
No, not Rosellidae, not in the whole dive, sorry. Rosellidae is more shallow. So, Asconema.
Yeah.
What was I saying? Rosellidae all the time?
For this part, yes. But
Okay, sorry.
I also thought we saw Rosellidae also at a higher depth, but no, they're in shallower water, so it's no surprise that we don't see them here.
Yeah, no, I don't think I have. This is nice.
Oh,
You
that's
see
an
this rocky outcrop? Yeah.
Mhm.
Like,
Yeah, some lava rocks here. Because we're still at the inactive vent site called Mohns Treasure. That was, once an active vent, hydrothermal vent area. And in some in the first transect we saw also the mineral rocks. Our geologists would probably really laugh at that expression, but yeah, we could see the metallic crusts.
You see that rock?
And that's really
Is
interesting.
it possible
here
to
as
stop here just to have a nice close up of that? Francie, what do you think? This,
Yeah, it's a nice
this
rock
and this.
wall.
Maybe we
Yeah.
can then
Wow, there's a lot of
just
sponges.
have a view.
Yeah.
Come across again. Use my camera, right? Turn my camera on.
Bridge RV, can we come to a stop?
It's also like like a little shelter
Yes.
building
Nice to
something.
It's a cozy spot.
Mm-hmm.
It reminds me to the Lord of the Rings. You know when the Nazgûl are hunting Aragorn and the Hobbits.
So we're going to land in there,
I
or
don't
not?
have that scene in mind.
No?
No.
That's the rock you want.
Sorry,
Yeah. And we can see nicely again the areas where they cling most on to the walls, where the current is probably best so that they can
Exactly.
get their food out of the water.
I wonder if this is a rock or something that keeps going up.
If it's also maybe an old chimney structure,
Yes, well,
so
I don't
part
know.
of it.
But, yeah. So I wonder if we are at the summit or if we could keep going up.
It's not really clear to see.
are you going to want me to land here?
Yeah. And we are not collecting anything, just to have an overview.
Can you pan around when you to the left of the camera, please? I need to see how close I am to the rock. Thank you.
Like that.
So we're going to stop here to look at this beautiful rock wall and the other rocks that we see here and all the life that is on them. So the pilots are looking closely also at their other cameras and so for example the camera that is below the raft to see the surface where they want to land upon and make sure that everything is well suitable for that.
Now wait a little bit till it settles. Brilliant.
So let's have a closer look at these rocks.
Something going on here.
Mm-hmm. Yeah, we have an interesting community. We have many different
This one to the right, the little tube. Okay.
Asconema? No?
It looks like, but it's a little bit different to me.
Why?
Well, it's smaller and the shape seems different, but it can be just because it's growing in a different thing.
What did they say?
Beautiful bowl.
Beautiful boulders.
Nice comment from the colleagues. Beautiful boulder maybe means we should turn the lasers off.
Yeah, can we turn the lasers off, please?
Oh,
Thank you.
Thank you.
So do you think that's a carnivorous sponge, the branchy thing next to the Lysodendrix?
Yes, maybe. Would be good to see.
It would have a lot of.
It's hunting.
So that would mean it has a lot of small hairs, or even like little sticky balls
Yeah.
on its little twigs branches.
which way it goes.
So the spicules project also, and the spicules, so the skeletal elements that the sponges have, microscopical silica, glass spines, help the sponge also to capture the prey, together with the mucus on the surface. So it's possible to see them hunting if you are patient, if you have enough patience. But yeah, maybe we don't have that much time. So
And what would we see then?
you would see just a little crustacean being trapped by the
Like a little amphipod?
or an amphipod, yeah, being trapped by the skin. Sticky surface.
And did you mean these sponges as cauliflower sponges before, or was that a different one?
Not really that one. A different morphology that I saw before, which was a little bit bigger, thicker, and with more like protuberances, more irregular shape. And this one, yeah, that's one that I'm not so
It
sure
has
that
an-
is.
No, is it a shrimp sitting on it?
It's just a shrimp. That one?
Yeah,
I think
or is
it's
it?
yeah, I think it's the tail of a shrimp.
Oh yeah, looks funny. Looks as if it's like an enemy is sticking out of it.
So one of those is our potential new species. I think that you see the tail of the shrimp. The tail of the
Yeah,
shrimp
yeah, you
just
can
behind.
also, yeah, I can see some of the antennae of the shrimp, I think, as well, sticking out. Yeah,
So just behind the tail, that's the new species. I think.
The
I'm
little
not
ball.
sure. Yeah.
Yeah.
Not sure. Also here in the middle you have this one that is to the right of the Lysodendrix. That
Yeah.
one that is smooth without the hole that we can see. That one I think is also our potential new species. Always with caution. Still a lot to do.
and why do you think the big ones look different than
No, I just thought, they feel a little bit different. It can be just the same.
Okay.
It's maybe because the way, the place they are growing. Do you want, Fancy, us to try to turn one of the lights off?
Oh yes. Oh, I've never been down in the shack where we turned the lights off. That's
So maybe
exciting.
Daniel wants us to turn the right or the left off.
Of the lights.
Yeah, because we can choose it. So well, let's
Maybe.
try it. Let's try.
Yeah.
Can we turn one of the lights off?
Yes, certainly. Which,
Uh,
do you mean to get that effect again, like
Left
last time?
one?
Yeah, maybe turn the left one.
Yeah. And then we can also try to turn both off afterwards.
Yeah, maybe we can see some luminescence.
Yeah.
Oh, the more often, bring one slowly.
Oh, check, dry lab.
Oh,
That sounds good.
Yep. Okay, copy that, thank you.
thank you.
Oh, we could just put them all off. and then
Our
slowly to get that
visual
effect, yeah.
colleagues approve as well.
Yeah.
Easier putting them all off and then switching one on, whatever one you like, we can make a note.
That works as well, yeah.
We also have LED lights on the T4 as well if you want Antonio's done it before with
LED, LED, okay.
yeah, so you can move the arm to a certain point and then you can sort of light up areas just with the arm.
Can we do that?
Yeah, you can try it.
Here you can see all the fan shapes to the left and all the other ones to the right. So current is probably coming from the left.
Yeah.
And here is like, do you want to compete for substrate or do you want to?
Ooh.
Look at that.
Okay. Oh, it's sparkling.
We still have some light. Now, no light. Maybe bioluminescence. You see all the bioluminescence?
Yes. So not sure if you, the viewers out there, can also see the bioluminescence here, because maybe you don't have such a dark surrounding as we do here in the shack, but we see a lot of sparkling everywhere, like little stars
Is it?
twinkling. Yes.
Is it? Huh?
And that's the bioluminescence of a lot of small little organisms everywhere. So yeah, even though there's no sunlight in the deep sea, it's far from being completely black without light, because a lot of organisms, I think most of them, at least more than the majority, it's thought that more than half of it has the ability to create bioluminescence.
Super cool.
Yeah.
Looks like the sky with the stars.
Just more short-term stars.
Yes.
Ooh, wow! What? Now we see more.
Wait. Something going on over there?
Yeah, those the sponges.
Ah, it's Antonia.
I'm turning on lights from
Ah.
the hang of the ROV.
Which one?
This hard.
Ah, okay. So now we have the arm,
look at that.
and oh wow, we have the arm coming of the ROV with its light on. Wow, what a stage!
We try with only one of the lights.
This one's, when things are on the seabed, this one's quite good. Antonio's got pictures.
Yeah, it's beautiful.
Just the seabeds.
Yeah, so if something was like sticking up from the seabed, you can sort of light up and create shadow.
Oh, great one.
Okay. That looks really good.
I
Yeah,
can show you some pictures.
yeah, yeah.
Now
happily.
Try the
Yeah, it looks like a stage setting. I think here, like the spotlight is just on now the main actor here, the rock, the spongy rock.
Port LED.
Yeah.
I think it was
It's
maybe
amazing what this
4
ROV can do.
LEDs or something.
And what the ROV pilots can do. Oh, so many light
But
effects.
then maybe we can turn the arm off.
Yeah.
Yeah, it was either one of them or maybe try the port Botec.
Uh-huh. Yeah. Can we put the arm?
Open the camera on.
So we need to rotate up.
Well,
There you go, you got a few more.
just one second. That's me.
The immersive cameras in?
Send you.
Yeah.
Can we collect this rock?
Yeah. You got 2 arms. We can target.
So we will put our immersive camera out as well to capture this view in a 180-degree format as well.
You can try different lights also if you want. You want to put this one off, try a different one.
Let's do that. Let's try with the right one.
Right one, yes. So left one off, just right one. Maybe one of the inners or something like that.
Yeah.
Try to play with this once again.
So we're playing around a bit here with the different light possibilities we have to put the scenery in different perspectives or in different
You need to try another light.
Hold on
illuminations.
1 second, because I'm trying to close the iris, but no, nothing happens, because then if you close too much, so yeah, no.
You can look to the lights and see if
Hold on a bit. Do we have more combinations of lights?
Yeah, you can do. So I'm going to put this one off.
Yeah, that's sponge.
Ah, yeah. I tilt it over, yeah.
And then go. We'll try that one yet. So we'll see this one.
That's nice, huh? This is beautiful.
Sorry?
This is a beautiful one.
That one?
I like this one.
You like
yeah.
that one, yes?
Yeah.
Me too.
That's the makeup.
Yeah.
So I can dim it if you wish.
Sorry?
I can dim it if you want.
Oh, that's right. Yeah.
Yeah. So,
Uh-huh.
that's
Too much. Yeah.
borderline there.
Rorschach dry lab. Can we push out the sextant and try and get the same from that one?
Yeah, so
The sexton's out at the moment, so you can have a look.
Julio in front.
Thank you.
Just that's to the right or to the left. There are
Oh,
more.
cool. We're now getting also some pictures of some past dives and how the pilots illuminated different animals of different structures on the seabed. Ooh.
To get more
really
light
cool.
on that. As in,
That's maybe for Jason to say. Jason, do you want more light? Jason, do you want more light?
You can bump it up a little, please, yeah.
So we're also consulting here a little bit with
okay, that's
our
maximum
visual expert
on that
colleagues.
light.
yeah, that one's with the
Very nice. Okay. When you say fancy, I think
Yeah.
for me.
Yeah. We can move on.
Move on?
Mm-hmm.
Guys, we move on? What do you think, sir? Shall we move on?
Yes, we can move on. Yeah.
Let's move on. Thank you.
Okay.
Yeah.
Thank you very much.
Thank you.
you just switch on the starboard and it comes away.
Oh, it's just the starboard and then go.
It's an inner. One of the inner ones you just turned on there, I think I was a lot of lost.
Oh, that's different. So that's a pan. So would it be that one? That one's just come on it?
I think it was one of the inners.
So we're getting ready
that
to move
direction.
on with the transect
So you wouldn't do the aft.
and the little light show that we put on here.
Yeah, he's on with the light that we used last time on the bridge.
starboard or starboard?
That was this time. I think the last time was maybe one of the inner lights, and it was like a stage kind of effect.
Yeah, I saw that. You want to try the last effect?
Oh, okay. We have one more effect.
What are you?
Because there's one of these, as you just said, that has a couple of these on.
For making the stage effect from...
Yeah, exactly.
Ah, cool.
Just go on again.
Ooh, that's moody.
I think it was that one. No, it's the main code, but
No, it's not the main code, it's one of the inners.
right. I only go as far as this one when you said that, okay? So,
Is it pilot plan and tilt off, right? Because that's halogen.
Pilot plan two.
everything is off now, apart from yes, that's how pilot pan and tilt
Yeah,
down.
to guide off things.
So we're
But
just
when you
playing a
just said
little
that,
bit
you
more
knew exactly what
with
I worked
the light
on that.
options that we have here. Before we move on, there are still different combinations and possibilities.
So it must be an hour, is it?
So that's the one we just
Nice.
So that was the one, that one, or we'll have the starboard one also. That's the one we did the other day. More LED outer.
Yeah, so those are really like stage lights now.
You cannot move it, right? You cannot.
No,
it's like that.
No,
Yeah,
it's
yeah.
fixed.
We moved that a bit. Like
Yeah, we can move, I can move forward on the other.
We've got the starboard one as well. We want to switch that one off and try the starboard one.
Yeah, maybe the starboard one
Yes.
captures it better.
That's the rock.
Yeah.
Starboard LED outer.
Ooh, cool.
Now, that's beautiful. Yeah.
Nice.
Very nice. Let's wait for 10 seconds.
Yeah.
I will just zoom in a little bit.
Perfect.
I think it's better to go from the port.
Port is left. Port is right.
No, port is left.
Because I'm with the Spanish, I know what we were doing.
Port left, starboard right. So I think when it's coming from that side,
It's maybe better.
it seems more of a cone. So
Yeah, but I think we're good.
we can move forward on the RV.
Yeah.
If I put the port on.
Nah, it's fine. It's
Yeah.
fine. Do you
We
want
keep it in mind for another.
for another?
Yeah. But thank you very much. It's really good to know
Yeah.
what's possible.
That's all.
Yeah.
I'm going to store it in the section. Doing the sexton left out for now?
Yeah. Mm-hmm. That's okay
Obviously,
for
if we
you.
use
Yes.
them in nips, we're going to have to put it.
Yes. Yes.
Will these on?
All
Yeah,
lights
please.
back on. Shall we explore a little bit how long this goes on? The big rock?
So you see the big rock on the
Yes.
left? Can we try to climb that rock?
Go up over it? Yeah,
Yeah.
we can do that.
That's fine.
So obviously you're going to have to come off the seabed a few, probably about 2 or 3 meters. To clear this rock in front. So, what would you like me to do?
Just to fly over that rock up.
Go straight up and over.
Yeah.
Okay. Yeah.
So we're really intrigued by the rock wall here on our left, on our port side, and we will have a look how it continues and fly a little bit over it. What's port and starboard in Spanish?
Babor y estribor.
I didn't catch that.
Babor y
Babor.
Babor y estribor.
Estribor.
Yeah.
Okay. Babor, estribor.
Okay. Yeah.
So it has the summit there already. Yeah.
So it won't go up.
Do you want me to go further up?
Yeah.
Yeah. Okay. No, go further up.
Which direction would be north to continue our transit? Would it be the same?
Come over to the left.
Yeah, to the left, to the port. The thing is that I don't know if we should trust that. But north is not right.
Yeah. So we're just bringing the ROV in the position to follow the rocky structures more.
Okay. We went up and now we're going down.
Yeah. Oh, there was a jelly, I think, in the picture. Yeah. And we get a nice view of our immersive camera, which has a really cool structure built on the ROV, so that it can be hydraulically pushed out and pulled in again, so that when it's pulled in, it doesn't interfere with the sampling boxes and the manipulator arms that we have on the sides for sampling. So we can actually have both just by pressing some buttons to have it in and out.
Come higher above that?
The highest possible. So just want to climb to the top.
Is this one here, is it? Is it this one?
Yeah. I think so.
And then you lose view of it and go much higher.
I mean,
Where would you like it to go?
the thing is that if it's going up,
Yeah.
then just go up
Go up, yeah.
with the mountain. Yeah. Okay.
Not really sure, although. Nothing on the
While the ROV is brought into position to look at the wall, we can also see right now in the picture the left arm, the manipulator arm from the ROV that usually is looking just like the right arm as well, that could grab things, but now it's adapted to hold the hose, the slurp, with which we can sample differently than just with the manipulator. So we can suck some samples into and move the hose directly to the sample.
So it's this way then. round. Yeah.
Okay, there we go.
Place for over it.
If you can go closer to the bottom. Awesome. Thank you. So now we are going uphill, right?
Yeah.
That is not showing our position. It's not moving, it's steady, the map.
Sometimes it lags a little bit until it updates and then we can see the position where we are. So on next to us there are actually a lot of different screens showing different things, but on one of them is our map with the transect that we have drawn of the area and where we want to go.
Another movement.
And it updates in
Direction
between where the ROF is
40 at 0.
and how far we've come on this transect, for example. But sometimes it takes a bit of time for updating it. Ooh, wow, there are a lot of sponges coming up there.
Look at that.
Sorry, is that 340?
Look at that. Look at that.
Yes, that's it.
My God.
How many do you think?
I don't know. It's a lot. A lot. My God. Look at that.
Wow, there's a big fan here.
Is that come to port? To the left.
Okay, thanks.
Fort, yeah. A bit more.
Lots of sponges. Many with there are so many crinoids on them. I first thought it's sediment, all the brownish, but it's all crinoids. All feather stars sitting up on the sponges and making use of them.
Yes. This is awesome. Look at that. I'll zoom in a little bit.
Wow. And a lot of shrimps flying around as well. Oops, we came a bit too close to the wall. But as we learned, sponges are very, very hardcore, and they just regrow their body parts, and they they can also regrow on other hard substrates again.
So the one that we see that is a little bit bigger, I think that is Stellodorits. Flabellata.
Stellodorits?
Stellodorits, yeah.
Flabellata?
Flabellata, yeah, I think.
Nice.
So maybe we can use that name for the moment.
Flabellata.
We haven't collected.
Flabellata, why? Because it's so
flabellated shape. I think it's a type of shape. Can we go to the right now and follow the rest? What I like, yeah, you see how the community is structured with all the fan shapes on the right side facing the current.
Yeah, beautiful fans. Really spun wide open to increase their surface to the current and then collect all the delicious little food.
Bit more board. Sorry. Okay. Yeah, like that. Perfect. Perfect. Right in the middle. Yeah, great.
Yeah, we're going back to the kind of start of the line there as well,
Well, yeah, but we haven't seen this ridge before, so I think it's worth to keep going for a little while, not much.
So from this far above we get a really good overview also of this area and how the community is structured. With little rhododendrons in between and really big fan-formed sponges.
Bridge RV.
Does this one have a hole in it? It's not
Yes,
something
that's
in
maybe
Seesternen.
Can we change the direction of the
somebody
movement
hit that
to
one.
260?
Someone was munching a hole in the sponge.
Sister over there.
Oh yeah, that one. Guilty.
Okay, fine.
Shall we turn around?
Yeah.
And continue?
Okay, can we go back to the line?
Okay.
Perfect. So so we have followed all the crest of this little mount.
And now we have a valley coming up.
Now we have a valley, so it will be a little bit of a blue transiting now without seeing much.
So we can probably
maybe
go
we
up.
can speed up
Yeah.
a little bit. Could we speed up now until we get to the line?
To the line, yeah.
So now we're going in the direction of the transect that we have planned, and there's a valley coming up, and we're gonna increase the speed a little bit to go over it, because there's a few hundred meters of this transect still coming up, so we can cover some distance also by going a bit more through the blue over the valley.
And start moving ahead and then see how it looks and see how
So that was this crest? This
Yeah, I think that we did like this.
Yeah.
And now we are going again.
And then there's this valley, maybe 100,
We did.
200 meters.
But here, this can be done by blue transiting
Yeah.
if we believe that there is nothing interesting. So maybe we can explore a little bit
Yeah.
at some point, I don't
Yeah.
know. And then, so the other team will see this.
Yeah.
Because otherwise,
Yeah, so we're looking into a smart folder where we printed all of the transects and all of the profiles for the transects, so we know the structure of the seabed and when highs and lows are coming.
so yeah, we were at this mountain. This is 1 meter resolution, so that's why it's so detailed.
Really nice, yeah.
May I see the next one?
Yep.
Yeah, this is 25 meter resolution.
Yeah, you can spot the difference.
Oh wait, no, this is T3. This is T2. Actually, we have done this one.
What?
This is the one we have done.
Yeah.
Yeah, I thought we.
Ah, no.
No,
Yeah.
no, this is the next one. Yeah, sorry.
Cool.
just gonna left of that line because the right hand line was slightly off the right.
May I have a look at our sheets for the different bio boxes?
I guess.
page again and we need to change direction. Sorry.
Oh God. I'm glad that you've given me instructions.
We're laughing because we always need to let the bridge know when we change our directions.
Okay, we stop this objective now, so we are now changing direction again, sorry. The new direction would be 010 at 0.2 knots.
So we give them some work to do to steer. Yeah, because sometimes on these exploratory dives we think, oh, there's something interesting. Can you please stop here? And then it builds up because not only the ROV needs to stop, but at some point also the vessel and the direction needs to be changed not only by the ROV but also by the vessel, and then it builds up, and then we think like, oh, okay, actually we're done here, we want to go now in the other direction, and then yeah, that builds up on the chain.
So where are we now?
So that's the track that we're on. If we look at
We are here in the last orange point on the left edge.
so now we should be.
We are converging to light, and now we are here.
You can actually go a little bit more to the north if you want. Yeah.
It's whatever you prefer.
Yeah.
I can bring my head round more if you wish, or
You can just go like that.
right.
So what's gonna happen when we bring the ROV back up with all the samples and the bio boxes?
Yeah, we'll need to sample everything. So every species has its own sampling procedure. For the genetic analysis, we want really good quality. So we use a special preservative, which is called RNA later. Then we have vouchers, which is the whole specimen if possible. That's for the museum to be deposited. Both the Uppsala, at the Uppsala University, there's the Museum of Evolution, where Paco and myself work. And in Madrid, the National History Museum, which is where Sergi works. So vouchers will be deposited there. So everybody, all the researchers in the world that want to have access to those samples can ask for a loan actually and do research. So that's accessible for everybody. Some samples we frozen, and those are for natural products studies. What else? We also check the speckles of the sponges to identify. For that we take a piece of tissue and we digest the organic matter with bleach, and then we take the speckles, which are microscopical elements, structures with many different shapes and sizes.
Made of glass so that they remain when the ethanol has eaten up everything else.
When the bleach has eaten all the organic tissue, the speckles will still remain. And every species has its own specular set, let's say, the set of speckles that characterize that species. And that's what we use to identify the species, together with molecular markers, which are tiny pieces of DNA that also are used to characterize species, together with the morphology. What else do we do, Francie, when samples arrive? We take pictures of everything, we annotate everything. Yeah, that's pretty much it. We try to be as fast as possible so the tissue is not damaged.
And there's not too much degradation already happened, especially also for RNA or genetic analysis.
Well, we are not working with RNA, but yeah, because we may want to have genomes of some of those species, then you need really high quality DNA, maybe. So yeah, all that. So that's the bottom, right?
Looks like.
Shall we get closer to the bottom and check out what's in there for a while?
Yeah.
Can we get closer? Yeah.
Just a little bit.
How
Looks
close?
like we have some
As close as
Some
you are
fan
comfortable.
sponges and
Just
some
with a bit of a bounce. 'cause I
of
don't
the
think
stock
they'll have-
crinoids
If
possibly.
they're half a metre, it could stick me strong to the sea there.
Half a meter. Or
Yeah,
it's
well, yeah.
it's too close. No,
Yeah, with a bounce, it could.
not too.
So try and keep it on about metre.
And a lot of little
That's fine.
critters that we cannot see even if we move much closer because they're either burrowed or dug into the sediment, living below it, living just above it, but being very, very small.
Oh, like,
But
yeah,
the
no,
accumulation
we don't
of
want that.
all the small animals that live like over a vast amount of these spaces in there, they accumulate to actually a really big and important part of the whole
Yeah.
ecosystem. So
As close as you feel comfortable.
yeah,
No
even
worries.
though
I
we
think
don't
that we will not
see that much, it
do this
doesn't
for a
mean
while,
there's
but
nothing
if
there.
it's just mad, then
Um,
go to the next spot.
Yeah. So, for example, there are different worms and mollusks in the seabed or on the seabed, and they cause bioturbation, so they mix the sediment through. And there are little crustaceans also living in or on the sediment. For example, little amphipods, or also isopods.
So that's
Yes.
So it's more or less 72, 73. Sorry, I'm trying to annotate the coordinates. More
Yep.
or less 27, 26. 26 close.
And also the marine snow, so all the organic matter that is falling from high above from the surface, ultimately to the sea floor, or carbon that is sequestered from the water in the seabed, is also playing an important. That's also playing an important role in the whole cycling of, the nutrient and chemical cycling of the oceans. So yeah, there are a lot of different important functions that we cannot see directly. But that's why it's also important to study them, because it's not visible at first glance what like the different interactions in the ecosystems are, and what one thing causes for the other, or why this is important for that. And to understand also then rippling effects that might build up when you disturb the system by taking maybe just one piece of a chain out, and what it causes for different effects then.
What's that?
I don't know whether it's little crinoids or anemones.
Yeah,
So
I don't know.
I was too
We can speed up a little bit.
Speed up, yeah?
A little bit, yeah. Fancy.
Yeah,
Shall we?
sounds good. Oh, there's a nice jelly coming up. Beautiful and not antennae, but
Right.
looks like little alien.
Is it speed up here or go faster? It's entirely up to you. Go faster.
We have a lot of jellies coming up. With the pulsating
That's what,
Buddies.
0.2, 0.4. Yeah.
Bridge, can we speed up to 0.4 for now?
It's too fast, fancy. 0.4.
I think it's okay, we can try.
Yeah, the vessel seems to be going across the line. We are definitely 10 degrees.
Yes. Yeah, we're ahead of the vessel as well.
Bridge RV, can we just adjust the heading 10 degrees to 0, 10, I think?
So we're
you were
synchronizing
doing-
again,
can you-
so
can
that
you tell me
our
what
speed
was
and the
the
vessel
direction
speed
that we were doing?
are adding up,
Heading
and not someone is
010.
running away from the other.
Okay, so can we go for 020?
Just ask if an LM because it feels like there are
Okay, thanks.
little snails there. So
Oh.
I will just ask Sergio.
Oh, it's just really good over. I mean, it's just up behind us.
Is that a sponge
Too long to
there? Yeah, I think so.
Yeah, that's one of those.
It's like when you do that
That's in
handful,
there.
even though you don't. And gold.
Where did you see the snails? Maybe the white?
I'm not sure it was a snail. It was kind of little thing.
Auto altitude.
But it was super small. So I don't know. I don't know what it was. You know, maybe it was nothing.
Would anyone like a drink?
I'm all right, boss.
I'm thank you.
I'll get up for breakfast.
Some holes, burrows, maybe? Looks like someone was digging there. Can you try to zoom in on this one that's coming up?
We also have changed direction of the vessel from 010
one
to
of-
020.
One of the stalked sponges? Ah,
Beautiful. That looks like a sponge to me.
yeah. Just has a little fan.
Could be. It could be a tenea. Could be
Tenea?
a tenea with the parathlanthus. Yeah, it looks like, but I'm not sure. We should collect to be sure. Can show Paco later.
Yeah.
I think
And we
there
have a
is
little
only
seastern.
one point of the laser.
Alright.
Oh.
It's maybe tilt up a wee bit. It's maybe caught somewhere.
Oh yeah, it's there.
Ah, it's in
Yeah.
the.
Go on the 16, push back in.
I don't know. That's for you
Hmm?
to say.
What? No, we have the laser back, so I think just leave it out for now. Thanks. Yeah.
That's one of those crinoids colonized by sponges.
Tilted over because they were too heavy.
Maybe it's that. Oh yeah.
Those suckers.
So we may just want to do transiting. Transit. What do you think?
Mm-hmm. Yeah, let's continue.
Yeah, good. Speed
Uh,
up.
We can speed up a little bit, yeah. If that's fine for you.
Yeah, we're
Are we
in a much
catching up?
the boat here, yeah.
Yeah.
That's okay.
Because
Otherwise,
the depth as well, there's a lot of
ah,
leaves, so the faster I go, the
okay.
more it'll bounce.
Okay.
then we stay like this.
Just like
Yeah.
this.
What are we at? At what speed are we at?
0.4.
Oh yeah. another of the jellies coming up. Yeah, cool.
Yeah,
I don't
no
think
idea.
so.
Species or the...
It's not a sponge.
No, it's not.
It's well,
Nidarian.
and another little jelly. Wow. Wee. The mantle was nice to see.
So there's an Entdecke there on the rock with the encrusted fence.
And a big sponge coming up.
Yeah.
Yeah, build a nice fan around the sponge, around the rock.
I think it's an Entdecke there. Yeah, you see that has a stem?
Yeah. Yeah, little stem, and then it widens already.
Yeah, and
very
ask
much.
on him over there.
Yeah. Oh yeah.
But this Entdecke name may change in the coming days.
In the
Now-
coming days.
Yeah, now that we have a sample, because we haven't had a sample of this before, so we are only based on previous paper by other researchers.
Oh, there's a little
But we
shrimp
want to double check that.
sitting in the fan. Yep. So you want to double check if this is indeed
Exactly.
Entdecke.
Yes. This is a provisional name for the moment.
Yeah.
There's harder looking stuff over the left. Do you want me to go more to the port or?
Yeah.
Yeah.
Report this.
Thank you. So there's another big rock coming up, where we can already see that the sponges like it.
And you see how all of them are in one side.
Yeah. Again,
Located.
And the Lysodendrix again more to the other, and the fans
Yeah.
more
So
the fan structure.
probably
to this.
Lysodendrix is not interested in compete with the fan-shaped.
Big.
It's okay with being where there's not so much food.
Because they can do whatever, like they can live with whatever from what we've seen now.
Probably, yeah. So that's the way. So they don't have to compete. They both get different adaptations,
Yeah.
and they're both happy.
And we also see a lot of sedimentation on this rock. Ooh, that's a huge one.
Looks like a Stellodorix, the huge one, but I'm not sure.
Like
I'm
what?
not sure.
Stellodorix?
Stellodorix, yeah, but I'm not sure.
Yeah. Ooh, sandstone.
Bridge Alvi.
Now we get a nice overview.
Go ahead.
Okay, can we do a movement direction west? 270 for 40 meters.
Too far.
40 meters,
Too far from the desert.
0.
That's okay.
This is.
Okay, thanks.
Jump, is it? Just carry on again up the line.
Yeah.
Shall we? Yeah.
Please. Thank you.
I'll hop over this and then come back.
Oh yeah, two of them. Yeah, we see two little fish resting. Maybe also feeding.
Just chilling.
Yeah. Oh, not chilling anymore. Chilling somewhere else. Oh, and another little one. So we're off into the blue. Do you have a favorite sponge?
No, I never thought about that.
You don't prefer one over the other? You like them all the same?
Yeah, I mean, yes.
It's usually got these four inches going on again.
05, 03.
44, yeah.
It's just really nothing. So,
Euro 15 at 0.4.
Okay, thanks.
So while we're moving, we can see a lot of little animals also flying through the water column, from little crustaceans and different jelly, cnidarians. Yeah, all of these mobile animals. And a lot of smaller ones that we can barely see.
fish.
Yep, well spotted.
That's our friend from before.
Trying to chill here.
Yeah.
Ah, damn it.
Find a place
They
to
found me.
pick it up.
Another jelly coming towards us. little ball. And a sponge.
Sponge.
Fish trying to chill. Is it an eel pout, I think? Because of its eel-like body structure. I think. Oh.
Ah, that was nice.
Yeah, helmet. Some sponges there in the distance. That looked like a puppet. Oh yeah.
Community, this
like in a cross shape. Okay. Sorry, I listed in dogs. The complicated ones.
And the fine shape, I don't know what it is.
Yeah.
I don't know if it's Migilius or Acinellida. It's a little bit fun. I think that Acinellida is there, the little ones. But I think it's Migilius.
It also had the orange parts, right?
Yes. It's cloud.
Is it typical for deep sea sponges to be more white in comparison to other sponges?
Yeah, there is less color in the deep sea. All the pigments tend to disappear, not always, of course. So all the sponges that have photosynthetic symbionts, those can only live very slight.
Like little algae that
like
also...
algae, yeah, exactly. It's just like corals.
Yeah.
So those are in the upper part of the water column, and those are usually very colorful. But as soon as the sunlight disappears, then there's no point in having photosynthetic symbionts anymore. So, yeah.
So also similar to deep sea corals, which are also more
Exactly.
white because they also don't have the symbionts anymore. Yeah.
Exactly.
So what makes, because we also saw colors in some sponges that were, for example, yellow, or some of them are a bit orange. How do they get their
So
color?
depends on the color, but usually we say that bluish and orangish colors are acquired by feeding. So there's something that is orange, or that has an orange pigment, and the sponge eat that thing, and then the color molecule
Like a flamingo.
is transferred, yeah, to the skin.
What
What's
is that?
that?
Can we
Is it-
stop, please?
Is it wood? No,
It's plastic.
it's trash.
That's trash, yeah.
Oh,
Or,
warning,
that's
trash.
that's a piece of some ice.
Can we stop here? Or you already stopped, right? Yeah.
Right.
Maybe even settle down and have a closer
So
look.
plastic. It's 9.
Plastic in the deep sea. Maybe we can read what's written on it.
Warning.
Emergency firework
Warning.
Not
missile.
Oh,
hazardous intent. Oh my god. What is that?
We'll
what
stop
is
for
it?
a while? Or
Yes.
Yeah, that's a little red. Like,
It says
RV?
hazardous
Can we
material.
stop for a while?
Full stop.
Hazardous material
Okay, thanks.
of the. Oh, my neck.
hazardous material.
This resembles. I can't. Can we tilt the monitor?
But it sounds like something you don't want to touch. It's warning all the time. What do we do? Shall we take this off or?
What do you mean take this off?
You know, guys.
We'll just wait and check.
I mean, just to not leave the thing
Yeah.
on
It looks
the
empty.
bottom. It looks new.
Yeah, fairly recent.
Yeah.
Yeah, there's not much sediment on it.
Got a serial number as well.
Damage to
Something
the.
about hazardous. So.
Yeah.
It says explosive something at the bottom.
Explosive.
Explosive.
This is that's explosive.
Or ordnance?
Ordnance, be
What does
like,
that mean?
be you ain't like basically ammunition or something, like a shell from something, or maybe a training route.
Ordnance is explosives. basically.
Oh, okay. Right.
There's no gunpowder or
Yeah, the ChatGPT doesn't know very well. It's cylindrical,
Maybe military.
underwater instrument.
Try with that. Is that full zoom in or?
No.
Yeah, you can see it over so ChatGPT can pick up with that.
Disassemble sonoboy.
Ah.
Sonoboy.
So it's like from a boy? Hazardous material.
It's
Handling
I'll tell you
of the
what,
sonoboy.
that's the diplomats from the Navy for sonar boys.
For sonar what?
Yeah,
Sonar-
listen out. For sepsis, stuff,
Like testing of.
yeah, maybe for.
Mm.
So yeah, this is to detect submarines.
Yeah.
It's
Drawn by
detecting, yeah,
aircraft.
yeah. It's for detecting underwater.
They detect
is
the
it-
objects.
So if is it something you fire off and then the shell is left over? And that's the shell? Or how can you imagine it?
I don't know. We'll find out later.
Not for us to play with, I think.
No.
It looks fairly recent as well.
Yeah.
It's not colonized.
Come up with it.
So unfortunately we have to leave it here. Because yeah, we don't know what would happen, or it has a lot of warnings on it.
Too many warnings for
Yeah,
my taste.
but this is also showing us that
want me
not
to close?
even the deep sea at 2,700 meters is free from human impacts.
Can we collect this
Or
sponge
from
that is here?
remnants.
Joking.
You can't see the certain number of able to tell.
And that, yeah.
Yeah.
Anything that we throw in the water could ultimately end up here in at the very deep sea bottom, where it will just stay for a long time.
I'm good with it if you are.
Yes,
Okay,
me
so
too. Yes.
There is a gap in between.
Well, we have that case of months.
Yeah.
We just shut the morning as well.
Oh, good.
Yeah.
Thank you. Careful.
Not American. Which I called correctly. It's American, not the human.
That sponge also has a cool structure. It's like a bowl. And the sea star was on its way to the sponge.
So that means that there is a submarine around.
Normally our sonar goes a bit funny. There is.
That happened before.
Keep going for the same direction as speed.
Yes.
Yes,
Yeah.
thank you.
Which are we?
Hey,
Okay, we can continue with the previous movement. So direction was 015 at 0.4 knots.
It's a hard kind of target just ahead of us now as well.
Hard at bottom coming up?
Yeah, like a rocky outcrop or something.
Okay.
You go across and have a look.
Oh, good. Yeah, maybe we can slow down while we are coming closer. We have a lot of little lizardendrix, I think.
Yes. It's the same community. The lizardendrix away from the current, and then the community of fabulate sponges, Actinellidae, Emigilius, and potentially Stellolorids.
Mm-hmm. Always when it changes for the community or for the sediment.
I try to. Yes.
So not only the cost. You can see the little feet running through the water. So we should be here, I think. Now we're closing in on this
Yes.
hill and then coming to the last third of the transect. And then there'll be a steeper part.
Yeah, that would be interesting.
Mhm.
But I don't think that would be for us to do.
Let's speed up.
No,
No.
20 minutes left.
Yeah.
Another 20 degrees. Reach ROV. Can we change the direction to 025?
Very good, Philip.
Okay, thanks.
Are those just pieces of sponges or?
That those are liegt that are broken.
Ah.
You see that there's a stem.
I don't see the stem. Ah, yes, on the
yeah.
behind. Yeah. So tilted over.
But just a guess.
Yeah.
Maybe wrong. They look pretty much.
So sponges are, in general, like a very old phylum, but they're also themselves as individuals. They can also grow really, really old, right?
Hundreds of years. Some. Yes.
Hundreds or, and some even think up to thousands or more. That's not yet proven
It's
or
not-
not yet shown.
It's not that clear,
Yeah.
but it's not crazy to think about it. Yeah, but we are sure about some hundreds of years.
Yeah.
Study done by Teresa Morganti in Arctic, but more northern than this, in the seamount, with some Tetractinielida. I think that was Geodia. Yeah, I don't remember the exact numbers, but there were a couple of hundreds.
Have you also dated sponges?
No, I never done.
Like shown how old they are? Yes.
No, I haven't never done that. No, for that
Oh, that's fish.
you need techniques that I don't control at all. Let's see start.
Oh, yes. Oh, it's a very white and void of pigments.
What's this noise? Yeah, so this is probably one of those sponges, but hard to say so far.
One of which sponges?
Just porifera. Yeah. Just a sponge. I don't
but
know.
one that we don't know yet.
Probably one that we know, but I wasn't
Okay.
sure because it was
Yeah.
very fast and very far.
Oh, there's a nice stem sponge.
Yeah, that's
Oh,
nearly
is it?
there.
Oh, it's on a rock actually.
Yes.
It looked from afar. Ooh, and there we have a nice prototype of a rock covered on all edges by the sponges.
Yeah, it's always the same community. Keep happening, keep repeating.
So we're slowly closing in on a more rocky area and a quite steep climb. Yeah, on this area that we're right now in a wider sense. It's called Mohns Treasure along Mohns Ridge, and it's an inactive vent area. So before we see a nice little sponge.
I don't have to move the camera that much.
Yeah.
I move the camera that much. Sorry.
Following every little sponge. Yeah, so before we have already seen on our first transect, and also here in between lava rocks and also metallic crusts that show the volcanic activity that was present in this area and the active hydrothermal vent activity.
Okay,
yeah.
rocky crop.
Hi.
Can
So we slow down?
Yeah, yes.
Thank you.
Okay. So this is like beautiful. So you can just move along the crests up if it's possible.
Yeah, and we see some Asconema as well. Here
Yes.
behind the big fan
Yes.
above in the left corner. So they're also more orangey brownish in comparison to all the most other sponges we see. Overkill with sponges. So much to see.
Yeah, please.
Oh, there we have a nice, not tulip, but
Can we slow down to
nice bubble.
Yeah, we should collect that.
Do you know what it is?
No, that's why we should collect it.
But how?
With the vacuum, maybe.
Okay, thanks.
But we cannot settle. Can we?
It's okay to settle. Try to collect that one. It's a bit hard.
Free fly. How far is it for your camera?
So we've seen a really interesting sponge over there, the one with the stem and then the bulb.
So, yeah,
Almost like
it's too hard.
a poppy. And we don't know what it is, and it would be really interesting to sample this one and find out more about it, but we are not sure it's possible
Can
here
we stop for now?
because it's so rocky and the terrain is a bit difficult. And we could put it together with others, right? Yeah.
No problem.
Paco speaking. The tulip sponges are Amphidiscilla monae, not collected yet.
Can we? Paco, do you want me to collect it, or it's okay, or it's a well-known
Not
species?
necessarily, no.
Okay. So we don't need it. Yeah.
Thanks, Paco.
Thank you, Paco.
So Paco Carenas was tuning in and giving us some advice on the sponge that we were seeing. So which one was it? Yeah.
Amphidiscula.
Let's see.
That's an exactinelli again, that's a glass sponge.
Mm-hmm. So we leave it there and continue our exploration.
Carnivorous sponge.
Just fly straight over.
Yeah, can you go port
Yeah.
to the very edge?
Don't mind everybody.
Thank you. It's perfect. You can follow the reach.
There's quite a big shrimp flying up there. It's okay. Oh, this one really built a really bowly fan.
Yes.
So it's really increasing its surface and building like almost a funnel.
Which direction are we going now? North, northwest.
Yeah, northwest.
Okay, that's fine because we will find all that rocky area there.
Okay.
We don't need to follow the line anymore.
Okay.
It's better to follow the rocks.
So going up.
Yeah, going up.
What speed are we going at? Maybe we can decrease a little bit.
Can we decrease the speed a little bit?
Yeah. We'll be a little slow to start with until we get the vessel going as well.
Okay.
So going uphill with it, yes.
Oh, this one. Asconemas.
Lots of big Asconemas.
Another exactly near it. Lollipop. You see?
Ah, yeah. But looked similar also to the one that we just saw.
Yeah, it's the same, I think.
Yeah. And another one over there.
One over there.
Yeah.
North and then 270, or you want to do the other one?
You could probably step off.
Oh.
So now we don't care about anymore about the line. Yeah. Just going up and
Bridge
following the
ROV.
rocks.
Can we go north 30 meters at 0.2? Maybe increase to 0.3.
And Sergi is coming back. Hey.
How are you doing?
Good.
Beautiful.
Yes. It is beautiful.
Oh no, I
We-
think you have to change.
Almost 10 minutes. Yeah.
Wow.
So if you want to
If you don't want to
grab something else. We're almost at the end of the second transect as well.
Second one. Oh, wow. Then it's a long transit, right?
Yeah, but now it's the whole area that I think we should explore. All these like little hills.
Yeah.
Yeah.
So we sped up a little bit in between, but we have now an interesting rocky area. And yeah, after that you have a longer transit to the next transect.
Did you happen to collect this sponge that you were interested in? This convoluted big one? Is that the one on the left?
Yeah, no, it's not that one. That one I think is that Cinellide, which is the one that I collected at the very beginning. I think. I mean, all this would be good to collect a little bit more to be sure, but and then the cauliflower one, I think is Stella Loritz.
That one that you mentioned?
Yeah, I think it's the one that colored on the ROV too. I think, but we haven't collected.
So chambers as the bioboxes as well as before.
Oh yeah.
Yeah.
So I think we're gonna collect 2 more of those.
So we haven't collected anything, basically.
Yeah.
So big room for you.
So is this Carson sponge, the one that carpets the
This
rocks?
This one.
No, on top. You see on top, it's brown.
Top. Okay.
Yeah.
This one.
This would be crinoid. Ah.
Oh, this.
No, that would be just sediment, right? Or is there a difference?
Fading.
There, you mean? Or just here?
Yeah, that one. Second one.
Where? This one?
No, that's
this is
just
sediment.
sediment. Yeah.
Really?
Mhm. So we actually, do we have 2 shift changes already going on?
There's nothing over there. Can you see some points?
Yeah, but you can explore a little bit still at the end of this one because we have a lot of rocks around before the big blue transit.
Yeah.
Mm-hmm. 10 to 12? 10 to 12?
No, it's actually going to be 10 past, so if I go up at 12,
Oh, okay.
it's already 5 past.
Let's give you a little bit
Yeah,
of
we'll check with Daniel when he wants me to.
So Anne-Hélène and Sergio are already behind us, ready to take over for the next shift. And that will also be the time when we change the tape again in about 5 minutes. Have you seen the
current here. It's current here.
the trash, the from the
Yeah.
missile? Or
I think it was too big for you to get it into the firebox.
yeah, and it
Something like
was
50,
a bit,
60 centimeters.
yeah, and we were also not feeling comfortable
Yeah.
taking it, but also if
With all
there
the
would
warnings.
probably, yeah, would have probably been difficult.
So just follow the crest. Tricky to know where we are.
So where? About here.
Okay, so,
So,
I wonder.
5 minutes.
Okay, thanks.
So we're getting closer on the one hand to the end of our transect,
This is north, right?
but we also have a lot of rocky outcrops over here, so we're discussing a little bit to follow these for a little while and just explore those a little bit.
So we would be north. So yeah.
That's a nice direction. Like that.
Well, I'm fine.
This one.
So for the change, do you want us to stop around here, see this,
Yeah.
and then we do the change? Okay.
There's also a really nice big
Stop around.
Stay on, sit down.
Yeah, we'll look at this community over there.
ooh, beautiful fans of the sponges. Looks like that Asconema is like a little bit in front of the fans of the sponge and then behind, catching
kind of
catching away.
Competing.
Yeah, and it's sitting on top of sitting on top of a Lizodendrix?
I don't think so.
No.
Somebody's at the base, right? But there's some massive.
Yeah, and this one in the back on the right.
How big they are.
And
Paco,
a lot
are
of
you
crinoids.
around? Is this a Sacocalids? Paco is not there.
This is Carlophagus.
Galapagos, yeah. Galapagos, sorry. Sactinelids are on my...
Yeah, so many
I have described
crinoids.
those that are sitting on those.
On the crinoids, on the Calophacus
On the
or
Calophacus.
Phacus themselves, yeah.
It's soft well.
We can stay a little bit here.
Yeah, we can
Just
stay
to
here.
have a look.
So we are seeing a lot of feather stars on these kind of sponges, and Annelien also just said that she has also found a lot of amphipods, of the little crab animals, crustaceans on those sponges as well. So yeah, we're settling down at the moment for the handover, and will probably also be interesting to have a little bit further look at those.
It's soft here. I don't know. You'd have to sit down now. Come up and reapproach it. because of the ledge, it's just
keeps going.
thrusters are right at the height of the sediment there. Come up a bit. Yes.
I mean, we'll have 2 minutes until we change, so just wait.
Yeah.
So the terrain is not so easy to
Maybe
settle
we go
down
from-
because
yeah, then we don't
it's
see
slopey, and yeah,
When you're coming up, the thrusters are right at the height here, so it just.
so we have to take care that the ROV has a safe landing and a steady surface as well.
Maybe if you put in this side, the current is going that way. We can clean up.
I can drive it. I think it'll still. Still up there. Grease, is it? I think that bird caught in there before. It's quite a slope.
Shall we extend the tape a little bit, because it would be some 20 seconds until we should normally change it, or is it possible to do it?
Yeah, I mean,
Yeah.
when I self-destructive, that's what
No, exactly. No, just checking from your end if we should just wait a little bit to settle. No self-destruction. No? That's good. Okay, so from my end would be, I would be ready to change, but whenever you are. Yeah.
Okay, there we go.
Stopped.
There you
Yes.
go. There you go. Perfect.
That's a beautiful Galapagos.
Oh wow.
So
But I don't see any amphipods. Not
and
only
the other side.
Yeah.
was I describing it, but Annenina on the other shift was also part of the description. So really, it was a teamwork from the others on board.
It was one species.
One new species of amphipod.
And this is common,
We've only found it on the Galapagos so far.
but several times or only
Once.
Shack Dry Lab,
No,
could you
twice.
pull in the sextant, please?
would be good to find more.
Yeah, it's not bad.
And maybe we can also turn off the lasers, please. Thank you. All right, we're ready.
So we're handing over.
Thank you very much.
So that's Sergi and Anna Helena back in the shack, and Franzi and Julio are going out to have a well-deserved break.
Can we sample the Calophycus?
Is it? Do we have room in the?
Yeah, okay.
If we have room, I think.
Which one is it?
The big one.
Big one? Yeah.
Yes. Paco, we will do our best to see if we can sample the Calophycus.
Creep forward a little bit.
Thank you.
I would definitely wait a little bit until it's
Yes, but let's wait
clear,
until we have a
so
clear
have the-
picture first, because it's a beautiful
Beautiful.
sponge. And I will spend the time zooming in and looking for my amphipods. Or, sorry,
Go ahead.
mine and Annenina's amphipods. Because Annenina alerts on the other shift. And I described a species of amphipod that lives on this together a few years ago. But it's still kind of dusty.
Yeah.
And here I only see crinoids sitting on it, but it's a beautiful big sponge.
You want only a piece of the sponge, right? Not
Like, I don't know,
You
one
want-
of these, where is-
One with the headline.
yeah, one of those. Any of those that you feel it's not tricky. Yeah, let's wait a couple of minutes until it's clear. It's almost.
Oh, no, it's all there for pictures. Still a month of it here, so.
And since we maybe have still the specialists in the virtual room on this, Paco, do you have more than Calophacus for a species?
It's pretty good sponge. Huh?
There is no response.
No response. Then I guess we go with the genus Calophacus.
It's almost about to be clear, right? Do you think these are the same crinoids that we saw?
No, I
1,000
think they're
meters? I don't
different.
think so.
So I think we can also get one of the crinoids.
Because those were bigger. I see that they are like
They're curling in a
bending.
little. Yeah,
Yeah, you see?
they're having a different behavior.
Yeah. Definitely, we need to probably slurp.
And I think also maybe either they're a different age, because these I don't see any of those what I call the pregnant parts, which I don't think. But I mean, the
Do you
others
need to do
have.
a close-up?
Yes, please.
8%.
Let's do a close-up on the crinoids. One of those.
Yeah, I think they're different.
Yeah.
It's a beautiful picture.
Mm-hmm.
Do you want to grab these as well?
Yes, please.
Sponge with this.
Yes, please.
But I don't know, what do you recommend? First slurp these and then...
No, I
Regular.
think picking them together because they're sitting and holding on. So let's hope it's holding on. I don't think the slurp will give.
Solvent's pictures, is it?
Yes, well,
Yeah.
Solvin had a picture of a similar, but not. I don't think it's the same species. That's why we want to check. But I think they're holding on pretty well. So I think we can try to just sample a part that this is sitting on. That's what came the last time when we sampled and got one.
This is, for instance, zero and stuff.
So it is going to be cast 8.
So we're going to try to sample a piece of this large sponge, not the entire, of course, but a small part. And at the same time try to get one of these crinoids because they don't seem to be the same as the ones
Isn't it better
we
this
had
one?
at 1,000 meter, which is, I mean,
The one you have on the
a
top.
good probability since we're now at 2,670 meters.
We may smash the crinoid.
Let's see how we can maybe get that without smashing.
Don't close it totally, because otherwise we smash the...
So if you just decide on which bio box. Oh, it went away. Oh, is it coming to us again?
It's stuck on it, like it might fall.
No, it's free.
You've got one leg on it,
Okay.
very fragile.
Yeah, you have the piece of sponge.
Yes.
I think so, right?
Small piece, yeah. It's a small piece.
Yeah.
Just a small, tiny bit.
More than enough.
Withdraw.
We can do this. Can we do this one?
I think that should be okay because it's something completely different.
Right.
So in the starboard
Top left.
front left. Please.
Sitting on a ledge here as well, and
Yeah.
kind of like thrust it all, push it away.
So we're sitting, tipping on the ledge, and the pilots are still doing delicate work. I think it's amazing work from the pilots again.
Zero thrust it. As far as you're able just to help him with the zooming out as well.
Sorry, yeah.
Yeah, sure.
Just because it's very flaky as well. Just right a wee bit if you can.
Which one?
In the top left one.
There's a little bit of spin as well.
Yeah.
Plenty.
Yeah, look, there is a little bit of sponge there.
Yeah. More than enough.
And that's the most kind sampling
Look at
of a crinoid I've seen. Let's just hope he lets go now. There you are. Thank you.
Great job.
Oh,
Oh.
he does
Trying
not
to
want
get
to
out.
let go.
Yeah. Those
Go down and find a
rhinos
place
are
to
able
sit.
to swim.
Yes, they are able to swim, but I think it's kind of stuck on the... Stuck.
Maybe it's slurp it.
There's no one to let go.
It's attached to there maybe.
Really
Yes.
likes Antonio.
Well, we are a lot of people who like Antonio, so that's okay. I can understand that crinoid.
Yeah.
Thank you. That was great sampling.
Yeah, he's in a drawer there.
Yep.
Yes. Thank you.
Can we stay a little bit here so that
We are sitting on the ledge, so they are working it the entire time, but let's try.
we can
Zoom in a little bit and see.
let's see what we get. What we see. So this
So-
Calfago has been already sampled.
Yes, but there is this thing in front of it that is that also a Calophagus or is that no? Because there is a lizard and Dorix there, right?
Yes, a lizard and Dorix. There are two at least. The thing at the base, this fan shape, probably is the same species that we've seen already many times. I'm not entirely sure.
So in my list of species of common species here, I see there is a Calophagus arcticus, and I am
It will
guessing this is the Calophagus arcticus. And I am sure Paco will jump in and correct us if we are wrong.
No, there's not much we can see here.
So we have these other fan-shaped
Yeah.
and some shrimp crawling around.
You spot any amphipod?
No.
Not really, right?
Which is sad. I was looking forward to seeing our Haliragus. We even called it after being on a sponge, so it's Haliragus spongia,
No.
which
Look
you
at this
know,
So the closer that you get,
You see
the
all
more
these-
conspicuous are
yeah.
the porous, where water is being filtered, so it goes, the water gets.
And this is what is giving the sponges their Latin group name, right?
Yeah,
Porifera.
Yeah. Bearers of the porous. Yes. And you get the whole thing.
Oh, that's pretty.
Can these swim out or anything like that?
They can sing. These crinoids can swim. They can let go with their holding legs, kind of, and then they use their arms to
Because
swim.
if I do more samples later, you'll just have to watch them.
Yes.
Yeah.
Doesn't like it.
Yeah, otherwise we can just
Especially if Antonio's not here.
Well, yeah. Maybe you have to stay on, Antonio.
Just looking for the planets.
If you are sitting in the shack, he will come with us.
Be quiet.
We can bring you a lot of coffee if you stay here.
Well, we have
That's a shrimp, and I think, can we zoom in on that shrimp? Because I think this is again that, yeah, I think again this is the Bithocaris leucopis, the one that was described at that Norwegian expedition to the they can go in number one. They can just continue being in number one.
Number one?
That's-
Yeah. We have something else, but
But it's completely different, so that's okay.
it's cast.
Oop,
So then
they are
you have to
going away.
Chamber 2, yeah.
Yeah, let's do chamber two.
Sure.
Yeah, let's just do it. One there and one there, and they're sort of crawling towards us, so it will be.
What's exactly
That's
what we want to...
tiny little things here.
And there is also one in the
There's another one
sand.
here
Oh
too.
yeah, there is another one at the little rock to the side.
Yeah,
So
it's
we
gonna be
can-
challenging, guys. but
Yeah.
We can guide you for sure.
So we are going to try to slurp the little white moving dots. They are amphipods.
That one disappeared, but we can
Yeah,
just
it's
go
here.
again. Ah, yeah.
Yeah,
Then
one
goes it.
there and one there, and the last is here. There,
Here.
there.
Yeah, yeah.
First one is close to the rock.
Oh yeah, that first rock, just if you...
Can you zoom in? The camera, please?
Yeah. Can you go on the rock? Where's that one? Right last. Oh yeah. So you
Up.
somewhere here.
Right in front of the rock there. Yes, perfect.
Yeah.
Zoom, there it is. There.
You can look now.
Try again.
Oh, you're a little bit
Nearer
nearer
to
to
the
us.
right. Yeah, went in.
No, it
but wait
didn't
a
go
second.
in, but it's coming.
Start sucking now though.
The rock is there.
That's you. Pick it up. Just try and move the rock if you can. Choke it.
It's still there.
Can you zoom a little
You
bit
get a
more?
stone
Or
out.
we are in the limit?
I'm gonna start sucking the stones out.
Ah, it's a stone.
So yeah,
Yeah.
Okay.
spit out the stone. Try again.
So there's one more here.
Yeah.
You still see it? Yeah.
Yes,
Yeah.
I see the little one here.
So on your screen now.
Up, a little bit more.
That's it.
A little bit more to the...
No, that's smoke.
Oh, there. Did you see it move?
Yeah.
Shout when you're ready.
Yeah.
Suck it.
Yay.
Yeah.
Yeah.
Yay. Cool. And then we
And
continue.
some other things are going in.
Here is one.
Yeah.
Oh, jumping back on the rock.
Ah, yeah, you can see better now. There's no one here.
Yeah, so there are two ones on that rock there.
Just start sucking now.
Yes.
Hey.
Go for it.
Yeah.
They
There's no
are
one
in.
also
Yes.
back in.
Perfect.
Can you just inspect down?
There is one
There's
as
no
well.
one
Yeah.
here. There's only one here too.
But now we do that one.
Yeah. You can just suck now.
So the thing is,
Yeah.
it looks like we are hovering up everything here, but the thing is that these are
And there's one also here.
actually probably quite abundant, so we are not taking the entire
A little bit farther up.
the entire family or anything. It's just that when
No.
we're finally zooming in a quiet place, we can see these things.
Yeah.
And that's what I talked about earlier. If I had had just a plankton net going over this area, I would have had probably a few hundred. So when we are picking up 5 now, that is still a very, very small amount of specimens, but very, very precious because
see
these
anything
are
else?
species.
Just, yeah.
These are species that we
Cannot see.
need
right?
to or want to make a genetic sequence from, in addition to checking exactly if they have. Of course, at this distance we cannot see the details that we normally need to look at to identify them, but we need to check if they are species we know from beforehand, and also even if they are species we know from beforehand, it is quite common that we do not yet have a genetic sequence of them, and having a genetic sequence will allow us to later recognize if they have been there or not when we are doing environmental DNA from either water or sponges like Sergio is doing, or if
No
from
one here, right?
sediment. No.
There's a few. Oh, yeah.
Yeah, maybe one is swimming in the water there. So the average size of an
Yeah, it went
arthropod
away. Yeah,
is less than a centimeter. The ones that I
Something is
normally work with are between 3 millimeters
good,
and
right?
5 millimeters in size. So it's very difficult to see any details, even with this wonderful camera we have, with a zoom the most in. I might get a feeling for an area or family, but apart from that, I would need to look at these specimens. Yes. In the microscope to be able to say what they are. And normally
Let me just first.
I would even have to check the mouth parts. So you can imagine if you look at the crustacean that is 5 millimeters long, then you need to look at the mouth parts. Of course, not as small as the spicules that we are looking at in sponges,
Swimming.
but it's still small pieces of animal.
I can't see anything.
I think this was a nice set of animals. Thank you so much.
Yeah.
Well done. See, more hearts to you.
Can we pick this?
Yes. When we are already here, can we also pick this rock right in front? Because I think it has... do you want to slurp it? Is it small enough that we
No,
can
because
slurp it?
it's got not just...
Okay, so maybe we can pick it.
Yeah.
I think it is because it has Lysodendorix on it,
And
right?
2 other sponges in there attached.
Yes. So we will do a sampling of the little rock. Maybe if we just do a little bit more slurping of just water to make sure they get all the way down to the chamber.
Yeah. It wanna break him if we keep slurping the water.
No, it will not break him. So we can just slurp so that we make sure that he's in the chamber. That would be great.
Stuff in there.
Nice.
Because it's only this in chamber 2, right? So then they will have a little bit of a dance in there, but nothing more.
2.
Yeah, we can see some stuff in there.
Okay, perfect. Thank you.
Any guess on the number of specimens that we collected? 5?
5. I would say 5. So we didn't find the Haliragus spongia, which is a slightly larger amphipod, and it would have probably been sitting on the Calophocus. But we did find a smaller amphipod sitting on the rock in front. And that is mainly also a good show of how when you zoom in and focus and you have a little bit of a idea about what things should look like, then you can
thing is that I cannot go.
see a lot of new
It's fine.
I
can zoom,
lot
but
of life
the most
that
I
we
can
don't
do.
see when we are further away. So now we're sampling a small rock with Sergi's favorite animal, the Lysodendorix, which is a sponge that lives a lot of places around here. So he is going to try to do population genetics to understand if they are well connected, these populations that are far away from each other, or if they are
What do you think?
separate from each other.
Maybe.
You should be able to get it as long as we get deep enough.
Yay!
Great job, guys. So you can go to the right hand side.
In top right,
Top
in
right.
the starboard would be a good place.
Front way. Yeah, but zoom out and up on that way. Really? Yeah. Okay, come on out.
And we can go a little bit over to this side, not too much. Can we go further that way? Yes.
as you like, pitch out a bit, pitch going up.
So I think everybody who have followed us for a little while now have heard about the Lysodendorix, the sponge that is breeding its offspring so that they are not having a free-living larval stage, and that still lives everywhere in this area.
Zoom in here so you get a picture of it.
In all of the Norwegian and Greenland Sea, and from depth
He's here.
Everything is here, yeah.
from in the depths from
That's what we wanted. That was awesome.
shallower than 1,000.
Shallower than 1,000, yeah. I
Yes,
think
and
so.
we now
Not
are
super
almost
shallow, I
at
guess.
3,000. So all of these, yay, sample in all of these depths and all of
Great job,
this
guys. Thanks a lot.
large geography. How can they be well connected when they don't have free swimming larvae? It's a little bit of a mystery, but we have the good detective Sergi to
Oh, yeah.
solve the problem. We just talked about what we are doing when we are sitting in the lab and sorting our samples. And I always listen to good detective stories, so I will soon listen to the story of Sergi solving the big detective story about the Lysodendorix.
It will take us a while, but yeah, we'll
But
get
it
there.
will be a nice story when it comes out, right?
Thank you so much, guys.
That was
what shall we
great.
do? Shall
Yes.
we continue? A little bit?
Okay, should we... We are at the end... Oh no, we have a little bit left of the... Let's just continue the very last part of this...
The line, yeah.
Of the line, and then we will do another skip and jump. So we will continue with our transect. We haven't even really started on the trans... Oh, yes, we are. But our shift... we have the lasers on again?
Yeah.
Yes. So we will just continue the little last part of this transect.
Bridge.
Yeah, go ahead.
Can I just very gently swing my head to the west, please?
Yeah, that's perfect, yeah. Oh, that's one thing for us.
So here we see that we are on this really amazing landscape, even though it seems kind of flat when we're looking just straight ahead. It's right to the side there is this little hill going down, and it's kind of steep, right? So
Not kind, it's steep.
it is steep. It's not just kind of steep.
Way
And
steep.
our pilots were balancing on that ledge. It's amazing what they are doing.
Oh, these are small Nigerians, right? Yes.
Are they anemone?
No. Those are the crinoids, right?
Yeah, these are the crinoids,
Yeah.
but that thing there, is that the sponge?
Sponge.
Oh,
Yeah,
the one
I
that,
think
oh
it's-
yeah, that's the Asconema that is not, yes, okay. So we have an Asconema and we have crinoids. And we have these fan-shaped things.
Hop over a span and then come back down.
Yes, we are going to do a little bit of a hop. That will be cool. And then we will get to another hill again, I think.
Oh yeah.
Yeah.
With that.
Oh yeah, look at the sonar, we are meeting something. We are using a sonar to know what we are hitting before we are hitting it. Something steep is coming. But look at that, it's completely full of sponges. And all different types as well. That's a lot of Lizardendorix, right? Lizardendorix everywhere. I will register them again.
There's a couple of fun-shaped ones too.
And is that sedimentation or is it a sponge that had sort of covered
I have
everything?
my doubts. Yeah, I think it's sedimentation. Potentially some encrusting sponges through there.
And then there are some of this Asconema.
Yep. That one in the middle is the Asconema Anna was referring to. You see the lasers?
The one that looks like a vase.
Yeah.
So this is becoming difficult because yesterday the vase-shaped sponges were completely white and there were Rosellida.
Yeah.
And today the vase-shaped sponges are more brown and they are in a completely different group of sponges if I learned correctly.
Yeah, indeed.
So these are Asconema and they would be glass sponges, yeah?
The other, the Rosellida.
The Rosellida are also glass sponges, but they are
different,
different parts of the, So okay, both are glass sponges. And the glass sponges are animals made completely out of glass. What's the red thing behind there?
Ooh. Let's find out. Something big.
Ah, that's a relatively big red thing. Well, that's
no, a shrimp.
That's a shrimp, but the other thing, that's also a shrimp.
Ah, no, that's a fish.
Is it a fish and not a shrimp?
Can you see the eyes? Or is just something that I'm making?
I was thinking we see a shrimp from the back.
It's too big, right, for being a shrimp.
That would be a big shrimp, but that's okay. Look, that size of that shrimp and this size of that, I think maybe.
Yeah, could be.
Yeah.
And I think potential carnivorous sponge in here.
Okay, let's see if it's a carnivorous sponge or let's see if it is a different thing. You haven't seen any carnivorous sponges
so
today.
far.
So you have been missing that?
Yeah, a little
bit. You wanted to talk about carnivorous sponges. Oh, there are bryozoans everywhere. When you zoom in, look at that. Bryozoans are super exciting animals.
Bryozoan or a carnivorous sponge?
No, I think they are bryozoans. Oh, look, there is a shrimp sailing along on a little piece of
on
something. But we are hanging out on an edge again, right? Or hovering in front of an edge. That shrimp is holding onto a piece of sponge, I think.
Yeah.
An upside down parachute. So the potential carnivorous sponges, you have been missing them. Is that because you think that we should have some sponges that eat the crustaceans? Oh yeah.
So many crustaceans.
Oh yeah. There's never enough crustaceans. And there are so many sponges. So here we see a lot of crinoids sitting on Lizardendorix.
Okay, let's see.
Lizardendorix and crinoids all over the place. Which is also interesting because earlier we were seeing the crinoids sitting on the Rosellida. And now here they're sitting on the Lizardendorix.
So just beside, now with the lasers, I think there's this branching carnivorous sponge that you may see now.
Oh yay! You got to see them, carnivorous sponges.
I think.
they are?
I can't remember. Paco may know, or Julio.
Yeah, if you are listening in, you can tell us what it is. If not, we will look at the video later. Yeah, look at this area. This is crazy. Whenever you get a rock wall, it becomes completely wild. Wow.
There's plenty of crinoids, even on top of the Lizodontorics. You see?
Yeah, see, they are now. Maybe the Lizodontorics here wanted to be fashionable as well.
Yeah.
Dress up a little bit, put on a crinoid, go to the ball. They already look like they have dressed up with all of the ruffles.
Getting ready for the weekend, right?
Yes. And it's only Thursday. Is it the same thing in Spain that people do Thursdays at the bars because it's almost a weekend?
It's called Juernes, which is the combination between Thursday and Friday in Spanish, of course.
So they are having a Juernes already.
Yeah.
Are these the same?
Yeah, I think so, yeah.
The same, these small stick-like things that you see with the lasers now, almost, yeah, next to the lasers?
Yeah.
They are also carnivorous sponges, right?
Small branches.
Yeah, okay, we call them carnivorous sponges. There is a lot whenever there is a empty rock face. It's not empty.
This is one of the peeling rocks for these guys.
Yeah.
Potentially thanks to current.
It's a good current here.
Yeah.
And there is a lot of things in the water. We can see lots of swimmers. I would say very many of those are small crustaceans of different varieties.
More into that direction to
Oh yeah, now we are going over this wild landscape again. Look at that. There is this almost like a little river of mud that has been sliding down the mountainside, and then we come to the next mountainous rock part covered with sponges again.
Is that the sea star? Over there? Oh, let's see it. No.
No, it was just a sponge. Another sponge.
What else?
But you got your carnivorous sponge today. I have to tell you all, Sergey is smiling in the shack right now. He's like, yeah, finally I got to see that one today as well. Now I can have a happy afternoon.
There's something we're missing, if you remember from previous dives, is the Assyrians, right?
Yes.
At least I couldn't see any.
I totally agree with you. We haven't seen Assyria today.
And in the dives that's around 1,000 meters depth, there was like a really abundant number of these guys.
Yeah.
2 or 3, even if you
Look at that beauty in the middle of the picture right now. This is this is a beautiful sponge, huh?
I think this has stop. Thank you. ¿Qué tal? Is it going?
I am going to leave you with another sponge guide for about half an hour or something. It will be sponge. So remember that there is other life than sponges around in the world. Might be hard to realize, but Julio is here to take my place for half an hour. And I will go and talk to the Swedish TV. So if you are in Sweden, you can, scary enough, see me in 2 channels at once. That would be scary. Don't do that. But listen in on the guys here, because they are going to tell you everything about the sponges.
Yeah.
I'll just keep this here.
Shall we have any petition? Do you know where the potential species could be? We could potentially go and pick it up.
It's probably all around, but we need a boulder, we need a little rock. So, not in this
Not
area.
here?
No, not here.
Can we go somewhere else? It's a huge rock. What else are we missing? Something wise?
No, not much. Maybe your design Dorits.
I would take another one, so that to make 8 in total. But we can just go a little bit in that direction. Yeah, let's explore this a little bit.
I think that we can try to find a really nice rock with maybe several things, not just one or two. So one that is really
We took one of
well
those
covered.
with a small Lisbon-Dorix plus 2 or 3 sponges.
okay.
But yeah, we can definitely try to find some other things.
I mean, if you took one, you can just check maybe. Because the new species are really all over the place. It is really impressive,
Yeah.
the whole area.
Is that the Stellodorix, the one?
I think that the orange one, yes.
So this fun shape individual that you see here close to the lasers, those are Stellodorix
Flabellata.
flavellata.
And we still have a question mark on the whitish one. That
There's
is.
some stuff in here.
Yeah.
Many boulders in here. Do you wanna
Yeah, maybe
get
we can
closer?
stop there. Yeah.
Close to that?
Simply now, right? Which way you want to face?
Ah,
this
yeah.
way?
Yeah, that's a good question.
Once I'm down the vessel.
Yeah.
That's hard to say.
Let's inspect a little bit this area. Is any?
I mean, if we find a rock with some other stuff, the more the better. So it's not that big. There's not much in there. Questa? Muy grande esta.
Hay cositas,
Ah,
Son,
eso. Bingo.
I don't know whether this is a single rock or.
It's quite huge.
You think we can try this one?
Yeah.
Or is it going to be too big? I mean, if that's 15.
Well, how about another one? Just Reba. There. Yeah. Are you looking straight on your camera like that? Yeah.
Zoom out.
Yeah, just to see which way.
It's that one here. So there's some.
Yeah, but is your camera straight or do you zoom out a bit just so you can see where you're looking?
I mean, it's a little bit large. Well, there are others nearby.
We can explore. We
Yeah.
just land in here and then
Yeah.
explore.
That's right. Right, just zoom in again and point where you want.
Yeah.
Oh, it's not.
Which one was? This one?
This one, but if we land, we just have a look.
Yeah, probably this one. I will check first whether there's anything you're interested in.
Maybe this one will be enough. It's bigger. Esta es la cosa.
Daniela.
Esta de aquí igual. Esta de aquí tiene una pinta también.
Sí.
Vamos a hacer un macro y a pensar. Vamos a hacer macro todo esto de aquí. Y no lo pensamos con calma. ¿Vale?
From there.
So I wanna wait a second to have like
Yeah.
an idea. Can you please zoom up the laser?
Yeah, 2 seconds. You turn off this one.
I mean,
Make it off.
this one is really
This
nice.
is beautiful.
Yeah, that's so. I think that you see that some are a little bit more smooth, like this. It's hard to say because there are many things here.
Do well.
You see this?
Yeah. Is that the one? Or similar?
I
Is
would
that the
say
one
so.
too?
I'm not sure. No, I don't think so.
Oh, no?
I don't think so. But
This
I'm
one
not sure.
potentially?
So this one potentially.
This may have it. Look, this stuff in here.
So we're out.
Here is good because we also have all this stuff.
But you are interested in that new potential species.
This one I think that is also maybe new. Maybe. So if that fits,
Let's do it.
well, we are
We're here to sample and then I'm happy.
let's just zoom in. So I was thinking that this one maybe is not in the rock, in the same rock.
But this one is, right?
No, but this one is a different thing. You see?
This is the Velata.
This is not for Velata.
Or something. No.
This is Emigilius, Schlotfeldes. This is the one we collect before.
Yeah.
We go to have more specimen just in case. And this looks like something that I identified yesterday. Maybe Nimedesmia.
And this is the same.
No,
This thing. No?
it's different. That's a 4-bass, I think.
I was just saying this small here, this in Croston, it's not the same as this one?
No, I don't think so. Look at the oscula and the star-shaped canals. This one, and then go to this one. You see that the oscula are different.
Yeah.
The color is also different. So no. I think that this is more like 4-bass and this is more like Imedesmia, but this is just a shot in the air. And these are the little calcarian that we saw the other day.
Yeah. There might be others in the rocks that we collected.
This is a Raniera. This is Emigilius. This is the one I think that we want. I'm not sure. That's a
That one
carnivorous
here?
sponge. Yeah.
Yeah, this one.
Sorry.
I will try.
I just took.
With the arm.
What do we think? Whole stone, is it?
Yeah.
Whole stone.
Do you think we're gonna need the
When Andre comes in, I'll bubble to the
well, I mean, the thing that you prefer the most. Where do you want, what box do you want it in?
What do you do? Scoop that last one, did you know? Just drop them.
Number 7. Oh, right. Yeah, we're going in the starboard aft.
Well, if it's the whole stone, I'll just see if I can pick it.
Right. Yeah,
You zoom out from here and just follow in my arm.
Yeah, that's good.
Sergey?
Yeah.
Just zoom out just so I can
Yeah,
see
sorry.
where
Oh, yeah. Is it too big?
I think so. It's too big.
that's not.
We can look for another one, huh? It's plenty. Careful with the sponge.
With the stone, yeah.
Perfect. Perfect, my lad.
I mean, they used to be photographed by surveyors. Back left. Okay.
¿Qué castes?
11, 11. Cast 11.
Lot 7.
Ya está, ya está. Amazing.
Perfect. Thank you so much.
Alright.
Yeah.
Yeah, good.
Super happy.
Okay.
When the lob's out there now. And Andre, you jump in here then. Pues ese es el mismo. Me preguntas eso que no lo veo, tío. Las coordenadas. No sé quién es. Solo me puedo.
Esto de aquí en 11, ¿verdad?
Sí. ¿Tienes el 56? Uf, qué buena esa. That was a really nice cast.
So, we're flying just on this here.
I aquí tenim 7, 12.
Yeah. I'll write down.
Oh, this is cast 11, barrel, boats, 7, rock.
Yes.
It's a list pretty complicated. Yeah. Super beautiful this area.
Es que tengo mis dudas entre dos. Yo creo que estos son polymasties. These are polymasties.
Ah, the ones with the oscula at the very center.
Spinularia actually is a polymastide called Spinularia njordi, something like that.
So those are the ones that you see roundish
Yes.
Specimens.
I think that those are, this is a species described by Alexander Plotkin in the Loki's castle 10 years ago, called Spinularia njordi or njordi. I'm not sure how to pronounce it. I think it's a Norwegian name. Very close to here.
Yeah.
Also same depth.
So it makes sense that it could be the same species.
Yeah, I'm pretty sure it's the same.
So we were, just before you came, we were inspecting the sea bottom close to the rocks because there were some amphipods in there. I cannot see any now, but this is one of the things that we should potentially be doing also in order to get samples for Anne-Hélène and Annelena and Jenny. have a look in here. Anything moving? Just let me know. And this is a carnivore sponge, right?
Yeah, I think so.
Which one is this? Furcata.
Avestopuma, I believe.
Which one? Furcata. Is that the one?
Yeah, I believe.
Yeah, let's change. Yeah, it's a beautiful one. Yeah.
I don't know what is this. Is it green?
Yeah, that's a stalk crinoid. We pick one at the very beginning of the life. I can see the tentacles or the crown
At the bottom, at the end.
Ah, yeah. It's difficult to see when you have the sediment back. Ah, yeah, you see. Look at that.
Where? Ah, the crinoid. Yeah.
Yeah,
There you
beautiful
go.
one. It's like a flower, you see?
Mm-hmm. Do you know the species?
Yeah, one.
For that one?
No. In fact, collected one just to try to identify it. Beautiful. And there's a shrimp on the back on the left. That's fine.
Many crusting sponges.
Yeah. Very bottom right. So these grayish spots in the rocks, those are encrusting sponges, right?
Yeah. This is where you really increase the biodiversity when you start looking at those. Because you may think that this is one species, but there are actually many species here. Yeah.
Yeah, there is another encrusting sponge there, right?
Well,
just below the Islandorix. Is that an encrusting sponge?
when I see this whitish border, I think that that's colonial Assyrian. Maybe.
It could be.
Hard to say, so far away, because I see some kind of a structure inside, you know. So it's probably colonial Assyrian.
Mm-hmm. Yeah, good point.
Now they shall endure it everywhere. That's the queen.
The master. We just continue exploring here. Look, this is a field of stalk crinoids. It's really beautiful. Like a garden of with flowers. Look at that.
Yes. They're facing towards us, all of them.
This is super beautiful. Coral? Coral?
No.
I don't think so.
Sponge. The Casconema over there.
Yeah.
Oh, this is nice spot, huh?
Super beautiful. That'd be fun. Get into the limits. Camera. Beautiful. Shall we move on?
Yeah, I'm
Maybe.
good. Yeah.
So we're happy to go? Move on? Yeah. Let's explore a little bit more ahead. Gotcha.
So how many species do we have here, Sergi?
In total here, I don't know, the megafauna-wise, I would say 10, probably.
I would say more. Megafauna, you mean without sponges?
With the sponges, of
With the
course.
sponges? No, more than 10.
More than 10, yeah. I mean, including the stalk, crinoids,
And the crystallia.
so Islandorix, the fan shape. At least there's 2 or 3 species fan shape. This makes like 5. The
So three
Asconema.
let's say 3 fan shapes.
Yeah.
Asconema, then the ball, Calophagus,
Calophagus 5.
Emigelius,
6.
Rebridge.
Stellodorids.
Hello. Can we have a movement of
Yeah.
0 to 0, 0.2 knots continuously?
6 or 7 encrusting sponges.
020 at 0.2 continuous. Okay.
Can you turn
Thank
on
you.
the laser, please?
Yes.
Okay.
Lasers on.
So 15 sponges, maybe something like that, and then all the rest.
Yeah.
Not bad. 3,000 meters almost.
So it's 2,670 meters so far.
6.
Yeah, this is the previous one that we collected, right? Or at least similar.
The one that had the
Caulifacus.
Caulifacus, yeah.
So, just a while ago, we collected a piece of a specimen for this species, the one where you see the lasers. This is Caulifacus.
No idea.
No idea of the species,
Don't remember.
but at least the genus we have it. This species has a stalk, really wide stalk that connects the sponge to the rock. And I think we're heading to an area with sediment and full of these cranials. See them.
So I'm very curious about what would this fan-shaped sponge be. We'll find out soon.
Do you think you're going to be able to identify it? I mean, it's one of these big species, really conspicuous.
Well, at least 2 genus, we should.
But I mean, chances for these species to be new are low, right? Given how frequent it is.
It's lower than the little ones.
Of course,
people tend to describe first things that are more obvious, more patent, larger. But I mean, it's also not rare to describe new species when you are at those depths. So.
Yeah.
And even if it's not a new species, you can barcode it for the first time maybe, and then you can place it phylogenetically in the tree of life. So these kind of questions you can answer.
Checking whether anyone is asking something.
Reach RV.
Have one of those lollipop
Can we
at
change the
Sentinel.
speed to
I don't remember the
0.3,
name.
please?
It's a beautiful world.
Thank you.
Ah, yeah. I think Paco mentioned something about this species.
Yeah, we have the
while
name
ago.
upstairs. Just call it lollipop Sentinel for the moment.
Yeah.
Those are nice, you know, they create like these fingers.
Yeah.
You see they're all facing the same direction.
Yeah.
And you have the Lysodendrorids lying where there's not so much current. So
Yeah.
I think that, you know, they are just saying you go here and we don't compete.
Yeah.
We don't need to fight. Probably Lysodendrorids is more, doesn't need so much food and stays on the second.
It's super obvious that you mentioned how they're facing backwards.
Yes.
So you can tell where the main currents come from. In this case, in this area.
Yeah. Super obvious, super beautiful.
We're gonna maximize the amount of food they're gonna be filtering.
Yeah.
Thanks to this precision against the current.
Yeah, it is.
It's super beautiful. It's, I remember this landscape during our second dive, right?
Yeah. So this is an habitat that we should really put our efforts in try to describe as well as possible.
Yeah.
Because you can see that stand is vast and this is creating an habitat
Yeah, for sure.
So these are the kinds of things that you are looking for when you want to protect an area, or look for reasons to protect an area. You find this, you say, oh,
This is definitely an area that you will identify as something to be protected for sure. These sponge grounds, these massive organisms in here, probably took for them to grow like this like hundreds of years, right?
Just safe going downhill now.
Yeah, no worries. So we have to go to fly a little bit up, since we are not going climbing, and then when we're going down, which is a little bit dangerous for the ROV. So you're counting to have an idea of how far you are getting.
Obviously, but I also got what's the back end of the ROV.
Yeah.
You come down too quick,
No, yeah. Talking about the yeah. I guess the currents here are not so strong. You can imagine, given the species that you find in here, mostly Sulfid-Hügel and just one or two fan-shaped sponges in here. So potentially less currents in here.
yes.
So something we've learned.
It is. For me, it's very beautiful to see how they are, through the years and the ages, building these reefs, or these gardens, these sponge gardens. Super well structured.
Yeah.
More like a sponge garden, I would say.
So in this case, we haven't seen any soft coral that we saw at the very first dives.
Yeah.
No gorgonians, no bamboo corals that we saw 1,000 meters depth. Just sponges. Or most of the diversity is sponges. At least the macrofauna, the megafauna, the fauna bigger. You can see with the ROV at this distance.
Do you think that you could tell which genetic adaptations the lizard have between the shallow and the depth populations?
Yeah, if there's any, we could potentially do that. So we've done that with another species, also from this area, Here, wurden, and we were studying Here, wurden from shallower, 1,000 meters depth, something like that. Deeper areas, like around 2,500 meters, and they were clearly expressing genes, and had clear differences at the microbiome level. So the microbes that they host are quite different, the ones from the shallow, the ones from deeper waters. So this points out to different adaptations of the individuals of the same species in different depths. And I'm pretty sure that we can just find something similar with this Sulfid-Hügel one once we study them in the lab.
And can you tell which genes are being expressed more or less?
Yeah. So ideally, we should have a reference genome of this species, something that we could potentially try to have. But otherwise, when you sequence these guys, you can compare the sequences that you're getting with already existent sequences from other species, and then try to identify which are the genes that are involved in these adaptations. Normally, genes related to pressure or to temperature or things like that are the ones that are going to be related to adaptation in these two different environments, shallower and quite deep habitats.
So it is about the permeability of the cell, maybe.
So we found in Here, that when comparing different depths, there was some genes related to composition of the lipids or the lipid membrane, making more viscous the membrane of these organisms when going farther deep. This is just to withstand the pressure that they have to live in. It's like 3,000 meters versus 1,000 meters. Mm-hmm. So some of these genes were related to this lipid metabolism with direct implications on this cell viscosity.
So what they control is the interface between the outer environment and the inner environment in the cell.
Yeah. So in this case, for Lysodendronics, we're going to be also making something similar. We're also going to investigate the microbiome in association with these individuals from different mathematical ranges.
Is it?
No, it's just the shape of the rock.
So does the microbiome change has something to do with the diversity of the microbes between the deep and the shallow ones?
Not in these cases. I mean, what we found, it was like slight differences. It's not that the composition of the microbiome is totally different, but it slightly changes with some species that are more resistant to deeper waters. But if you analyze microbiome from one species of individuals, shallow and deep, you're going to have a similar signature of composition of these microbes. So we are not expecting dramatic changes when comparing them, but probably slight fine-tuned changes in the community of these sponges.
Yeah, Lysodendronics.
Yeah, I think Lysodendronics is one of these low microbial abundant species, while Here was a high microbial abundant species with more than 90% of the tissue being bacteria, which is like a crazy amount of bacteria. So look at that, it's like a small hill,
super cool,
made of
66
sediment, right?
How much we are up since last 70, 66, so it's only 5 meters for the moment.
Beautiful wall. It's so muddy in here, so you don't see the rock. It's completely covered by sediment, I guess compacted sediment.
Those platforms are interesting, huh?
Yeah.
Below lava, beneath.
Reach ROV.
Great.
Can we slow down to 0.2, please?
Yeah, watch out. Oops. sediment
the
here.
layer.
Yeah.
Yeah, I wonder. Yeah, the
So
summit will be covered of the fan-shaped
Yeah,
ones.
you see, this is the
Yeah, look.
again facing the strongest current. These fan-shaped sponges that you can see. Beautiful. All together with Lisodendrones and
Yeah.
other stuff. Vertical wall. Over there.
Yeah, or just to check now, just for you to check, just for you to check with the camera,
Oh yeah,
to see if it's worth going or not.
it's more or less the same as the actual outcast.
Just lying on top of it. It's a rock, just a rock.
Hi guys.
Hey.
Hello, how was that
Thank you for
interview?
taking my space.
No problem.
Okay. See you later.
See ya. So we took a beautiful rock
Okay.
with potentially Julio's new species.
I just talked about our potential new species on the Swedish news.
Oh, really?
Yeah.
So how was the interview? Did you like?
It's always a little bit scary to be live on TV.
Mm-hmm.
I've only done it 2 times before, so that was
You
woohoo.
have experience at least.
Okay. Back to the beautiful subsea
You
world.
see now. We were just explaining the main disposition of this fan-shaped sponge against the current. So we guess that the main current is going to flow from the very bottom until up the hill.
Mm-hmm. And we are nearing the end of the transect, right? Yeah. But this is such a beautiful area that we might want to sit down a little bit and look at it,
Yeah.
or depending on if it's possible or not.
Yeah. Then try to find more amphipods.
Did you talk about sponges the entire time I was not here? Yes. I am sure you guys out there have had a wonderful nerdy sponge time. There seems to be another soft bottom. I mean, if we
Yeah.
are really at the... this is probably coming and going all the time, right?
I mean, we can always turn a little bit and see whether we have
Yeah.
these big boulders.
Is that another of the Silicordula?
Hmm.
Down at the very bottom? Or maybe up here, right before the Aravina?
Can always stop and check. But those are maybe these savelids with together with
Yeah. Or maybe they're still the stalked crinoids
yeah.
that we have been seeing the entire day.
Yeah, we saw a bunch of them when we stopped for the rock. They were really beautiful.
This part is all sandy.
Yeah.
Yeah, this looks sandy. So let's maybe.
Where are we now? Are we at the
We
top?
are
Yeah.
at the top of the
I
line.
think this was your transect here.
Yeah.
And we're up there now, so
Yeah.
we're literally that far.
Yeah, let's just
Shall we do?
find, if you could turn up and see if there is maybe a little bit of rock or something
Yeah.
a little bit up, and then we can sit and have a little bit of look. Because then if we are at the end of the transect, we should, after a while at least, move on to the next transect.
Yeah. It's gonna take us a while to get there.
Yeah.
I think it was something around 900 meters, which is a lot, given the...
And then we can type in, so let's type in now, remember to do that.
Some hard contacts coming up. So 10 meters.
Yeah, let's go to some hard things and see if we can find animals sitting on them. And if it's possible to land close to something hard. Or is it too steep?
Yeah. It's alright, as long as they're going uphill, they'll be okay.
Okay, then we could have a little bit of a look and zoom in on a couple of things, and then we can go for a little bit of a skip and jump over to the next transect.
There's some rocks.
There are some rocks. But we're really going uphill again, right?
Yeah, very much so.
Yeah. Let's find a place where it's comfortable to either stop or maybe we can just...
No,
So which direction is the next?
It is up.
The next dive is... Yeah, so this is Mohns Treasure, and we are from here, and we will go to the next. So
But
yeah,
there's
so it's
900
actually,
meters.
but it's like northwest to the next.
Yeah.
So it's okay to go a little bit uphill here because that's already northwest, isn't it?
Yeah.
So if we just say technically we're ending the transect here, and then we go a little bit toward the next next location, and we can maybe have a little stop somewhere where it's more comfortable for you guys to stop. But the end is 11:29.
It's just all sand now because no hard contact already. So we're heading to these ones. So that's good heading. Okay. Okay. Up here sort of thing.
Yeah.
Okay.
Somewhere around there.
Somewhere.
Yeah.
Somewhere around out there. So when we have looked a little bit at the map, we can tell you how far it is to the next start of transect, and on the way to that place we will try to find something where we can stop and have a little bit of a look. But right now we're going to fly over some of this very cool, very rich, oh, is that another seeping maybe? Soft sediment.
Yeah, that's fine.
And it's just that this soft sediment is even more interesting when you put it under the microscope. Yes, there is another Calophacus. Yes.
Can you stop here, please?
Yeah.
Can you just hang a little bit and let's zoom in on that
Just in
one?
front of the...
Bridge.
Like here?
No, it's on the hill. We can't stop.
No, but here it's a bit flat.
Is it? No. No, it's really steep.
If you see it's too steep, then
Trying to head this way forward. I don't know if it's gonna lose us.
Okay, we're going to see if we can look at the Calophacus, that big
That's what I got it now.
sponge. Oh, that is a nice burrow up there as well. Now we're sort of zoomed in and everything, so let's...
yeah, sorry,
That's your fault.
we didn't say in advance.
Yeah, well, they give us. I think we'll be able to sit down and clear the area.
holofocus here. Thank you so much.
That's right. Maybe 15 pounds. Just 50. 50. 50. 50. 15. 15.
Yeah, cheap.
That's €5 each.
So now we're facing holofocus. Before we were like taking a shot from the very back. Look. So, where were these amphipods living?
They were sitting on the outside like that shrimp is sitting now. So if you zoom in and look at the shrimp, yeah, look at there. Maybe there is an amphipod there. Then...
We're gonna use the slurp.
Yeah, but first let's have a little bit of a looking.
Let me check. I think it's that's a
yes.
crustacean.
I think it's a crustacean. And that's amazing. So now can we please take off the lasers?
Thank
you.
There's another one here.
Yes, oh perfect.
Okay.
This is what we want.
Go away.
If they have the, it's crawling around, so please, yes. Let's look a little bit at this and maybe we want to slurp a little bit from this.
You might get it all, but...
That's okay.
Oh, really?
So the reason why I wanted to slurp this is because I think this amphipod that we are seeing crawling on the small pink thing, not the big pink thing, is an amphipod that Nina Lurtz and I described a few years ago from this same species.
There's two in here.
Yes. So I think Nina is sitting up in the dry lab, and I hope that you are happy seeing
this
it.
is a decapod.
That's a decapod, yeah,
that,
but the other one. So there is a big shrimp coming, but right next to it. Let's collect them because there is an amphipod. There is an amphipod there as well. Behind the shrimp, there and there. You see that pink thing there?
This?
No, that. Yes.
Yeah, these two things.
That's what we want. And there is another one here.
Yeah.
If you look here, there is another one.
Yeah.
Which? 3.
So this is the first refind of that species that we described. So I'm really excited now. This is super cool.
Awesome.
Yes, very much so.
Number 2 is empty, right?
No.
Number 2 has other amphipods in it.
Okay.
Number 3.
So please, number 3.
Okay.
So we've done that before with the shrimps and the amphipods. Isn't that you have to put the slurp like super close? Just a little bit far away, like 3-4 centimeters away.
Yeah.
And you start slurping, and once you see that it's in, you continue slurping a little bit so that they can reach to the very end.
Right. Okay. Zoom out.
So I can see the amphipods more.
They want number 2.
1, 2, and 3. So basically...
So the small ones are like here and there, they're moving a little bit around.
Yes, they are movers. And if I am right, I think maybe these might be, then they should be a species called Halliragus spongia that were described by Lurtz, Tandberg and 2 students. So that will be exciting to get up in the lab. I am already super excited now to have this in the lab.
Just rotate that wrist. That's it. Keep going.
Oh.
So right now they are hiding, but the
You see one here.
Pilots can see from a camera that is right above that arm with the suction, so they can see a little bit on the backside of what we are seeing. So yeah, I think it's crawling around there as well. This is super amazing. I am so happy.
We need to slurp them.
Yeah.
Right there. It's that bit there, just along that line there. Come in.
You see? Yeah.
Yeah.
See that there? That little thing there. Right there.
This is super exciting.
Which one shall we start with?
Oh, we
This
start.
one?
No, we start with whatever is. Yeah, just there. I think it's coming. Yeah.
A little bit up.
Go up, go up.
Yeah.
There you have it. Whoa, 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 whoa.
Oh.
Hold on. Okay.
There, it went down there.
Yeah.
You see, it's down on the.
Down here?
Yeah, down here. There's a pink thing there.
From there, I think.
Yeah.
You can do it.
There,
you can do it.
yeah.
Just
Just slurp.
Slurp, there we go.
No, you're on
No,
the
no, no,
backside. Uh-huh.
Come a bit
Oh, we're
closer.
super excited now.
There. Sorry?
Don't slurp now, please.
Don't slurp.
Let's go backwards. Go backwards. You have it. It's here.
Okay.
Don't slurp. Just go backwards a little bit. Slurp.
No. Yeah.
Slurping.
Oh, wait a second. I think we got it right.
Let's see. And there is one on top there now crawling.
Yeah.
Right there. There.
Slurp a little from the distance. Not too close. Don't slurp. Don't.
No. We scared that shrimp a little bit away, but then it didn't get sampled, so that was
I think
good
it's
for it.
blocked now with the
No,
sponge, right?
that's okay.
Bit of
Sponge is
sponge,
not, yeah, but that's okay. It's flowing away.
Yeah, but it's not gonna go.
No, but
Very
just if we continue slurping what is in there, there is at least one or two in there.
Yeah.
Yeah. That's cool. We found it again.
It's coming as well.
Oh, this is really
There is something there. Yeah. There is somebody down there. Yes, the red thing right under. So
I cannot see it now.
no, it was right behind
Just
here.
there.
Yeah.
It's way in our road, she says.
Here?
Yes, a little bit up actually, but
Up?
yeah, but that's okay. Like that, and just put on some strap and let's see.
Like
Okay.
that?
Yeah.
Yes.
Okay.
Slurp?
Yeah.
Yes, please. Okay, cool. And at the same time, we have a little bit more sample of
Yeah.
Calophocus.
Okay. Stop slurping.
Thank you.
Thank you so much.
Yay.
Has it gone?
That's great.
Do you want to slurp a little bit more in the
just,
water?
Just slurp in the water so we make sure
Did it
that
go
it
through?
comes
Yeah,
into
yeah.
the...
Alright. So it's 11, 14. Shoot down. So it is SS3.
Yes.
Can you write
I
down
can
the
put it in. Oops, yeah.
coordinates? I will write it. Write it down.
Yeah, good.
Thank you. That was great. I have been hoping to find that again.
Mm-hmm.
You know, when you describe something new and then you never see it again, you don't know if it's actually a common thing or it's not. But now we have shown that it always comes on this, which is it explains a lot more for us.
Moving on.
Do you love the image transit?
Oh,
Do you love the
Yes.
image transit?
So we're gonna go.
We're gonna
to the other one.
Yeah,
That
let's
direction.
just move in the direction of the next transect, please. Yeah.
Great. So the work is
I will
done.
bring, because this is empty for, I think we have emptied out a lot of the lead when we opened it, so I will bring new lead for it.
That is
sorry?
Yes.
Yeah. So that's 117.
There's beautiful fish laying in waiting, just
It's
hanging
been a while
out.
since we didn't see a fish.
Yeah, well, it's always good to see a fish, right?
Is that the same species we've seen already?
Just so I know, I'm heading in the right direction.
Could be. Yeah, I think so.
Yeah,
that,
swimming
yeah.
away. So everywhere where we see one of these sponges, there is a little rock hanging under, and being the living place for that sponge.
Is there a rock, right?
Hardly Ridge.
Or maybe a hole because it has all this fresh sediment around it.
Of the ship.
No, a rock, but it's a new rock.
0.2 knots.
There's a little fish there.
350 at 0.2.
Thank you.
Huge sponges.
Oh yeah. Okay, so did we put the lasers back on maybe? We can get that done when we are ready.
They're on. Yeah.
They're on, thank you. Asconema.
3 in a row, you see? 1, 2, 3. One
Yeah.
here.
And the shocking experience of seeing a lizard and Dorix.
Oh. Again.
Again. How about that? Are your drinks
Totally
full of
unexpected.
lizard and Dorix? Do you see them when you close your eyes?
Yeah.
Yeah? And is it the nightmare or is it a good dream then?
Hopefully a good dream, yeah.
It's a good dream. That's nice.
But I didn't know about the existence of this species until we started identifying or looking at the videographies. Mostly it was Paco that decided that this species is common enough, as we have seen it is.
It's very common, so that was a very good call from Paco. Yeah.
So this way we started collecting this species from the very beginning because we knew that it was occurring in all along the transit. We are exploring at different depths.
Oh, look at all of these fan-shaped sponges sitting right at the edge of that rock. So now we are back into that harder area. And I think we will maybe move over this hard hill full of pebbles and cobbles, maybe most of all. 1 or 2 boulder and some hard rock. So then of course is the question, do we have to play hard rock when we are looking at hard rock? Those of you who are back home can decide that's really cool. That's a lava thing, right? That is so volcanic.
Dorix is the one, or the orange one.
Great ROV.
The other ones will have a name,
ROV.
at least for one.
Can we slow down to 0.1,
But we
please?
will soon.
0.1.
What?
But we can generally just move as you feel best toward the start of next
That
transit.
direction. It's gonna take us a while to get there.
Yeah.
It's gonna be our next shift. How long
Yes.
until we change the tape?
It's 16 minutes until the next shift, so. But we got the exciting amphipods. I am so happy about that.
Really looking
Yay!
forward to seeing them too.
I am too. This is going to be cool. We are going to have an amphipod party in the lab. Looking at our beauties.
What was that? Is it the same fish we've seen already?
Oh, that's very relaxed. Just hanging out at the seafloor there.
Yeah, I think it was the same one.
Yeah, oops, not relaxed anymore. So when I try to do some gym, I do interval training because they say that this is the best. Do you think that's what the fish is doing as well? Relaxing, doing an interval, relaxing, doing an interval?
Probably, yeah.
Yeah.
It's cold down there, so just to keep him warm, right?
Well, they don't. The things that we have gotten a question on in the chat here is how it is with the animals that we are sampling when we are bringing them up to the vessel and we don't have a pressure. In the same pressure as on the outside, and there is no compensation anywhere there, so it's no bad thing for them to be taken up. And also we
340, please.
we go kind of slow on the way up as well, so we see that these animals are still alive when they come to the ship, and they don't get deformed or anything like that because of this
Can we
not
change
being
also the speed to 0.2, please?
an animal with a swim bladder.
Yeah, I totally agree. And I believe temperature is more crucial than keeping them at similar temperature than the one that is facing every day is minus 0.5 degrees Celsius. It's more important than probably this pressure.
So we
we're not
try-
adapted to higher temperatures.
Yes, so we keep them in very cold water until we preserve them for further studies. And that is, of course, part of the thing with sampling. We have to preserve the specimens to bring them home to our labs and to keep them even in many years later. So we very often preserve things now in ethanol to make sure that we can also examine the DNA from these animals. Sometimes we put, I think for the sponges you subsample a little bit and also sample in formaldehyde and
Glutaraldehyde.
glutaraldehyde to be able to look at other details than you can when you look in ethanol.
Yeah.
But we make sure to not make the animals suffer a long time in warm water, or
the animals or pieces of the animal, because
yes, for sponges we are looking only at pieces of animals now,
cases
and
we just collected in some of the cases just portion of these animals that will be used for these studies, and we kept the whole animal, whole specimen in its habitat.
And of course sponges are the kind of species that animals that can regenerate what we took away
quite easily. I mean, it's going to take these animals a while, but they will for sure regenerate whole specimen very soon.
So these animals are still down there and living happily, we hope, or they will be happy again because they will regenerate again. Those amphipods that we caught, so some of them swam away and will still live down there, but the ones that we sampled will be first examined in cold water where we know that there is enough oxygen for them, and then when we decide we have done enough of examination of the live animal, we will preserve them in ethanol to make sure that we can use the same animal a long time for research. So they will also become part of museum collections so that other scientists later can come and look at the same animal, making sure we don't need to do a lot of new samplings all the time. So I am looking at all of these white dots, and I'm wondering if they are sponges or if they're not sponges. I am seeing sponges everywhere, and I think it's because I'm sitting here with you all the day.
You are like getting crazy.
You are, you're taking over my brain and making it see sponges everywhere.
Come to the dark side.
The force is strong in me. The crustacean force is strong.
Not entirely sure about these being lollipops, the white.
No, they're maybe not, right?
I don't think
I'm just
like
hoping because I know that the lollipop sponges, the Stylocordula, quite often have amphipods sitting on them, of course.
Yeah.
So that's the force. But you are coming over to the good side here, seeing amphipods everywhere. That's good.
And Elena, and Nina, and Jenny to have enough samples so that to keep themselves busy.
Yeah. If not, we will, run around the vessel and sing.
We don't want this to happen.
I have been known to sing around. So maybe it's not too bad. I could sing a lullaby for you. And you are too long in the lab.
I will fall asleep.
I do this to my students when we have student cruises and they are too long in the lab. I just sit in the lab and I start singing lullabies and I see them dropping off. There is another fish. I think it's siesta time for the fishes.
Yeah, there's plenty of those.
So we have 8 minutes left of the tape.
This is one of these amphipods that you study went away.
That was almost like in a ballot box. Poof. I voted for the ROV. What's that? What's that? It's just darker sediment, right? Okay.
Transit 799. I think it's 9799. Yeah.
Shrimp.
Just what I'm trying to do.
Yeah.
You can try it, Jen. Just be careful when you come over this end here, look. Yeah, just be careful. Yeah. Do that, Jen.
Bridge ROV.
RV bridge,
Can we increase to 0.4, please?
increasing 0.4.
Thank you.
Hello. We collected a few amphipods from Holophacus.
The species that Anna described earlier. Yeah, isn't that cool?
Yeah. So this is like a second report.
Second report, yeah.
Same area, also more or less.
Oh, it
Bridge
was...
ROV, can we change the bearing to 315, please?
No, that was a little bit south of Iceland, I think. So, but along the same... yeah.
So it's blue transit.
Yeah.
Yes.
There's the very beginning.
Did you get lunch?
Yes.
That's good.
The soup is very good.
The fish is okay. And there is chicken.
I think the fish is going to be lovely. I miss eating more fish. So that might be good.
So we didn't say that we were transiting the... What did you...
I am putting in the transit and I have put in the... yes, we don't have the start here.
Yeah.
And we don't have location for that one.
But we have these.
Yes, and then you have to remember to put in... You have to remember to put in the start of the transect here with
Okay.
data.
Yeah. Genau.
5 minutes to tape change.
Okay.
That transit will be interesting because we are going really up.
Yeah.
About 500 meters, something like that.
And that will mainly be on the next shift.
Yes.
But that's okay. We can sit in the dry lab and watch. And people who are watching us live can just continue to be in the same place because very soon we will be on the next transect that has been planned by Julio, who is here to take over from us really soon now. And he has planned a really exciting third transect for the day. What is this? Can you zoom in on that? Are they Seriantids?
Yeah.
I think they're Seriantida. So these are anemone that like to... They have a tube under.
Yeah, that's first time that we see them.
Yeah,
today, right?
cool. They are large. Look at that. Yeah. Seriantida, and I think now we are getting those little sponges. So this is a different soft bottom community than we have seen until today on this dive.
Yeah, indeed.
Because all of a sudden we have the anemone, the tube anemone, the Seriantida. No, it's okay. And we are seeing maybe also some of those. Maybe I'm just dreaming, but yeah, look at those. We have some white dots there. No, they are
Not those.
no, but we are seeing Seriantida.
I'm gonna leave the notification like this.
Yeah, that's cool.
Francie will be back.
Yeah.
She's just asking if we should keep streaming the transit.
Yeah. That's fine.
That was nice to see. That was really cool. A little
Yeah.
bit of new community there.
Ending with new stuff.
Yay, that's always good. We're ending on a high.
Do you wanna take over?
Yeah, go, because otherwise you won't find any focus.
You wanna come with me?
I will stay until Francie comes. That's okay.
Okay.
So Julio is taking over for Sergi.
Oh,
It was great.
hello.
So this is Francis.
This is me.
Thank you so much.
This is nothing,
Right,
right?
welcome.
Yeah, this is transit to the next transect. But we saw the cool Seriantide, which was good.
Do you want me to
So
fetch?
let me just move this here so we have enough space.
Bye.
Right.
Enjoy lunch.
There was a seriante?
Yeah, several. There was a field of Seriantide.
Are those shining bioluminescent?
I am not sure. We do know that other anemone sometimes are bioluminescent. Actually, in the deep sea, quite a lot of the animals would be bioluminescent, but many of them are mainly visible bioluminescent when they get a little bit disturbed, or if they are, like hitting a screen or something like that. So there are several different reasons for being bioluminescent, either to attract food or to scare away enemies or to inform enemies that you don't need to eat me, or maybe just to hide. There's this really cool thing that some animals are doing to become almost invisible, so they are using counter shading, so a little bit higher up in the water column, around 1,000 meters, your stomach can maybe have a little bit of bioluminescence, because if your predator is sitting under you and wanted to eat you, you are looking just as light as the light coming down from over you, and then you become invisible in a way.
Everything is invented.
It is so cool. I always wanted to have Harry Potter's cloak of invisibility, and that's what they do have.
Really, a little bit shiny on your bottom.
I would like to be shiny on my belly. Shiny belly would be nice,
Shiny
huh?
belly.
Okay, 1 minute until tape change.
What is this?
If you're going into the blue transit,
Yeah?
if you're going into the blue transit, we will just join the video, and the video will stay on, but we can stop the recording.
So,
Yes.
so we will have a shift change in tape change, and then a shift change in about 20 seconds. And then Franzi will take over for me.
Actually, we will probably go in the blue transit now.
And then we might go into blue transit to the next transect, because Julio has decided on really cool next transect. But now we should change the tape, please.
Yeah?
Yeah.
1, 2, 3.
Be a bit too far away, maybe.
How long do you think the blue transit will be? So it's like 800 meters. Yeah. Unless we found something interesting in the way. It's like this.
45 minutes to an hour.
Yeah.
Yeah, okay.
Roughly.
Yeah.
Roughly.
But we will continue, we will continue recording, right? We are continuing recording, right?
Yes.
What about the stream?
The screen will continue, but they will have a treat.
Sorry?
Bring Charlie V.
ahead, RV.
Can we change the bearing to 325, please?
Changing to 325.
Okay. 1, 2, 3.
So we started the new tape, and I think what was happening is we are going into a blue transit, so we will lift the ROV a little bit off the seafloor to be able to move slightly faster toward the next transect that Julio has been looking up for us, designing, picked out from all of the wonderful bathymetric maps that are already there. And we will keep the live stream going, but if you need to go and get a cup of coffee, this might be the time to do it. Because we will, of course you will see the video all the time, but we will turn off the sound now for a little while, so you don't have to listen to us chattering.
Yeah.
And it will probably take 45 minutes, maybe a bit more.
Yeah, so check in if you're super excited, maybe check in 40 minutes, start to tune in and have us in the background, and you will hear when we get to the next new place, right? Yeah.
See you soon.
See you soon. Let's do it.
Okay.
Thank you.
Normally the dry lab, you should be able to hear us now.
So we're pretty much. That's the ship. Yeah. Alright. Let's keep going. There's all that line.
Dry lab, can
That corner. Right.
the two red dots on the microphone, can you hear us?
Shift cam.
Hear you all loud and good, thank you.
Shift cam is on one. Alright.
Thank you.
So I can take care of this. You take care of the annotations.
Will do. Tell me when you're ready.
Yeah,
Okay?
and zoom and stuff. We're ready?
We're ready.
We're ready.
Okay, let's do this.
Let's lift off.
We're now on our third transect, 2,548 meters.
And we are a crustacean expert. Can I call you a crustacean expert, or is that more specific than that?
you may. May I say that's a sponge expert, Paco?
Absolutely. And we have Andrea as our pilot.
Hello.
And Patty,
Co-pilot.
co-pilot.
And we have white sponges on rocks.
Yes. So we are still climbing on the little hills around Mohns Treasure, uphill.
What are these sponges?
Amphidiscalamonae. Where is the book? The tulip one. That's on the left side.
So a
Or
tulip.
you can write tulip glass sponge. Yeah, yeah.
Tulip. I may zoom in a little bit.
Yeah. Yes. Again, the tulip sponge here, and Asconema megaatrialia, those vase-shaped brown glass sponges that we see here and there. And for those who have seen our previous dives, we see a little gray sponge that's covering a lot of the rocks, and we're trying to put a name on it. So
It's a fish.
that's a fish.
Recorded. Good
Yeah.
idea. Hello, Grant. Visiting us.
Would you like
Which
to
is
see
nice.
this?
I would like the file from the transect start stop file.
This one.
Thank you.
Mhm.
Do we have an extra sheet for? No, this one. We should.
That's the one, yeah.
Yeah, so you follow on here, please. So this is our start.
This is T3.
This is T3 with
Same dive.
2,540 meters now.
21st. So we started at
Lots of sponges.
Yeah. This is sponge country again.
Yes. Rolf, can we slow down a little bit, please? Then the viewers don't get dizzy when we zoom in.
Crinoid on
That's on it.
and something.
Crinoid and a decapod.
Yeah.
First I thought it's a sea spider, but it's another crinoid back there, also on the sponge.
It's
So they're trying to get higher in the water column. So they climb up on the sponges or on any hard substrate they can find. Because the current is slightly different from directly on the seafloor. And they grab the little particles out of the water. Oh.
You need more time, Anna?
No. We may continue, please.
Okay. That's filled.
Can I grab
That's
that
an
that you just completed, that piece of paper? And I'll bring it back.
Sure. So a lot of the what I'm calling now the pom-pom polar sponge is those white bushes. There's a lot of them here.
Hi guys.
Hello, Jenny.
Hello.
And this is typically a volcanic rock field. And apparently sponges love it because there's so much hard substrate to overgrow. So if you were looking for your animals, where would you look for in this type of habitat?
Well, some of them sit on sponges.
Mm-hmm.
And, well, I'm working with amphipods. Some of them sit inside sponges, so if you take them apart. Lots of them on soft sediment. But they have all kind of life forms. So if you have like the scavenger guild, you'll find them when there's something like a dead fish or a whale falling down.
Mm-hmm.
So they're attracted by the odor, the
Mhm.
smell. And then
Do you
you'll
know-
find lots at once.
Do you know if they eat dead sponges?
I am not sure, but some definitely eat sponges. I'm not sure because they have the spicules of the sponge in the stomach.
Mm-hmm. Can they live in the crevices between the rocks as well?
Well, for sure.
Yeah?
I think a lot lives between those rocks and crevices. It's most of this life here is so small that we can't see it immediately. And all these wonders occur when you look through the microscope in more detail. Are these crinoids again, coming up on the left?
Yeah, with some shrimps on the left as well.
The red things floating around are shrimp. And the brown parts
And
are
in the middle, this piece, what looks like a piece of wood, is a stem from a glass sponge that probably died, but the stem stayed, and it will disintegrate, and the spicules will probably fill the crevices here.
Is that us again? That one?
That is, yeah, it's probably fallen, yeah. Yeah. I mean, it's probable that sometimes you have landslides, and maybe they take up and make the sponges fall. I don't really know.
Well, Paco likes to point out how old sponges are. They are the oldest, but I have to say that crinoids are also super old. They're also from the time of the dinosaurs.
Yes, that is true.
I know.
But sponges are one of the oldest, because there's still a huge debate in the scientific literature between is it ctenophores or is it sponges that are the first animals to emerge on our planet? And it's been a debate that's been going on for more than 20 years, and we still don't have a clear answer. So every year there's one or two papers that say yes, another paper says no. So it's still very unclear, which is really interesting, I think, because if sponges appeared after, that means they probably have simplified their bodies secondarily in evolution, and that is very interesting.
It is. There is a debate of the origin of life, and it started in the ocean for sure. And most likely, or very possibly, at some height of thermal vent sites. And here we are only 30 kilometers away from the active vent sites that we'll visit next days. This is an inactive site, which has been discovered, I think 2002.
Mohns Treasure, you mean?
Yes.
I don't know, actually. Do you know, Jenny, when Mohns Treasure was discovered?
No,
I think-
I'm just thinking.
I think it was 2002.
I talked to Anna-Helene about it yesterday, and she said that she remembers very well the discussion about how to name this site when they first discovered it. But I forgot.
Look at this little amphipod.
Oh.
Is it an amphipod?
Yes, it is.
Wow, a big one.
It had such a red stripe in the middle.
No, I didn't ask her myself
Okay.
when it happened, but oh, there's another one.
Yeah. So those little red.
Oh.
There's a shrimp or a mysidae. I don't know how to recognize them.
A decapod.
Or decapod. Yeah.
Photopod.
So right now, with my microscope, I'm trying to identify these gray patches of sponges that you find on all rocks. I've been drawing the spicules and looking through the literature. Can't find it now, which is getting exciting because it's so common, but maybe it doesn't have a name yet.
I'll just zoom in a little bit. Maybe.
Wow.
Have you got a name for the fan-shaped sponges?
No, but we collected it today, so next time, until next time, we might have a genus name, and then we'll take it from there. But at least I really want to know from which group of sponge they belong to. They don't have a name yet.
It's a stalked crinoid.
Oh yeah.
On the right side.
Yeah, yeah.
Speaking about crinoids, they come in there are two main shapes. The one of the stalked crinoids are also called sea lilies, and the other ones are the feather stars. The latter can move around. They can swim. Oh, what's that? Sorry, too fast.
This looks very
This
steep.
goes up. What a pretty amphipod.
Oh wow.
Come on, go right for the lens. That's a nice one. Come on, sit. Can we stop, please? I would love to. Can you stop?
Yeah.
Please.
Do you want me to stop?
Well, I would love to have a picture of the amphipod. If it sits down right in front of us, I would be.
Yeah.
What are you
No,
doing?
but because it's zoomed, if you
Sorry.
wanted to move less, yeah.
It's too fast for us.
Yeah.
Wow, this is beautiful.
Now we'll have a little plume, which will settle quickly.
Wow.
You're stepping a little bit
So
You're stopping a little bit. We'll stop a little bit and have a play what we can discover by zooming up close.
yeah, we can enjoy the view as soon as it's cleared out. So this dense accumulation of sponges can be named depending on the community we find in them. Usually when there's 2-3 key species of sponges, we name it a sponge ground. If it's more diverse, like it's the case in Antarctica, for instance, where there's sponges, very dense accumulations of sponges, but a lot of different species, then we talk of sponge garden. And here, since we don't really know all the species, that's something we will have to investigate. Is it a sponge ground? Is it a sponge garden? So far, it seems to me that there's not a huge diversity of sponges, so it might be a sponge ground. So here we have a close-up on the polar pom-pom sponge. Ooh, should we zoom there?
Yeah, just a second. I wanted to get the overview first,
Okay.
so what other animals we can see. Yeah, no, okay, move there, please. So, all good.
Yeah, so the brown sponge, I think, is Hemigelius arcofer. It's a species we've identified here on the boat. It's been described a long time ago, but we had never seen it alive. So actually, for our viewers here, you're also probably one of the first humans to see that species alive, pumping, filtering the water. And that's pretty exciting.
I'm not quite sure. It's hard to see. Is that an anemone?
That's an anemone, I think. Yeah, on top of it. Yeah.
no.
There. Yeah. Yeah.
I'm actually surprised.
Can you zoom in a little bit more?
Nope.
No, you're maximum. Okay.
Next. I'm surprised that there are so few bottle stars and sea cucumbers, because quite often in this kind of terrain you would see them when zooming in.
So here again we have Hemigelius arcofer, but this time a much more developed specimen.
Would you mind?
Yeah, it has produces these fibers that come out of it, giving it the impression that it's very hairy, like the woolly mammoth. we could call it the woolly sponge.
Wow.
It does look lovely. And
I
you
really
see,
want to touch it.
you'll have some spicules.
So again, it's the
But
first
look at
time.
this. So I'm trying to zoom in on the soft coral actually. So this is not a sponge, the left parts.
Yeah, we have colonial Ascidians, soft coral, and the woolly sponge.
How's it called again?
Hemigelius arcofer.
Okay.
And I didn't know that last week. So it's because we collected it, and with Julio we've been trying to find a name for it.
Yeah.
And we finally found which species it was the last few days. And now we see it everywhere because it's one of the really common ones. So that's one advantage of exploring the deep sea with a robot, is that we can start to put names on things we see down there.
Yes. We may continue.
Yeah.
Thank you.
Sure.
I better load my soft coral. And Ascidian.
And woolly sponge.
I think that's one of my favorites so far. It's really cool.
Yeah, it is.
The name is not though. I I need to write it down.
Yeah. I can write it down and show you later. Yeah.
Wow.
There's the tulip sponge again at the bottom of the screen. It's a glass sponge with a stem. Very beautiful and delicate sponge.
So most of the life you can see here is filter feeding or suspension feeding.
Yeah, absolutely.
And all this nutrition comes from the top, falling down. Oh, let's zoom in. So there's an anemone.
I think. Like this one? Or was it smaller?
No, it's all good.
Okay.
Another one. Some more crinoids.
So many.
Yeah.
Wow.
Crinoids have this many arms, and like most of they're related to like sea stars or sea urchins. And they can regrow their arms. So when one breaks off, it regrows. It's a nice ability.
Do crinoids have predators? Do you know if
they have predators?
Well, I guess sea stars, because in the previous dive they saw sea stars. But the crinoids that are not stocked, without a stem, are they also eaten by sea stars? Do we know?
I'm trying to think if I've seen an image. I haven't.
Because that's a question we often get, is who eats who in those environments?
Yes.
Who's the apex predator? Because there's no tyrant, there's no dinosaur, there's no lion. So who is at the top of the chain? What do you think?
Well, if you go to the very deep, it's amphipods, I have to say.
Amphipods?
Actually, don't you laugh, because if you go down the deepest trenches, you know, below 6,000 meters, if you go down to the Hadal, the fish can't cope anymore. Oh, very occasionally they do. And then you get the large amphipods. So I often show them as top predators. It's like a lion in the savannah.
I have another possibility for you. I would say sea stars.
Nope, they can't cope so deep.
If an amphipod battles with a sea star, who wins?
The amphipod, of course, the sea star can't even cope when there's no salt. You know, they give up in the Baltic. So they need a very salty environment. And amphipods, on the other hand, occur also in freshwater and in caves.
Okay, so they're more adaptable, you're saying?
2 points per station, yeah? Okay. They're more adaptable, and they have adapted everywhere. But this is sponge heaven.
It is. It is, and again, I'm saying that this type of habitat I have never seen before. We think it might be a new type of habitat that should be described, but for this we need to know which species are present. We need to understand the community that we're seeing. So this morning, I know that my colleagues have already collected a handful of these sponges. And we will continue to do so.
Do you think these thin fan-shaped sponges are a different species than the thick fan-shaped sponges?
Yes. Yes, and I know they've been collected this morning, so I hope I will have a name or a genus name for you tomorrow. But right now they've been people have been taking pictures, previous expeditions, Norwegian expeditions, so they've seen them, but they didn't collect, so we don't know what they are.
Okay.
So I'm very excited when the robot will come with those samples.
Okay.
Tonight, probably. I'm very excited to look at these.
Many of those sponges will reproduce by larvae. So
there's all types of reproduction. So some will have larvae that will swim away and settle somewhere else. Some will release little babies, already formed little babies. And many of them will just send their sperm and oocytes in the water. So fecundation will take place in the water column. And then it will produce a larvae again that will be taken up by the currents, water currents.
the same is true for the crinoids, or for many specimens that we see here. And often these larvae need a hard substrate to settle. So when they're looking for a landing place, it can be a rock. That's also why this habitat is so super suitable for them, because you have all this hard rock scattered amongst the soft sediment.
Absolutely. But I don't know if you've noticed, a lot of sponges are very clean. There's nothing growing on them. And on the other hand, you have some sponges that are very social in a way that they attract a lot of other organisms, and they build these whole little communities. And the difference between these two types of sponges is still poorly understood. We think that some sponges are happy to have other animals growing on them for protection against predators, maybe sea stars, maybe amphipods.
There's a woolly one again. There was a woolly one.
Aha. Right there at the
Yes.
bottom? Yeah. Yes.
There it
Yeah.
is.
And the anemone on the left.
And the sponges that are clean probably produce a chemical to deter animals from sitting on them. And these chemicals are very interesting to us because we could actually reuse them, maybe to make products that will deter also organisms from sitting on the surface. And I'm thinking, of course, of the paint that you use for boats, for boat hulls, because currently we have a lot of paintings that are very toxic to the environment. But if we could find a more natural compound, especially coming from the sea, that would be very interesting. So there's a lot of research groups in the world trying to extract, we call them anti-fouling compounds, for applications, for instance, for the ship industry. And many of these anti-fouling compounds are coming from sponges, because you can see how clean they are. They clearly have found a chemical to
Can we stop? Please?
clean themselves.
Please. Can we stop, please? Thank you.
I can already hear you getting very excited.
I am excited.
And
I love
see
seeing
why.
this live.
I see why.
So please enlighten me. I don't see why. I see the sponges.
It's a sponge.
Oh no, no. This one? This little guy?
No, to the top right. Sitting. Sorry. I'll show you. It's right in the middle. Now turning at 6 o'clock. It's a decapod. Turning up.
Yeah.
It's upside down.
Yeah, I see it now. Yeah.
It's quite cool.
I clearly don't have an eye for these small crustaceans. Not yet.
You can see the eyes glowing and the long
there's
antenna.
an Alfie joining the party. To the
Yes, but
Read your reveal, stop please.
But it's swimming along. It's very hard to see when they
might
don't sit
settle.
down.
Come on, buddy.
that we talked about larvae.
It's actually looking at us. You see? It's towards us. It's looking at the camera.
Yeah, I'm focused
For
on the
sure.
alphabet.
Yeah, it's in the alphabet I'm talking about.
It's
Yes,
set down.
Oh, now you finally set up.
Yes.
How could you mind steering a bit to the right?
Yeah.
It's actually a good question why they have eyes, because we're now in 2,500 meter depth. We don't know. Some of them. Stop! I'll try and zoom. I'm zoomed out. Some of them stay down in the dark all the time. One assumption is that there's a lot of bioluminescence going on and illuminates parts, so they can attract partners or irritate predators or attract prey themselves. There are many theories.
Would you like to try to collect that amphipod?
Nope.
No?
We're too far.
Or too far?
We're too
yeah,
far.
now I see, yes.
We're too far, and the arm of the robot will not go that far. Maybe continue. Okay, thank you.
Great, ROV, can we resume the move, please?
Resuming move.
Well, we talked about larvae before, but the critters that we just saw in red, the shrimp and the amphipod. Well, it's actually only true for the amphipod. They don't have larvae. They have a brood pouch, and they take care of their young.
So it's the mother that takes care of the young?
Yes. This question might sound a bit bizarre because often in the animal kingdom it's the mother, except for the sea spiders that we encountered yesterday or the day before. And it's a dad. We're now waiting for the ship to catch up with the ROV. We might as well zoom then. Yeah.
So on the image you can see two little red dots. These are the lasers coming from the robot. And they are 20 centimeters apart?
Yeah, 20.
20 centimeters apart, so that gives you, in the audience, a feeling for the size of things.
Like this critter.
Which one did you see?
Oh, just out. I'm really surprised that we don't see any battle stars anymore. None.
Look, sorry, I just wanted to look there.
That's it.
Okay.
Ready, off you go.
Yeah?
Yes, we're ready.
Yeah. So bridge ROV.
ROV raised.
Yeah, we get a bearing change to 310.
310.
Thank you.
So it's very tricky because we are connected to the boat with a cable, and so the boat is moving with us, following us, and we constantly, I mean the drivers here have to constantly tell the boat to either stop or move faster or move slower, depending on what we're doing with the robot. So it's
I forgot the English word. What's it called? An
umbilical cord?
Umbilical cord.
Yeah, kind of.
Well, it is.
Like the mother to a child.
It is. It's.
And here in the ROV, in the robot shack, where we control the robot, we are actually sitting on the boat. We are in the container that's on the boat. So, oh, jellyfish.
Yeah.
ROV raised. Paddy, just to confirm you want speed 0.1.
Yeah, thank you.
There's another tulip sponge on the top right. This globular glass sponge sitting on the stalk.
That's the big round, the donut-shaped thing. The donut-shaped sponge. No.
That one? Is that the
That's what
tulip?
I've been, yeah, the tulip. Yeah, yeah.
That's big.
Yeah. Yeah.
You have very strange tulips in your country.
Oh, now I see it. Sometimes the perspective is funny.
Mm-hmm.
It looked a bit like a donut for like a couple of seconds, and then it changed to...
Are you hungry, Jenny?
No. But actually, Francie and me were talking about donuts at breakfast, so maybe I'm a bit
Influenced.
influenced there, yeah.
So here the polar pompon is everywhere. And yesterday we got a couple of specimens from that interesting species, and we found that it was reproducing. So our researcher, Sergi, was looking under the stereo microscope, and he started to see things inside the sponge, which could be eggs or larvae in development. So that's very interesting, because sponges, like many other animals, have a reproductive stage. They don't reproduce all year long, because reproduction requires a lot of energy and food. So,
But we don't yet know if this is coupled to a time of the year, or if this
absolutely
is year-round, because there's so few samples that it's always just a glimpse of chance discovery.
We should collect all year long to be sure, to actually pinpoint to which season they reproduce.
That is true for all organisms that we see down here.
Ask anyone.
This one?
No, to the, that is now moving left in the...
Ah, there. Yeah.
Megatriana.
Yeah.
Thank you. 2 weeks into the cruise and I got it.
The name comes from atrium,
Okay,
but huge atrium. Atrium is like a vase shape.
right.
So mega atrialia,
Ah,
which comes from atrium.
that's why the double A. Megatrialia.
Yes, exactly. Yeah, I should have explained that to you when I gave you the name. But it's a good way to remember it.
Now it will stick with me forever. Yeah, now Marco, this was the
Well,
key to...
We're learning lots of new names from each other. Most of the deep sea critters don't have English names because they're so uncommon. And our jobs as taxonomists is naming new species. Oh, there's a jelly.
Jelly.
Oh, our tape has to be changed here in 10 minutes.
I really like polar pompon though. That's a funny one.
We've been discussing this with my sponge colleagues.
We'll just remember, we'll keep it in UTC yet.
Okay.
Grant, you want to stay? And because the tape has to be changed.
I can do some tape.
What do you need?
No worries. I can bring it up.
Oh, I can do it. It's 10 minutes left.
The screen where I'm entering just went black. So it's like no. It's come back again.
Sorry?
That's the blender.
There's the woolly sponge.
This is the back.
Nice ones, eh?
Verticalis.
Deep sea shrimps.
Yes.
So as you can see, the polar pompon, they are easy to grab and collect, but there's so many other little sponges sitting on those rocks that are really
What's that? Is that
tiny,
a sponge? Can
tiny.
you spell to the left? Thank you. That's an anemone. Upside
There's
down.
the woolly sponge next to it.
Yes.
So a lot of these little crusts on the rocks are sponges as well, and they are complicated to collect. So usually we take a small stone to be able to collect it, because otherwise it's impossible to scrape those stones. Here's another woolly sponge.
Yes, definitely.
Very nice.
There was something going on.
Quite
I think that one had a parasite.
Yeah.
It really looked like it.
Against the current, like you can do, you can then find the current one.
Where you use the current ships.
How easy is it to stop to collect on such a steep? It's possible?
What do you want to get?
I'm just thinking ahead to know if I tell you to stop, it's possible or not.
I'm just asking because if it's too close, I don't know if the slurp would be too close, but we can try or
because of that patch.
Okay. Okay. It's not too bad.
Okay. Thank you. Wow, it's so beautiful.
Yeah, it's incredible.
And I mean, you would not think of a better polar scene as these white sponges everywhere, like snow on rocks.
Look at the tulip. To the left.
To the left, yeah, absolutely.
Really cool. Wow.
Wow.
We really have amazing pilots here too, huh? Flying this slow, just so slowly
It
and
is
steady.
So smooth.
Amazing. Thank you.
Climbing up the hill.
Yeah.
Running up that hill.
Running up the hill, exactly.
It's just amazing.
It's hard to see on a camera in that angle, but this is a very steep slope.
I've noticed.
Yeah. Keep doing your bit.
I started with 204.
Almost everything that we can see here is a sponge.
Yep. But the big difference with the top of the seamounts that we've seen previously is that there is no spicule mat made of the dead sponges. Here it doesn't seem to take place. And that's interesting as well. Here it's just rocks and the sponges sitting on them. While at the top of the mountains it's sponges sitting on dead sponges, on the remains of dead sponges. They've created their own hard bottom.
These were crinoids again.
Oh, small underwater cave.
How many fans?
Mm. So we are at 2,481 meters. Wow, look at the amount of small sponges and feather stars.
Excuse me.
And shrimps. Oh, this is very rich.
Turn off the lasers just for a short moment.
Okay. Should I zoom out a little bit or?
Okay. Well,
yeah, like this,
zoom
yeah.
out,
Perfect.
and then we'll stop.
Stop?
Yeah. Yeah,
Yes,
if you
please.
can go down a little bit to see the...
Okay. There's some plume chasing this. But
Okay.
we can wait it a little bit.
It's quite interesting how they go on the edge of
Yeah.
the rock.
Yeah.
Obviously the best spot
Yeah.
for the...
Radar, we all stop, please.
It's a good moment to change the tape.
Yeah. So we do that?
Yep.
Okie dokie. So we're going to stop in 3, 2, 1, stop.
Tell the dry dive, guys.
Yeah, we're changing the tape.
Shall we tell Jason? Mike?
I think we are hearing.
They can hear us well.
Ah, beautiful. Why are you laughing?
Lots of colors.
Yeah, yeah. It's a good thing the crustaceans are there. They bring much more colors than the sponges in this area.
I think it's
Okay, recording on mic on in 3, 2, 1.
That's true for many situations in life. Crustaceans bring more colors. Oops. As we said, these are filter feeding, and they have some mucus on their arms, on the feather stars, and little small cetae, and they're catching anything they can. So, say plankton, but also eggs, or even tiny fish.
Mm-hmm. Can I zoom on the shrimps? So what do they eat? The shrimps?
But look at these feather stars, how they're stuffing the... they've got this one opening in the middle, and when they caught something, they stuff it in. So you can see the arms going into their mouth.
Mm-hmm.
Like sucking the units off,
yeah,
and then releasing them again, catch more. You see the left one there, it has nearly all arms in. So
yeah,
she just had a feast.
Mm-hmm.
What is this wormy thing here? There's a white thing under the under the crinoids, which we can't see properly. Oops. And you also see you see all this orange little units on the rock.
Worms?
I assume they're tubes of worms.
Yeah, tubes of worms.
Look at me.
Yeah, he was jealous.
Like a dog paddle swim.
So in the previous dive, we have collected those hanging round-shaped sponges, and very interesting. They produce a lot of mucus, a lot of gooey texture, kind of viscous texture, and we don't really know why. But they produce a lot when we brought them on board.
And that's a real nuisance if you have other samples in the same pot, pottle.
Yeah.
Because we are also doing DNA extractions, and what you don't want your clean, lovely little animals to have mucus all around them. Because when you then do the sequencing, you sequence sponge mucus. That's what we're trying to avoid. So that's why we're also sampling in different units. Can we zoom out again? A little bit to the left, please. This one. What is this?
The stick, you mean?
Yes.
Not sure.
I don't know either.
Maybe remains of a stock, actually of a sponge stock. Probably.
That's what the sponge. That's what I think as well.
Yeah. So again, these gray patches that you see here are sponges as well. We call them encrusting sponges. They are little crusts, so they don't grow much more than that. They can expand on the surface. And this particular one we have collected, and we're currently trying to put a name on it. And we've failed so far, which is kind of exciting because maybe it's not been described yet. But we'll see. We will continue to... We will update you on what this species is.
Down here it's mainly sponges encrusting. In the more shallow regions you also have the encrusting corals. And then in the photic zone, so when there's still light, you have the encrusting algae. Often all competing for space. But down here it's sponges only. Zooming out.
So what do you think the shrimps are doing? Are they just picking up stuff on top of the sponges, or they're just sorting and eating really tiny organisms? Or
I have to disappoint you, I don't think they really care about the sponge.
Okay.
I think they just use it as, well, wherever they can sit.
Aha, so they're actually eating in the water column?
Yes.
Aha, okay.
Filtering. So.
Okay.
Shall we fly on?
Yeah, we can fly off. Oh, look at these fantastic volcanic rocks with clean cuts. This is basalt for most of them, and you find basalt on the upper part of the oceanic crust. It's the magma that solidified really rapidly in contact with the seawater.
Photograph from the back.
Where? Oh. Yeah. Right. The sponges are smaller here.
there's still a lot of encrusting ones. We're now at 2,480 meters.
Bless you.
Thank you. You can see a little red jellyfish.
Yeah, little red jellyfish
swimming in.
On your right. Oh, and the second one.
Yeah.
Or yeah. That was different.
It was definitely something different. I'm not sure if it was a jellyfish.
Oh, but look at that one.
That one.
Beautiful. Ooh. It was a shy one. It was very shy. It seems to me that the crinoids are also very social, because I mean, several could live on another neighboring rock, but they like to be close to each other.
Well, the crinoids are the feather stars.
Yeah. I think that's also a glass sponge stalk, maybe from Colophacus.
With the feather star just crawling up?
Yeah. It's taking advantage of the remaining stalk.
Well, feather stars occur in all oceans.
Mm-hmm.
Especially actually in the tropics. And there are around 600 species. How many sponge species are?
10,000.
Yeah. The same as amphipods, by the way. I'm not making them. It's also around 10,000. So on our internal battle here. Two. Well, I think Paco wins here in this territory.
No, but I mean the number of mollusk and crustacean cannot. I mean we cannot match that. They're super hyper diverse compared to sponges.
Yes. But you've got a phylum.
Yeah,
and I have an
we
order,
do.
so it's
But I have to say this as well, is that 10,000 sponge species described, yes, but probably that same number of undescribed still to be discovered and described and named.
That is not only true for sponges, of course, this is true for all these groups. That the number of undescribed at least matches the undescribed.
And that's why we're still finding new species in those habitats, especially here, which is pretty much unexplored. Here we have a beautiful round rock. This is a typical pillow lava. So you will find pillow lavas on top of the oceanic crust, and they're typical of magma coming up and solidifying super rapidly when they meet the water. And that's how you identify old oceanic crust. So if you wander off in the Alps, you will find pillow lavas, because before there was an ocean between Eastern and Western Europe, and this ocean closed up, and the oceanic crust that was in the middle was mixed with everything else, and now you find it on top of the Alps. You will find pillow lavas, indicating that there was a sea here before. So how long have you been working with amphipods?
Oh, ages.
Was this but from the start?
Can I say last century? Yeah. Sounds terrible. more than 20 years.
Did you do your PhD on amphipods?
Oh, on groups of crustaceans, actually in Greenland.
So
So
not-
masters and then my PhD on Antarctic
Aha.
stuff. Mainly amphipods, true.
How many species have you described since?
I have to admit I have not counted, but around 50, I guess.
Around 50. Okay, you have to
50.
check. You have to check your homepage on Wikispecies. You should have your author name there, and maybe someone did that count for you, because they're really good at that.
It's my lack of vanity that I don't know the species. I do know how I would potentially check that.
But I'm telling you, probably you have a web page on Wikispecies, and people write the new species you've described, and then you can easily count them.
We look into worms.
Yeah, can we continue the move, please? Point 1.
And
31010.1.
Thank you.
It's a fantastic web page. It's the World Register of Marine Species. And if you have doubts of name spelling or when things were described or who described it and what publication, it is great to look. Also getting more features and where things occur.
Yeah. So it's called WORMS, the acronym of World Register of Marine Species, and I think last week or this week they just passed the bar of 250,000 species in the database.
Really?
Yeah.
Yes.
So
yes.
250,000 marine animals now registered in the World Register of Marine Species.
Wow.
Yes.
So that's pretty much the same number of plants on our planet. So it's a lot.
Wow.
Yes. You're an editor?
Sorry?
Are you an editor on worms?
I am an editor. Are you an editor?
Yes, I am. So that's why we know this, because we got this newsletter we're celebrating. It is a fantastic page.
So those pages are maintained and curated by people like us, experts
for their group.
Yeah.
And I think they're worth gold, especially when you're looking up names that have changed over time and things get moved, like to another genus when more species have been found. Then you can take track of that. And you also see what other groups have been described.
I think this is Sacocalix.
I will enter.
Can we collect that one?
Sorry, can we sit down here with the rock?
Too far?
Or is it too...
We can try, yeah.
So I've been looking for that sponge since the beginning.
Yes.
Want to slurp?
No, it will be destroyed in 10,000 pieces. So
can we use the shovel?
Yeah, you can use the
so
shovel.
it is very light
Where is it going to go?
and very fragile. There is an empty box on either side, so whatever suits you better.
Port side of the shovel.
Okay.
What's that shovel?
Port side. 1, 2, 3 or 4.
So it's probably connected to the rock by a stalk.
Yeah.
So if
it floats.
if you can break the stalk, then I think you can get it. But it's going to be very light, so that's why it might be a bit tricky.
Might be attached to the rock, so you might get the entire rock.
Stop there, please.
Yeah, but that's a big rock.
Yeah, I can't.
We don't have the.
I can't try.
Can we do a close up first?
Yeah.
And
Sorry? Yeah, it's 50.
Just move it to center first, and then I'll zoom. Thank you. Oop, now the rock is moving.
we're moving away.
We're moving away.
Sorry.
No worries.
Just trying to look at the umbilical.
Yeah.
These primates are living a feast.
It's a little bit higher.
Yeah,
Here.
that one.
So So as you can see, that's a very steep slope, and you have to be a really good driver to try to settle on this slope. Let's see if there's some of your animals in there. Why not? Maybe there's something inside there, right?
Yes, there is, but that's why it's so fantastic to get them fully intact.
I can center it a bit more.
Okay.
Like this?
Yes. That's a little shrimp on top.
Yeah, I see that.
Pretty?
But there's 1, 2, maybe 3 other things there, no?
I see them. I do see them.
Yeah.
We can't focus that any further. So
No.
I can try the manual. Sorry, it might be out of focus now for a little while.
We're trying to focus on the... No. Okay.
Well, come on. I'm trying.
Yeah. So you can see little crustaceans in the sponge, either for shelter or to eat it. And I've been looking for that sponge because, again, we think we know the genus, but we don't have a species name for it. It's a glass sponge. Glass sponges are usually very thin, so very fragile. Could we have the lasers just to have an idea of the size? Perfect, thank you. So you can see that sponge is about 20 centimeters large.
Anna, do you see your arm?
Yes, of course.
This one?
Yes.
You know, there was no reaction, so I was, I had to check in.
But I
If,
silently moved the camera over so to get the zoom.
Okay, I'm gonna leave.
Look at this pretty creature. Come on, go to Paco's sponge and just sit on it while we sample. He's hiding.
Yeah.
He knows what you're up to.
Are you all zoomed in?
Yeah.
Almost.
Wow.
Oh, yeah.
So the little bright lights that you see is the eye, right?
Yes, it's crawling to the left and has its long antenna moved forward.
Do they have one eye or 2 eyes or complex eyes?
Depending on the group, this one has 2, one on both sides. I assume it's the family Calliopiidae. They're quite common in this area. And Anne-Helene and I have described some, also from this area, also sitting on sponges.
Mm-hmm.
So, if this is 20 centimeters, where do you want the shovel to come from? From the right?
I don't know.
This will be tricky.
The shovel will come from your right arm? Yeah. Then you will place it in one of the boxes on this side as well.
What I'm going to have to do first is take the shovel out and then re-grip it. It's facing the wrong way.
Okay.
Are you ready?
Yeah, I'm ready.
I'm ready as well.
Yeah, we're both ready.
How shall I call the sponge?
Saccocalyx. S-A-C-O-L-Y-X. Saccocalyx.
the other pictures.
Sampling.
Otherwise,
And it's now 30.
So the robot is trying to catch the scoop, and for that it needs to be in the right position to grab it. So that's the first thing they would do. Are these chocolates for us?
Yes, for everyone.
I actually think they're further off pilots.
You know, I have half one left.
They're eating here constantly.
Excuse
Are
me.
these German cookies?
I was feeding the pilots with some chocolate pieces as well, not only myself.
Do you want to share this? Do you want a piece? Or
It's let them eat, but not
No,
during
no.
their operation.
Yeah,
Now
exactly, I
Now,
don't want
they-
to
now they
disturb them.
really have to concentrate.
oh no! Who is moving the arm?
can you zoom in a little bit?
Yes.
Zooming for the sponge?
Yeah, okay.
This one.
That's actually
Show on that screen which one it is.
Yeah, I've shown them.
Yes.
This one, right?
Exactly.
Yes.
Yeah.
Yes.
Yeah. So you can imagine how difficult it is to subsample a deep sea organism.
Well, 2.5 kilometers above.
Oh.
Oh.
Wow.
Yeah.
That's it.
You're
Oh,
a
magic.
chef.
Wow.
Perfect.
That was picture book. Sampling.
So
Tell which one.
Okay.
Yeah. So it's going to be box 8. So that's on your right bottom side.
Which one?
This one?
This
This
one.
one.
Yeah. It's currently empty, and that will be cast 13.
Oh, we lost it. We lost it.
Mm.
Can you see another one just while we're sitting?
No. Was it taken up by current?
Yes. Yes.
Sometimes.
Yeah.
Sometimes we find things on the rock. When we found something that. Is there another one?
We will try to find another one. Right now I don't see any more. Yeah, that's the challenge of collecting in the deep sea.
It looks so good.
It's not like going into a forest and picking up plants. It's a bit more work.
That was a jellyfish.
You want to put the shovel in?
Wherever the sponge will end up, it will regrow, like it will settle again.
It might
Yeah.
resettle somewhere else.
Okay.
Sponges can heal very rapidly and very efficiently.
Also, if you take a piece of a large sponge, it will regrow.
Yeah, absolutely.
Actually, like coral branch, it also regrows like an apple tree. Almost. Almost. I'm just zooming in while we're sitting.
Lots of these jellyfish.
Yeah.
You should learn
There's-
of the species.
There's two.
Yeah, there are many. Really many.
Okay. Very good.
Wow.
This is incredible.
Yeah.
Do you want anything?
It's like a painting.
Rorschach from Dryland, can you please put out the signal for a while?
Yeah, okay.
Okay.
So we are deploying our fisheye camera to get a nice shot at this cliff, this amazing cliff.
It's interesting how the cryonites are all on one side, on the feather stars.
Could you try to stay
And
on
the
that
right
side?
side, as well as the filter. More filter feeding is happening clearly on that side.
Oh, that's amazing! Look at that.
It is a steep cliff. Okay.
I think that's again
Camera
my species.
Oh,
But
wow.
could it on a flatter place, please?
Yeah.
It's
Yeah,
easier
yeah.
to stop.
No, but it's good. It means it's around. So
Oh,
we
these jellyfish.
yeah. And all these feather
We
stars.
continue the move, please.
Is the camera out?
Yes, it's there. So we need to take it in next time we collect. We shouldn't forget. Wow.
Can we turn on the lasers, please?
You're on.
Thank you.
Apparently the feather stars love it. They prefer that side.
Oh, look how many jellyfish.
Yeah.
1, 2, 3, 4, 5, 6.
Amazing.
So how deep it is? 2,450 meters.
That is very deep.
Do you know how many species of amphipods are described every year on average?
No, I don't.
No?
There have been a lot of discussion lately about cryptic species. So, to separate species from each other, when to call it a new species, because especially a lot of the groundwater ones look extremely alike, but they have a genetic distance, so they differ in molecular terms. That's very tricky to describe.
Two different species of jellyfish.
And an anemone?
So there's the red one, and then there's one that's
There
more
was-
transparent, or
Yeah, there was another species.
this one maybe, or I don't know. I think we saw another one that was transparent, more transparent.
Yes.
Oh, we can zoom in on it. Oh yes, finally.
Don't yet, before the zoom.
Finally.
It makes it so wobbly when
They're really
zoom.
active.
Yes, very pretty.
Mm-hmm. So the thing standing out now on the camera is the fisheye camera, in case some of you are wondering. And it's extremely fragile, so we don't when we don't need it, we try to pull it back inside the robot. But when it's scenery like this, it's really nice to have this 180 degrees view of the landscape.
We have a lot of monitors here, but we cannot see what this camera is filming.
Ooh, fish!
Slightly.
That's an amphipod, right?
Yes.
Is there a common name for amphipods? Like a general term, non-scientific term?
Yes, there are in different languages. As I think beach hopper.
Beach hopper.
Is the English term.
Sometimes I mistake a crinoid for a sea spider. And I just get all excited until
Me
I
too.
realize
Me too. I do exactly the same.
It's,
After we've seen so many sea spiders, I accept. They've got a similar color, a similar size, and they've also got lots of arms and lots of legs that are bent just before the middle part.
Exactly.
I had it a couple of times now that I had that mistake.
to be honest, this morning when I woke up and started to watch the video footage.
Paco.
I'm trying to follow the jellyfish.
They'll come to us.
Well, like this, you've spotted the huge lights on top of the ROV, without which we wouldn't see anything.
There was the second species. Yeah.
Of jellyfish?
Yes.
You asked for the name of the amphipods. Our Norwegian colleague Ann-Helene just told me in Norwegian they're called hoppekrebs. I don't know how to pronounce it, but hoppe, I like that.
So a little bit like beach hoppers, but
yes. But I think the beach, well, I don't like the beach as much as the hoppe. And in German it's a Flohkrebs. Look at this one to the right. What's that? Can we zoom in on that? What's this?
so there's a woolly sponge, a pompon sponge.
Lots of feather stars.
Escape.
You can see here that it's quite steep in the distance. I think sponges like the company.
All the feather stars?
Yeah.
Just entertaining them? Well, you may think so. I don't. They might be really annoyed that they don't get enough, so much food anymore if they're covered by feather stars.
But maybe
you
the
know,
feather
and
stars
not-
clean them as well. I don't know.
You wish they eat for themselves.
Yeah.
On your previous stop, these are very small stone sponges. So if you happen to find a small stone or on your next stop, if there are small sponges like this, it can be very small.
Yeah, I saw
Maybe
them.
they will make it to the surface.
Yeah.
And that would work wonderful for the picture.
Yeah. Yeah.
Not so big, very small things.
Yeah.
Small stone sponges.
We will try.
Nee. Okay, verstanden.
Keep up the good work, that's amazing.
Look at this one.
Thanks.
Look at this one. It's covered as well.
Immer mega Antrifolie.
Yeah,
that's good. Enjoy.
So let's find a little rock.
It wasn't a joke. That was actually finding a sample suitable
Yeah.
rock size.
So we have a lot of photographers on board, and they want to make nice pictures of these animals. So they also have requests for some species. And it's very steep and it's very hard rock.
Like being in front of a wall.
Yeah.
Going up.
So it's pretty much impossible to sample here because you can't get a grip on much.
Just climbed 30 meters because it's now 2,418.
tulip sponge. Didn't see it. Oh, there it is again. I don't know if it's the same fish.
I only locked it once. Will this camera stay out?
No, as soon as we will collect something, we need to bring it in.
When we collect something? No.
Yeah.
Okay.
But I think right now, since we can't really collect on the cliff, we just want a nice panoramic view.
It's quite large.
Yeah.
Looks like a potato.
Yeah.
So we can start.
Asconema mega atrialia. This is a glass sponge. For instance, something like this, you collect, or it's too complicated?
I would have to have some thrusts.
So what you're going to have is you're going to have the ROV right up just there, but the arm has only got that much space to move.
Yeah, okay.
I think in the interest of this precious camera, we don't want to do that. That's why I'm asking if we can pull it back in, because if we get this kind of sampling, we can't do. Like on the fly, it will scratch. It will be too close.
Yeah. No. Absolutely.
We've moved the camera in first before we've done any arm work.
Now it's a little bit more flat, so
okay.
If you find something.
Yeah, then I'll try to find the one that got away.
Sorry about that.
Might be that, but I'm not sure. Okay.
It looks like lace.
Yeah. So is there a lot of current right now?
Well, when we sit down, we put the thrusters on to suck the dirt into the thrusters to keep visibility. Because it was so light and it was moving in the scoop, it flew out and couldn't even shut the thrusters off, otherwise we'd have lost it completely.
Yeah.
So it's a balancing act.
Yeah.
But anything light like that probably would be best in the port drawer because we've got room to manoeuvre. So if you do get another sample like that, best to put it into the port drawer if you can.
But then with the arm?
Then the arm will get to it a lot better than right on itself.
Yeah.
As you go out further with the arm, you've got more dexterity. Closer in, you've got very little movement, so it's more tricky.
Okay.
Okay, thank you.
Here's another tulip glass sponge.
With another shrimp on top.
Mm.
The visibility is fantastic. Think of all these sponges, they feed on what's in the water column. So it's great that we can see so far. I think it was a feather star moving to the right.
Wow, lots of tulip sponges here.
I'll just zoom. What about this one on top?
Mm-hmm.
No.
Mm. Here it's maybe easier to sample because it seems you can take a rock. They're
Probably
detached.
will keep
But do you see
the sponge.
I'm trying to
healthier.
find
Yeah, of
something
course.
for. I'm trying to find something for solvent.
For solvent?
Mm.
So not. I thought the focus now is on your sponge that got away.
Yeah. But I'm also looking in case we find something
together.
Yeah. Mm-hmm. Oh wow, look at that.
And up we go.
Whoa. Beautiful. Wow, look at those
feather stars on the
feather
right.
stars on the crest of the rock. It's beautiful. Yeah, it's probably on the ridge of that rock that the current is the fastest, maybe, and brings more food.
Just as we mentioned, the good visibility, it starts to get a bit murky.
Mm.
And that's why I don't think the feather stars have an association with the sponges, because they're also on the rock.
Mm. Yeah, it's true. They're just trying to get higher in the water column. Mm.
Zoom in again.
possible to collect one of these two if you set it here? Or it's also a bit too...
Would grab it or
Yeah, to grab it, yeah. Because they're going to be also very light.
move the sextant in.
Oh, yes.
Dry lab is this one, right?
Dry lab ROV.
Right out here?
Yeah, we're now going to sort of try and collect one of these mushrooms, and so we're going to be sitting on the rock face, so we're going to pull the sextant in.
That's fine. Pulling the sextant, but never say mushroom above squatted.
Tulip. A tulip is better.
I think I've just been told off.
So we're going to try to collect one of these tulip sponges. We haven't collected any since the beginning of our expedition, Deep Arctic expedition. We think we know the species, Amphidiscella monai, but to my knowledge no one has really looked at the genetics. So this would be a fantastic opportunity to get a fresh specimen for genetics. Too steep.
It's too steep to land here. We'll just continue flying.
If I have to land here, so see where the tulip would be.
We'll find another one, and it's better.
What do you think, Rob?
Dry lab. Do you want the sextant out again?
No, we're setting now.
Oh, is that?
Yeah.
Okay, I thought it's just steep.
Just to see if we can.
Or these or the other one?
Yeah.
That one's probably doable.
Yeah?
First we will like to zoom in.
Yeah.
Photo shoot. And is it brittle?
No, but it is probably flexible and very fragile. So it's going to be very light like the previous one.
Are we good to go? Then lasers off, please.
And there should be a little stalk behind it.
Don't.
No.
We could bring the rock, maybe.
Yes, but it seems like a big rock.
Yeah.
Let's enjoy that view for a moment. What's this? Hanging out.
It's a sponge, but I don't know what's coming out.
That's what I meant.
I think I've seen this. I have no idea. It's like a sponge growing inside another sponge.
But they are on several. Look down here. There's also one hanging out.
Oh,
I
yeah.
first thought it's a worm, but it's another sponge.
I mean, it could be because I think that's one of the sponge producing mucus. Could it be mucus? It's a bit strange.
But it's on several. Look. We have three, but we don't quite know what it is.
Yeah. Do you want flashes?
No, thanks.
Thank you. Thank you. Yeah. Thank you.
Thank you.
Super pretty.
Everyone is seeing this.
Do you think this is one of your?
I don't know. It's so faint.
Mm.
It could well be. But
it could be.
It could be, but it's so light that I
Because it's
can't-
moving inside the tulip sponge.
Inside the tulip sponge is a critter, but we don't know what it is. But chances are high, yes. It is. Also, the eyes are on the right side, and it's moving in the direction.
Yeah.
And they move forward, so they always go in the direction of their head.
And this one?
Yep. Well spotted.
Mm-hmm.
And they're the similar size, so that's also another indication. So are you going for that one?
Yes. Yes.
Just checking.
Yeah.
Okay.
The other ones are almost impossible to connect.
Okay.
You ready?
I'm ready.
Ready when you are.
Yeah.
What's the cost?
13.
Thanks.
Bridge, can we come to an all stop there, please?
All stop.
That's it, yeah.
So since we're in sampling mode, we've asked the ship to stop and remain above us, above the robot. So normally just behind it, it should have a stalk. Ah, yeah. You see it. Perfect. Yes.
Yes, yes, yes.
And that should be rather solid. But it is thin.
Are we trying for the rock or the stem of the tulip?
Try for the rock, to be honest.
I think the rock is going to be too big.
Yeah.
Or I'm not sure it's even detached.
It's attached to the nuclei of the Earth. Maybe this side. This top.
Just tried that, but it won't stop me from there.
Yeah. Perfect. Perfect. What is more convenient for you, right or left?
Yeah, at the moment, right. It's the left one, yeah.
Left one. Okay, so that's going to be your upper left box.
The very first one to the left, yeah. Oh, is there the mug in there?
Yeah, there's a mug. Okay. But is the mug? Is there something?
Cup.
There shouldn't be anything in the mug
Just a
yet.
cup.
I don't think.
The coffee mug.
Yes.
No, but then
Put it on top.
Oh, right.
let's put it somewhere else
Can we
because
put it
we
on
don't
top
want to
of
mix
this
it
one?
with the sediments.
No, because if you...
Can we put it this one?
Or this one, because they're. I mean,
Any of the other
yeah,
two
any
on
of
the
these
right
two
side
would be
will be
good.
fine.
Okay.
Thank you. Just right mug.
Yeah.
We're talking about our coffee mug here. That's another
Can you
sampling
zoom
unit.
with your camera,
Yep.
please?
Yep.
Yep. Sorry.
Oh, I've got some other stuff with it as well.
Nice. That's fine.
Oh.
That. Oh, and lots of critters inside. Look at it.
Can you pan down any more?
Yeah.
That'll do nicely. That's big.
It is bigger than I thought.
Yeah.
Right, so I
I
have to
wouldn't mind turn it upside down. Yeah.
Yeah.
So they.
Then maybe it's better in that. Yeah,
Okay.
There's a bit more space.
Oh no, they kind of. Yes, put it. If it doesn't fit there, then go to the coffee mug one.
No, but I think it should fit in there.
Anyone is actually fine. Will be.
Look, you see it fits quite well. Actually, here it's a bit.
We have a different. Well, we have different cameras, so it's not as easy to see from the main one.
Yeah, perfect.
Yeah, you're doing really well. Perfect sampling.
Yes.
So quite. There, I would have liked to go in. It's like flower picking.
Ooh. Perfect.
And they were
perfect.
Thanks a lot.
So it's cast 13.
In 2,383 meters. Let's continue our flight. Thank you. We talked about our coffee mug. That is actually a sampling unit. It looks like a mug, and there's a net attached, but it's only suitable for soft bottom. When it's scoop up some sediment. Not here.
Yes, continue. Yep, continuing, yeah.
Some anemones. Zoom in a little bit on those. Oops. Here on our left, light pink.
Right here?
Yeah, just no, I'm just registering them, so just to say what I'm doing. And more feather stars. Shrimp.
So feather stars are the same group as sea stars, sea urchins, sea cucumbers. They all belong to the same group.
They fossilize really well. That's why it's possible to know.
The crinoids.
Yes.
Yeah. So sponges don't fossilize very well.
No, leave it around for pods. We have to give that point away.
Apart from one group of sponge called the rock sponges. They're super hard and they fossilize very well. And we can go way back in the Trias, Jurassic, Cretaceous, and we find lots of those rock sponges. So at the same times that dinosaurs ruled the terrestrial world, we can imagine that maybe sponges ruled one part of the underwater world during the Cretaceous and Jurassic.
Next to the feather stars.
Next to the feather stars. Probably some amphipods as well. No?
No. They took their time. Hello, Jenny.
Hello, guys.
Do you want to swap?
Oh, yes, please.
Okay. I'll swap with you now.
Yep. That sounds good.
Okay, cool. Let's go. Enjoy.
Thank you. How's it going?
Good.
Good. How many?
Jenny has joined us.
Hello.
Maybe you can say where you come from. Or Which institute?
Yes, sure. My name is Jenny Neuhaus and I am from the Senckenberg Society for Nature Research in Germany. And I am based in Hamburg. And I have a background in deep sea research on benthic invertebrates, which are small animals that live associated with the seafloor. And we are staring at them like that's all benthic animals that we're currently seeing, and I cannot get enough of this. It's just amazing. We've already seen so many feather stars today, and I don't know, they just amaze me. They're so beautiful.
I mean, this view is extraordinary.
Yes, it really is. But I have to be honest, I don't think I've ever seen that many feather stars in one patch before. I know this is not rare, but to my eyes it is, because...
No, it's really beautiful.
Yeah.
Could we have the lasers on back on, please? Thank you.
Would you mind stopping there for a sec?
Yeah.
I would really love to get a shot of that, of this little edge. May I?
Sure, absolutely.
Is that okay?
Yes. So we have the privilege in the Rough Shack that we can actually, the scientists can control the zoom and the direction of the camera. So we are the ones to blame if if you have zoom movements that are a bit too fast.
There are two species
No,
of
I see them.
jellyfish in the picture.
No, I see them.
Yes, and you see how different, they're really different.
Only 4 tentacles.
Oh,
that
there's
are the
another one. There's this third species maybe. Oh, okay, that one.
But could it be
quickly.
ctenophores? They were a bit too fast.
Yeah, they're very fast floaters. Little dance, thank you. Okay, now I wanted to zoom in on the feather stars again, because I just think they are so incredibly beautiful. Especially along this edge.
Mm-hmm.
Wow.
Great, Jordi. Can we continue the move, please?
Can you move 310 at point 1?
Thanks, Baz.
Amazing. Thank you for the stop. We may continue. So currently we have Andrea and Rob as pilots, as ROV pilots. And they're doing an amazing job flying the ROV in this environment.
Look at the background.
Wow.
It's just cliff after cliff.
Yes.
And all of them absolutely covered with life.
Yeah. Really amazing.
So I hope you will never think again that the deep sea is empty.
Yeah.
It's your choice if you want to go to the next rock or to the further one.
So either here or either there? Is that what you're saying?
Yeah.
I'd say port side.
Okay. Yeah.
Good.
Let's check out what's up. I've already logged the fish.
Other than change of that. Yeah.
What are we looking at?
Well, he's looking at us.
Hey, buddy.
Not easy to follow.
No. Okay. We've got a little glimpse. Actually, where's our species guide? Because I personally don't think I have seen that one. Now it's gone.
Yeah.
Alrighty. I have logged eel pout, but that could be wrong. In fact, you know, because I think it really resembled the sea snail, like a bit.
Mm-hmm.
Paraliparis.
Yeah, maybe it was this one.
Not sure. I haven't worked with fish ever, so it's.
Yeah, me neither.
Yeah.
That's a very steep wall.
Amazing.
Completely vertical.
You can look in my camera.
That's the tulip again.
Yeah.
That's a different one from the lollipop sponges, right?
Yeah.
So we call them tulip sponges. Okay.
Yeah.
So for those that might just recently have joined this live stream, we are logging the species that we see.
And we're a little bit on the outskirts of Mohns Treasure,
Yeah.
which is on the Arctic mid-Atlantic ridge, which is the border between two tectonic plates. The North American plate on one side and the Eurasian plate on the other side. This is where new oceanic crust is created, but very slowly. So this is what we call an ultra-slow ridge, meaning that only less than 2 centimeters of crust is created per year. So it's really not much. Not much volcanic activity here. The maximum speed you can find in the world will be in the Pacific, where it can be up to 14 centimeters per year that is created, meaning that the tectonic plates will move and will spread apart much more rapidly than here. Here it's ultra slow.
Right. And we're currently at 2,357 meter steps.
So all the rocks you see are volcanic rocks, because of course there were a lot of volcanoes in this area that produced a lot of magma, and when the magma solidifies, it becomes a volcanic rock. And all of these black rocks that you're seeing with us are volcanic rocks. What we generalize as basalt, usually.
Oh my God, look at this high density of crinoids and sponges. I mean, this is incredible.
Crinoid city.
Yeah, really. Oh wow, I will log that.
I no idea.
Oh.
The fish is following us.
So it's going to appear if you look down.
Yeah, there it is. There we go. Oh, beautiful. Yeah.
Yeah, like Jenny said earlier.
Oh, it bumped into a sponge. Oh.
He's blind.
Yeah.
Oh, little guy. But at least it was soft, right?
Yeah.
Ooh.
I would call this one the clown nose. I don't know why it makes me think of a nose of a clown.
Let's try if we
you know, we have some jelly experts that are really yeah, it's difficult. Sorry, guys.
It's challenging. Yeah.
There's
another one.
Oh, they're both of them.
And it's tumbling away.
And sorry. We'll try again. There's another one coming up. So this will be now a bit crazy camera movement here because...
And it's gone.
And it's gone again. Let's focus on the benthic sessile animals that are not moving away that quickly. I really can't get enough of this.
You know what we should keep our eyes open for? Storozoa. Because last time we saw them in this environment.
Yes, you're correct.
And we didn't collect, so...
Yeah, you're right. So there's this bell-shaped Staurozoa, they're called. It's cnidarians, like they're in the same group as jellyfish.
Yeah. Maybe we call them the stocked jellyfish.
Yeah.
Is that not a thing? I think so.
They are really pretty. They have these little pom-poms shaped.
Yeah, the tips, the tentacles. Yeah.
Exactly. It's hard to describe until you see one, and then it's really clear.
Yeah.
We can do a bit of zoom, like a gentle zoom to see if we spot anything.
So what change the ship's heading to 270 west? Yeah.
There's always this balance between zooming in and trying to spot. Oh, jellyfish! Look.
That's the other species you were talking about. Yeah.
What I want to say is that there's a balance between the zoom that we need to see smaller critters. Now there are plenty. And the movement of the ROV and the video in general that obviously increases the more we zoom. So but I haven't spotted any, even though when we zoomed in, I haven't seen any.
Me neither.
Okay. Let's give it another try here. For the jelly guys.
Oh wow! It's keeping its Oh, perfect.
Yeah.
Oh, that was but that was good. We had it in focus.
Okay.
For 2 seconds, and then you saw those 4 tentacles up in the air.
Yeah. Look, there's the fish again. Over there to the right. It's hiding. It's lying flat on the slope.
Oh, I thought that was a rock. Okay.
I think it's a fish.
Yeah.
It looks soft to me.
Yeah, clean surface. Yeah, it's the fish. It is.
Okay.
That's the one.
Having a little break. Maybe it's the one that got hit. Like hit its head on the sponge. Yeah, well.
Do you have the water temperature here given?
No, you guys have it there.
On the CTD.
We do have the water temperature.
Let's have a reading on that.
If I am not mistaken, we have a minus 0.65 degrees Celsius.
Yeah. So negative temperatures right now at 2,300 meters depth.
Oh.
Oh.
Hey.
Beautiful. Boom.
Oh. Yeah. They are obviously not used to the light. So they get a bit dizzy from it, but they will recover. It's just for a short period of time that we are illuminating them and their habitat quite intensely, but
yes,
that's just we can't avoid this.
So usually everything is dark here. The light and the sunlight cannot reach these depths. So all these animals constantly live in the dark.
Yeah. I have to say, of all dives we've done so far, since we arrived today, like since our shift started, this has been the most constant habitat, I would say.
Yeah, I mean we saw a very similar habitat on our previous seamount at the same depths.
Yeah.
It is very distinct and very. Oh, I think this is the one I wanted to.
Which one?
Do you think you could get it? It's this one.
Are we trying to sit?
Just see if we have.
Sure. So, this species is special to you?
Yeah, because I don't know the species. It's been reported. I've seen a couple of pictures, but it's not been identified to the species level. So I'm curious.
Try, I don't know. Which one is it?
It's possibly that one, but I.
There you go.
It's this one. So it must be. Yeah, it must. No.
It's to the left of the rock. It's that one.
That.
Yeah.
Bridge RV, can we pause, please?
I'll stop.
I'll just zoom out a bit so we see what's happening, so we can orientate. There's another specimen.
Are you
Oh yeah.
In case we don't catch that one, we might get
Are you
that
going to
one.
grab or slurp? Are you going to grab or slurp?
Slurp, no, it will be destroyed.
How about the scoop?
Scoop we tried, but they are so light that you scoop them super well and then they fly away.
The thrush just pulls them.
Yeah.
So the sponge has about 20 centimeters in diameter, so what we see on the video often looks very large to us. But when the specimens get on deck and we have them in our buckets, we first see the real size of them. It's sometimes really hard to imagine how big they actually are. We might be able to just grab the stone.
Yeah, maybe grabbing the stone could be an option. I don't know if it's sitting on.
If it's
see where you go.
Yeah.
Let's see. Let's have a still. Just give us a minute, Rob. We'll just have a little still.
I think that's a baby woolly sponge.
There's more room.
Can you tell which box where we have it?
Oh yeah.
Which side would you prefer?
We're going to go to start. I think it's more.
So this one, yeah, bottom right is empty.
Could you please turn off the lasers if you have the chance? Thank you.
Oh, and there's a little shrimp on it.
Yes, it's a nice still image of your specimen. And we've got what I believe are anemones. We have some other sponge species. Which cast is this? 14?
14.
All right. So whenever we collect animals, we give the event, the sampling event, a number, so we can retrieve the information we need for depth and location and time afterwards when they're processed and analyzed.
Marco, will this break if we hit the hole up?
It will break a little, but that's okay. If you cannot grab the rock, try to grab it from the bottom. It will break a little, but that's okay because the rest of the sponge will be.
When we go to let it go, then it doesn't fall off.
Yeah, that's
so we need it attached to something.
Ah, yeah.
I'm sorry, but I found a picnic on it. Look at it.
Oh yeah.
Okay, so when you look, you have the sponge, and to the left of the sponge, let's say at about 8 o'clock, there is a sea spider that is very well camouflaged on the rock, and it has just the same coloration as the debris or like the sediment that is on the rock, but it is a sea spider. And I think I'm maxed out on the zoom, but
And during our live stream, was it yesterday? It was yesterday, where you saw a lot of pycnogonids.
yes, we did.
So if you want to rewatch that pycnogonid action, you have to go back to our previous live stream, where we saw actually adults carrying their babies, which was pretty amazing.
Yeah, that was incredible. Shall we?
I'm ready.
Okay, me too.
Yeah. So.
Is it okay? If
Yeah, absolutely.
You're fine.
Yeah.
Okay.
The ROV needs for the sampling. We need 2 people. Andrea is maintaining the robot stable in the water column, while a pilot is using the arm to catch the sponge. So it's a 2-person's job, and it's very tricky, as you can imagine.
And then we have us steering the camera.
Yes. Jenny controlling the camera, and me being a bit nervous for the sponge.
Moral support all the time. And the decapod can leave now. It's alright. Escape while you can.
Ooh.
Yeah, it is very fragile.
I wasn't expecting that the rock would break so easily.
No, I wouldn't.
Wow.
Yeah, it is. It's very hard to predict sometimes what is going to happen when we just touch the sediments. When we just look, the decapod disappeared now. That's good. Bye.
Yeah, maybe this part is part of the whole rock.
That's sticking up, right?
Yeah. Because then it would be really.
It now. If it's too difficult, it's too difficult.
Just clear a little bit. Yeah. We're gonna clear a little bit.
Sure. I mean, this is basically like in the mountains, right? You touch one stone and they all keep rolling, and then we have these sediments in the water, so they create a lot of dust.
No? Uh-huh. So it's right. Okay, it didn't go too far. I think I see it. Yeah.
Just slid down a little. Now the little edge is gone. It was nice to grab it. Let's see if we manage. There we go.
of the whole robot. I want to do this. You know?
Yeah, exactly. Turn on the fan and just get it out of our way. Hi, buddy.
So again, we're collecting this species because we don't know what species it is, and without collecting it, we cannot give it a name. But once we've given it a name and we see it again, then we'll be able to name it directly on the movie, on our observation. So
without collecting it.
Without collecting it. So
yeah.
We need at least one individual.
Ah, it's clearing. Maybe not. It might be the clay lump that is in the claw still.
Mm.
So yeah, there's always lots of patience asked from everybody to retrieve these very, very precious samples. So you might now start to get an idea of how precious they are, given that we are at 2,341 meters depth and need, I don't know, lots of, yeah, energy, resources,
logistics,
skills, everything needs to fall into place to be able to collect single specimens from the seafloor, to explore their diversity.
Okay.
We're giving it another try. And you cannot see much at the moment, but the pilots have a bit of a better view on the specimen. It's a tricky one.
It's a very tricky one, yeah. You have to imagine we're on a cliff and we have this huge robot, 3 meters large, that's stabilized itself in the water column.
I mean,
yeah, I think you have it.
Oh my, we got it.
Yeah. This one is totally empty. The other ones have lots of sponges already.
one is good.
No. And I'm afraid to put it there because there's already apparently other sponges, it says.
It's all from the forwards, so we just fall back.
I'm not sure what's in there.
One thing off the ground, we just fail.
So basically what's happening is that
Really slowly.
these drawers have been
Or you could actually put
pushed
it there.
out
I think right there there's
in the
only
front.
stone.
And then they have 4 compartments, and
Yeah, but then do that.
we have to be very careful with where we put specimens to keep track on what we sampled in order to separate it again when it comes on deck.
Now we can't put the drawer out, so I shorter down.
B7.
It's actually going on here at the moment.
No, look.
Ah, I see. Okay. I didn't want to confuse you, I'm sorry.
It's okay.
Sorry, Papa.
Just turn your thrusters off. Go thrusters off. Try and get down a bit more pitch forward again.
At the minimum.
That's it.
Yes.
Ah, perfect.
Brilliant.
Thank you so much.
Just all the way and actually start working the jaw.
So that's going to be a very precious specimen. Thank you to the
forward thrust of the engine as it comes in.
to the robot drivers.
The arm's going to go on the
To our pilots. Yeah.
shoulder up more. As you start.
Thank you guys.
Yeah, thank you so much.
This is a trailer.
Yeah. Are the lasers back on? Thank you. We're ready whenever you are.
Just swirl the arm.
Good job, Rob. That was very challenging with the bad visibility. And we continue our journey along this volcanic mount.
Bruce RV, can we continue with the move, please?
Move 310.1.
On the chamber having anything on summer. Because I turned this side.
Yeah, come back on your
Leaning back there for a second. It's really. That was intense. I'm very nervous. Like because it's so precious and it's so easy to lose them in the current.
No, they're extremely precious specimens.
I think that deserves a piece of chocolate, actually.
Let's have a piece of chocolate to celebrate.
Yeah. There you go.
Thank you.
Anyone wants a piece of chocolate?
Oh, thanks.
So we're back on track, huh?
So I have another sponge expert that's watching us right now, and who's giving me advice on what these some of these sponges could be, because he's a glass sponge expert.
Oh.
Celso Domingos, if you hear me, thank you very much. You know him? No. He's from Brazil.
Nice to meet you, sir.
And he's doing his PhD on glass sponges from the Atlantic. So I always listen to his suggestions. He knows them better than me.
Did he have a guess already?
On several that we found.
Oh, wow.
Yeah.
That's really cool. I'm really eager to hear from our jellyfish experts about especially the red jellyfish that has been quite abundant in this area, but also the others.
Not going forward.
Just checking because sometimes messages roll in on species. Is that a... yeah. I thought maybe we had something else in the water column, but it is a decapod. Oh! Stay with us!
Always a pleasure.
Yeah.
A pinch of color.
Very pink. It's very nice. So, probably many of you know this already, but lots of species in the deep sea have reddish colors because red is the first color that disappears with depth. You can already see it at diving depth, basically, when you dive at 30 meters and you bring a color chart down with you of the rainbow colors, you will see that the red color just fades away. You can't really recognize it anymore, which is the perfect camouflage in the deep sea. And although there's complete darkness, there will always be some species that can produce light or even see in this darkness. And so you gotta hide, or you will be eaten.
What is your favorite animal?
Oh,
there.
In this particular
I think I know the
environment?
answer.
Yeah,
but
you
just
do. You do.
I'm just asking.
So it really depends on the habitat, but right here, right now, I have to go for the feather stars because they really amaze me.
Then I was not expecting that answer. Okay.
I am full of surprises. No, but yeah, here is the feather stars. But like yesterday, it definitely were the sea spiders. It really depends. I have lots of favorites. It's very hard to tell sometimes. I also really love sea stars, and I am a bit sad that I missed the sea star from this morning. Feasting on
they're not
feather
at all,
star. No,
not at all there.
They need more. The Hymenasta, the specimen that we observed, they need more soft bottom. They thrive more in soft sedimented plains rather than rocky substrates.
So that makes me think also that we still have the mug to use. We haven't used it.
Right, right.
So you tell us when you think it's a good place, a good stretch of mud we could sample.
Yeah, I'm not sure.
And we have the cores, the two cores.
Right.
Still.
We still need to do some push coring. That's right. Thank you for reminding me. That is quite a good point there.
And what about scaphopods? Have you found any here when you
No.
were sorting?
No.
Why is that? They don't live in the deep sea?
They
or?
do, they do. There are plenty of scaphopods in the sediments. Maybe even more than we think. But you might need different gear.
Okay.
You know, you might need gear that takes more of a larger sediment sample to sample them. Although there are sometimes species that live more on top of the sediment than inside, or they can crawl or move on top of the sediment. And if we would encounter this, that would be extremely special. I don't think we will. You need to gently deploy a sled on a flat sedimented plain, where you then take a very small fraction of the seafloor on deck, and when you then sort through that sediment,
that's when you find them.
Yes, that's when you find them.
Oh, look at that!
Oh, hey, oi, oh! It's like you see it and then it's already gone.
Maybe you can put it in the off-up. Fish.
Oh, yeah.
Fish to the camera.
Fish straight up. Still sponge grounds on, what do we call this? Say, yeah, let's keep the push course and the mugs in mind, but just maybe fly a bit first. Yeah.
We're still just at the beginning of the transect.
we should cover some track. It's a tulip again.
So the rough pilots are changing chairs. Of course, they cannot be driving and concentrating all the time. It's an intense job.
What's that? It was Kirognas, I think. It was moving like would be nice with a little cephalopod now, you know, once in a while. They could drop by like this morning. And this dive has been going on for many hours, and during the morning hours our team encountered a Dumbo octopus. That must have been a very special experience.
Yeah, I was sleeping, so I didn't see it. You were sleeping as well.
Yes.
Yeah.
There's always someone asleep on the ship. And there's always something happening on the ship too. Yeah. I would actually say that, I mean, there's still lots of sponges, and they still cover the ground. But I would say maybe we have a bit less than we had previously.
Yeah.
Oh no. Yeah.
Maybe because we have a bit more sediment as well. So sponges don't like, some sponges don't like the sediment. They prefer to settle on a hard substrate, such as rocks or other sponges. So we're pretty much the second people maybe to film this area. The first one came to the same locality, but a bit outside of it. So we are still the first humans to see this place. But thanks to the first people that came there, we knew it would be worth it. So we already knew it would be quite rich in sponges and so on. And that's why we picked that area as well. So every time an expedition comes to these places, our knowledge increases a little bit.
And it's very interesting because we have been diving now since the very early morning, something like 6 a.m. roughly.
Yeah.
And how many meters of the seafloor have we actually covered? Like maybe 1.5 kilometers, maybe.
Yeah.
So imagine that you need 10 hours to cover 1.5 kilometers.
So that's slower than if you needed to walk on that mountain. You would be much faster than us with a robot.
You could do this 3 times. Like it is, and of course we could go faster, but then we wouldn't see anything, and we wouldn't be able to identify anything. And speaking of which, I can actually zoom a bit in. The zoom is wonky again. You know, it's like sometimes it's zooming itself.
Are you raising this chair? Just back to the front version. Yeah. That's the front.
Yeah. Yeah. So again, patience.
Okay. Shoots forward.
Very few anemones now too. Like, and the crinoids are not that abundant anymore. Another jelly.
Oh, and this one is staying.
No. Are you kidding me? I was just about to, zoom in on him.
It saw you coming.
Or her. And gone it is. Gone it is. Askonema.
I will like that.
I could actually press the button, but I kind of like to type it because I memorize it more when I write the name more often.
Oh, it's just a shrimp.
So all the, maybe you would like to repeat that because I really love that name. The white sponges that are quite abundant here in the picture right now, what are they called?
So I call them the polar pompon sponges. But their scientific name is Lisodendorix complicata. One of the researchers on board, Sergi Taboada, is planning a population genomic study on this species because it's pretty widespread. You find it at different depths, so it's an ideal candidate for this type of study to see how the different seamounts along the ridge are connected and how are these also connected to further south in Iceland. So that's why we are collecting several specimens of that particular species, because when you do population studies you need a good number of specimens to represent, to have an idea of the diversity within a species.
Polar pompons. That is one of my favorite words for today. That's for sure. Either I haven't been in the shack when you mentioned this before, or I don't know. We've only called it Lysidendorix, but the polar pom-pom thing is new. That's so fun. And we have a beautiful Asconema.
Yeah.
And the crinoids are back, at least one. For those with trained eyes, I guess it could be spotted immediately. I will make a note of that. Now it's changing again. I have the feeling that now the rock is more. There's more rock.
There's more rock. That's the key, I think.
Yeah. Oh, beautiful tulip. It's not really push core area, is it?
Not really. Really on hard bottom here.
Yeah, the crinoids start to come back. Oi, what's this?
Oh.
The net?
Could we stop and watch this? This. We would like to do a close-up on this thing.
It's on you?
Yep. Here?
That one. What's that? The zoom is a bit. It's going. Okay. What is this? Oh, this is
Could these
pretty.
be eggs? You know?
It could.
Chains of eggs.
Could. I first thought it might be like some sponge because that could be possible.
No.
Wow, this is beautiful. The crinoid lifting its arm. Okay, so we have.
And I think all the knobs on the sides is Tentorium semisuberites, which doesn't look like that at all when we bring it to the surface. Of course, here they are fully developed, pumping, feeding in the water column. So all of these are Tentorium.
Right.
Here.
Beautiful. Amphipod.
Amphipod.
Yippee!
Yeah.
Let's just get it a bit back into the picture. The zoom is a bit doing its own thing again. It's a bit. Yeah. Yeah, it's.
Please turn it off.
Yeah.
Thank you. We can continue. Yeah.
I don't know. The interesting structure. Maybe some.
Okay, go Jenny.
You. Maybe someone knows. Eggs is always a good guess. It's.
Yeah. Okay, Bridge. We resume the move, please.
Okay, resume move.
But it's good we got this on tape.
Yeah.
Speaking of which, change tape in 5 minutes.
Yep.
That's good timing. And they want us to communicate via the comms to the dry lab that there's a tape change. You can just address it as dry lab.
You mean with the comment here? Okay.
Because they're not always listening to
to what we're
us.
saying.
Yeah. Okay, no,
Yeah.
it's fine. I don't know.
This thing you mean?
Yeah.
Yeah, it's a bit strange.
It is. Yeah, no, well, it's alright.
Oh, that's a big rock.
Yeah, it really is. Wow. So we are very slowly but steadily ascending actually. So now we are at 2,295 meter steps. I think when we started our shift we were at around 2,400, something like that.
Yeah, it's just like climbing a huge mountain.
Yeah.
Underwater, though.
Is that a sponge too?
I think it's Asconema. It's just a bit cleaner colored.
Oh! Hey guys! Oh! Oh, they will attach again somewhere.
Ah, but a lot of these big sponges. It's really good we collected one because now I'm.
Yeah.
I'm not sure we have the right identification for it. Maybe.
So you collected one of these large
tulip sponges.
Bell-shaped.
Yeah, we did.
Beautiful.
Look at that. The overhang.
Yes. Really beautiful. Wow.
So is this your first time in the Arctic Ocean?
With an ROV, yes. That's definitely yes. But I have been sampling in the Arctic before. It's 8 years ago. We were on board the Helmar Hansen research vessel north of Svalbard, and that was very special to see this high Arctic fauna.
Yeah, that's very high. That's probably higher than what we are now. We're at 73 degrees north.
Yeah, we were at, I think it was definitely 79 degrees.
Okay.
I think maybe one station was 80
Yeah.
degrees north.
Wow, that's very high up north.
Yeah, very much. Okay, we have a tape change in 1 minute.
Okay.
I mean, we could do it just now, couldn't we?
Yeah, okay, so 3, 2, 1, stop.
Rob, could you press the comms? Is that possible? Or
Dry Lab ROV, that's a tape change for the Kite Pro.
thank you.
So I did all that without moving.
Jelly.
Okay, I'm recording 3, 2, 1, recording.
All right.
Perfect.
There you go, Paco.
Thank you. I will write it down as well.
Yes.
So see time.
16, 17.
So we are of course recording everything that you are seeing today.
Just as this beautiful. You may continue. I'm just like.
Oh, are these stocked?
Yes.
Oops.
Osa.
There.
Yes.
They're really difficult to see.
Could we stop here for a bit, please? Because actually, this would be nice for a push core.
Yeah. Yeah.
What do you think, guys? Could we sit and do some push cores?
Not a chance.
Not a chance. Okay. Alrighty. No worries.
It's all rock.
Yeah. All good. All good.
Let's move off again.
Oh, wait.
No, let's have a zoom in on that rock.
Okay.
Do you think we could collect this? I think.
How? Also, can we pick it?
Yeah, pick it.
So maybe you can do that. Or you start with it.
Because I think that's a species we haven't collected yet, and it's a sponge for sure. And Celso also, following us, told me this was a glass sponge, and I've never seen it before. So here we have a nice opportunity.
Are you talking about the ball-shaped
No.
or the other one?
The other one.
Okay.
That looks a bit, I don't know what to say.
Yeah.
It has a brown ring, and in the middle it's grayish,
Okay.
whitish.
Okay. I see. How would you collect it? With the manipulator?
Yeah, with the manipulator, it's probably because, I mean, it's sitting directly on the rock.
Mm. Could we turn off the lasers for a second there, please?
Off.
Cheers. What do you guys think?
Only way would be to come in parallel to the rock and just pick it, but it will damage it.
Yeah, it will damage it a little bit, probably, and it's probably fragile, but
Alright, so start
I think this might be our best chance.
Can we slurp it?
We can try to slurp it. Okay.
Do we have any free?
Yeah, they're very free. So I know that I shouldn't put anything on top of the amphipods.
No.
This one is a crust on the rock, so here we could put maybe something, but in those 2 and 3 we have amphipods.
Exactly, so it could be 1.
1, 4, or 5.
Your call. But don't you think grabbing is better than slurping? Wouldn't it fall into...
I'm afraid it's super light, like the other, and that if we let it go just a little bit, it's just gonna... While with a slurp, it might be a bit damaged, but for sure everything will go in the box.
Okay, your call.
Yeah, let's try the
Chamber
slurp.
4, chamber 5.
Even 1. There's already there's a rock, but we can put this on top of it.
I think I've just found an Ascidian.
Do you want chocolate? What? Do you want chocolate?
Or a baby Asconet. Or actually maybe it's the same thing. Maybe this one is a bit bigger and has become transparent. I don't know.
It is.
Could this be
oh, sorry, it's not me. It's the Zoom.
Oh yeah.
Sorry, it's sometimes our Zoom has its own life.
Could you? I can't see the.
Yeah, sure.
Coordinates. You're a bit
No
closer
worries.
to the screen.
Yes, it's.
Yeah.
7, 10.
It's easy to get uphill. So we're reaching up high for this, guys. We just got to see if we can actually physically reach it.
Sure.
There you go.
And if you take the little ball next to it as well.
If we can, yeah.
If you can.
Am I free to operate?
Yes, please, go ahead. We might be too far away. I think we are. I think it's too far. So here comes the slurp.
So it's basically like a vacuum cleaner, and it will specifically
Zoom in there, guys, in HD.
Yeah. Is that alright?
Yeah.
We're a little bit far out, but yeah. Ready? On.
Almost. Yeah.
Yeah.
Amazing. Wow. So it's Craniella.
possibly. It could also be something else. So
Okay.
Craniella is usually a bit orange. This one is more white. So
Yeah, so
So we are currently collecting two different species together because they
on.
they grow together.
I think it's stuck against the rock. Yeah.
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 There we go.
There you go.
Perfect.
And off.
Well
Amazing.
done.
Thank you so much.
Fantastic.
It's
So this,
it's ecologically important to collect them together, or it could, because they were growing together. Very good.
good.
So I will request. Could you request the push course again? Maybe we can do a push core.
Do you think that if we do a push core here, there's not enough? No. Okay.
Okay.
Yeah.
Let's just take a time to zoom in on the tulip before we go, huh?
Sure.
Okay.
Beautiful.
It's zooming a bit. I need to turn off the zoom afterwards. It's a bit off. Yeah, it's actually. Yeah. Is that a sea urchin? No, it's also a sponge.
Yeah.
Rotate the shoulder. Yeah.
It's
very picturesque.
Yes, it is. It is.
Look at that with the little, even with the dust, I think it has a nice.
It's a sparkle.
Yeah. Nice touch.
Yeah. All right. I think we're good to go, aren't we?
Yeah.
After
absolutely.
one little shot here. Beautiful. We may turn the lasers back on.
On.
And we're good to go.
Yeah. Okay.
Yep.
Okay, Rob, you get going. I'll get the rest of that. Yeah, it's just the link. Okay, Bridge, resume the move, please. Resume the move.
Yeah. You must be right that was a baby Asconema. Because what Anna Helena mentioned was that we haven't seen any tunicates, any Ascidians during this dive, which is interesting because we had so many of them previously at other sites that we visited. So
especially in the bamboo coral.
They were everywhere.
Field, they were everywhere.
In fact, we sampled one very pretty specimen. Look. Yeah, I will just keep it steady now. Oh. Hi.
As if we had known.
Hello.
Where are we? You're in transit 3 still, right?
Yes.
Okay, so still early on.
We are snailing ourselves up.
Okay, good.
And we still have quite a lot of space in the boxes.
Oh, you do?
Yeah.
Okay.
So we can continue.
Depends on your plan.
Yeah, that's why we're here. We're trying to make a schedule now.
Yeah.
Or having a time set for ROV to come back on deck. And in theory, just a rough calculation that we did would be that you guys would have another 2.5 hours until 8 o'clock.
Okay. Sure. That sounds.
Although we said 8 o'clock, I think that would be.
No, it didn't. No.
So, to any
End
of
of-
our
no, end of
fish experts, what are we looking at?
Till
8?
Yeah, that would be.
Sure.
I'm sure Annie would let it push out then longer. Okay.
I mean, we can.
To prepare, once she comes
I
up
think I
on
will
deck
write
and you've had,
your
you've got the
parts
samples
still,
cleaned up and
but
all of that.
I am tempted to.
Comfortably think you may need. I know you won't know 4 or 5.
This picture is a bit
4 or
bad
5 hours.
because
So
we
if you
can't
had
really
6
tell.
hours minimum, then that would be pretty
Difficult.
Yeah, of course that's okay.
If anyone live has any ideas on the fish species, we appreciate
That
comments.
works out pretty much right because we've got to do